பசங்களா, welcome all of you to NEET Tamil Nadu. எப்படி இருக்கீங்க? நல்லா இருக்கீங்களா? So welcome all of you to reproduction unit மொத்தமா one shotல. That means we are going to complete sexual reproduction, flowering plants, human reproduction and reproductive health. So in the chapter weightage பார்க்கும்போது students, in the entire unit has at least 10 questions you can expect from this unit. So that means 40 marks in your NEET 2026 can be from this chapters. So அதனால இன்னைக்கு வந்து நம்ம full revision with the already handwritten notes, plus we are also going to solve questions as well. So are you all ready? Are you all ready? Please let me know if I'm audible, visible, everything is okay, because I will not know if everything is okay or not. Okay, so adinala quickly let me know in the chats if everything is audible, visible, everything is clear, and then we can directly get into the class. Okay. Hi students, hi, good evening, good evening. So most of you would have come back from your school and college. Okay, some of them might have half day, some of them might have holiday. So yes. So yes, it is going to be a little longer class. So I want all of you to be there, okay, till the end. Don't run away, because 40 marks does not come from, 2 hours of study. Okay, then everyone would have become a doctor. Right, so it is going to take time. Make sure that you all join and also share it with your friends right now itself. Okay, share it with your offline friends, online friends, every friends. Yes. Good evening, good evening. Everything is perfect. Let's start. Okay, so today we are going to start with sexual reproduction, then we'll go to human reproduction, then to reproductive health. Okay, mic is so tangled. Okay, mic is so tangled, just like our backlogs. Okay. Okay, no problem. Okay, students. So done? Okay, so let's start with sexual reproduction in flowering plants. So in the unit la vanda, how many questions you can expect? You can expect at least 10 questions, and 10 questions na 40 marks. Okay, 40 marks in NEET 2026. In the division vanda konja change agum students. Maybe from human reproduction you can expect 5 questions, and from sexual reproduction you can expect 4 questions or 3 questions and 2 questions from reproductive health. So in the madri or partition irukum. This is not like, this cannot be like the exact numbers. Numbers can change, but make sure that you're studying. Okay, make sure that you're studying. That is what all that matters. Okay, all that matters. Let me also just inform the Telegram students as well. Okay, yes ma'am, I will complete with you. Yes, that is the goal. That is the goal. Okay, 1 second. Yes, done. Okay, so yes, let's start. So students, sexual reproduction in flowering plants. Okay, so sexual reproduction is actually formation of gamete and fertilization happens. So what is sexual reproduction means? First you form the gametes, adukapram other fertilization will happen. Fertilization will lead to zygote formation or embryo formation, and adukapram we are going to ultimately reach somewhere, right? So here you can remember sexual reproduction na nama in the procedure illa sexual reproduction, why are we doing? And the procedure mainly students for variation. Ma'am, variation kaga da nama vanda sexual reproduction panna தேவை. Like what is the reason for it to happen? That is the reason for it to happen. Now, angiosperms, that is all flowering plants will do sexual reproduction. In the line one, the NCERT la irukku, so this can come as assertion reason or statement based question. Okay, or statement based question. So adanala make a note of it. All flowering plants can do sexual reproduction. Ipo namakku NCERT paakumbodhu students, look here. Okay, look at the NCERT. NCERT, it says all flowering plants show sexual reproduction. So this is a very, very important line of NCERT. All flowering points, okay, plants points, because you people are asking me. Okay, ma'am, please say whether we want to take notes. You can just write it in your NCERT that it is important, it is not important, like that you can write it. Okay, other than that you don't have to think a lot, because my notes will already be given. Okay, so you don't have to worry about the notes. My notes will already be given to all of you. Okay, notes will be given. So all you have to do is just make sure that you're watching and listening and understanding. Okay, you're listening and understanding. That is very, very important. Okay, now look here. This is one important question. Now, to a biologist, okay, to a biologist, flowers are morphological and embryological marvels. That is the site of sexual reproduction. So students, nama human reproduction la eppadi nama fallopian tube patti illena nama seminiferous tubule patti importance kudupe adde madri da nama vandu flowers also we should say that it is the site of sexual reproduction. So if a question comes, which is the site for sexual reproduction, your answer should be flowers are the site of sexual reproduction. So I will mainly focus on all the question point of view. I will also focus on the concept, and then we will be doing at the end of the, like after finishing one chapter, you will be solving at least 10-15 questions. So this way the entire revision will be very clean. Okay, that concept is understood, practice is done, then you will start the next chapter. Okay, clear? Okay. Question practice will be after the revision. Okay, question practice will be after the revision. Before that, you all should quickly smash the like button. Okay, attendance should be marked. Okay, attendance should be marked. Okay, now look here students, how flowers are formed. How are these flowers formed? In the flowers on the students, நம்ம shootல வந்து normally என்ன இருக்கும்? Ma'am, in the shoot there will be SAM. Who is SAM students? Shoot apical meristem. Normally ஒரு plantல வந்து இந்த plant students இங்க வந்து apical meristem இருக்கும், intercalary meristem இருக்கும், இங்க வந்து lateral meristem இருக்கும். இந்த meristemனா என்ன? இந்த meristemனா actively dividing cells. meristemனா என்ன? Actively dividing cells. Okay. இந்த actively dividing cells வந்து என்ன form பண்ணும் students? They will form the leaves. But when the plant has decided, okay, நம்ம வந்து flower produce பண்ணணும், அந்த processல what is going to happen? In the shoot apical meristem வந்து slowly it will start. Okay, hormones will be released. That is internal cues. That means from the flower inside, okay, let us internally the flower will tell that bro, we are ready to make a flower. You start making hormones according to it. நம்ம வந்து change ஆகணும், நம்ம வந்து morphologically change ஆகணும். So அதனால நீ வந்து signal like give the signal, அதுக்கப்புறம் நம்ம வந்து next process continue பண்ணலாம். So hormones are going to be released, which are the internal cues. Now students, this will lead to the formation of something called as the floral primordium, which will lead to the formation of what? Floral primordium. Okay, understood? This is the normal speed. Okay, normal speed. Ved, if you want to see YouTube lectures in Ved, then you have to download the app, and then in Ved itself you have to search YouTube. It is still loading students. By tonight I think most of the lectures will be there. I think my lectures are there from Vedantu NEET English. I think we need Tamil Nadu lectures are still not added. Okay, yes. Quickly subscribe and smash the like button. Okay, so இன்னைக்கு வந்து students here floral primordium will be formed. அதுக்கப்புறம் slowly you will start seeing the floral meristems. Floral meristems meristems are actively dividing cell. So floral meristems means these people will be ultimately going to form the flower, right? So slowly you will start seeing what? Floral meristems you will start seeing, and then students they will undergo differentiation and ultimately form the flower. So how is flower actually formed? How is flower actually formed? The SAM, again after all the hormonal changes, floral primordium will be formed, and the floral primordium, then later we have formed the development of floral meristem. அதுக்கப்புறம் differentiation க்கு அப்புறம் நம்ம வந்து flower formation நடக்கும். Okay, clear to all of you? Okay, clear? Meristems are actively dividing cells. Okay, what are meristems? Shoot apical meristem. Okay, shoot apical meristem. In the shoot apical meristem, and the meristem means what students? Actively dividing cells. Okay, actively dividing cells. Clear? Okay, yes, the energy should be there. Okay, chumma don't sit in the class. You should put fire emojis. If you're understanding, put thumbs up, put a lot of heart. Adu madri da in the class continue agum. If you people don't have energy, then I will not have energy. I will not have the motivation to do any more live sessions. So adanala I want all of you to have that constant motivation. Okay, clear? So here you can see students, now we are going to study how a flower looks. Okay, and here's, wait, I cannot see your chats, 1 minute. Wait. Yes, now I can see your chats. Yes, attendance has to come. Okay. Attendance has to come. Yes, okay. Ma'am, what is floral primordium? Floral primordium is nothing but like a initial stage, dear, before the actual flower formation, or a small, like a small bump, okay, like a small bump, it will be present, and this small bump is the ultimate where the flower will be formed. So, or initial stage, I can say. Okay, this is like a initial stage. Okay, initial stage. We say no precursor, adu madri da. Now, flower. Okay, this is a flower. So, look at the structure of flower, all of you. In the structure of flower, the students, we have a pedicel. The flower kudukumboda, that means if you have to give chintu mintu flower, you will hold the flower at the pedicel. Where will you hold the flower at? You will hold the flower at the pedicel. So, when you hold that flower, that portion is called as the pedicel. Clear? That portion is called as what? Portion is called as the pedicel. Then students, there is a swollen portion, and the swollen portion ke enna kupidua. Okay, it is going to be called the thalamus or the receptacle. Okay, thalamus or the receptacle. Adikapram students, here you can see two small leaf like structures which are not leaf, but then they're just structures like that which are called as your sepals. Okay, these sepals are actually when they are going to help students. Nama flower on the bud stage la irukumbodhu, you will notice that rose and all, have you seen? It will be usually like this. Okay, it will be usually like this. Only this portion will be red in color. Rest will be covered by green. So sepal da protect pandranga. Okay, va? Okay. So there is sepal. Adikapra students, just above that there is again a swollen portion which is called the ovary, and inside the ovary what will be there? Ovules will be there. From the ovules stretching there is one portion which is called the style, and style kapram there is a landing place which is called as what? The stigma, which is called as the stigma. So stigma, style, ovary and ovules together they form what, students? They will form the carpel or the pistil of the flower. They will form what? Carpel or the pistil of the flower. Okay, then look here. After that, you will see this orange color structures. No, what are these orange color structures? These are the filaments. These are what? They are the filaments. And after the filaments, here what you are seeing is what? This is the anther. Okay, this is the anther. This anther and filament together, when the enna form agum, students, they form the stamen. They form the stamen. Clear to all of you? Understood so much? Okay, so these are the major portions that you have to understand. Okay, so stigma style ovary is the carpel or the pistil. Adikapram anther and filament together forms the stamen. Okay, stamen. Then students, this colorful structure that you are seeing is going to be the petal. Colorful structure is going to be the petal. So, these are the parts of the flower. Just parts of the flower is not enough. You should know that the flower has 4 whorls. Flower has 4 whorls. Okay, 4 whorls. Just like imagine a box. Okay, or a gift box. And the gift box la the outer gift wrapping irukum. Inside, like inside that there will be a box. Inside there will be one more box. Okay, that means what? They are the 4 whorls of that. Okay, so adu madri da 4 whorls. First whorl one, the students, calyx. Calyx under calyx, who is going to make the unit? Who is going to make the unit? Sepals are going to make the unit. Okay. Adikapram corolla, which is going to be petals. Adikapram enna? Corolla, which is going to be the petals. So, yella whorls illame units present ayirukum. Okay, yella whorl me units present ayirukum. So, calyx la vanda sepal da. Corolla la vanda petal da. Adikapram androecium, stamen. Okay, in androecium, stamen is going to be multi-units. Okay, gynoecium, pistil or carpel. So, if I ask you, which are the essential parts of the flower? What will you tell? Now, all of you tell me in your life, which is the most essential thing? Or in your NEET preparation, what is the most essential thing? Quickly tell me in the chat box. Let me see your answers. In your life or in your NEET preparation, what is the essential thing that, oh my God, without this I cannot go forward? Adu your essential things. Just tell me 2 things. Come on. Quickly, let me see. I want interaction in the chat box. Otherwise, I will not do the live. Okay, I will not do the live. I will put recorded content. Okay, I will put recorded. You people watch it recorded. Okay. And don't forget, Tuesday, there's a big surprise coming for all of you. I think all of you are a part of day 3. Up to day 3 challenge is done. Day 4, day 5 challenge also is going to happen. So, adanale make sure that you are, joining on Tuesday. Okay. Yes, very good. Consistency and discipline, consistency and effort, consistency and NCERT. This is our life most essential thing. Ma'am, for my NEET preparation, I need consistency and NCERT. Okay? Okay, that's awesome. Hema Vashini. Okay? Your lectures, ma'am. Okay. So, notes PDF I'll give you after the session. Okay, at the end of the session the notes PDF will be in the description box. Okay, now when the students look here, essential part of the flower one, the Androecium and Gynoecium, the ones who have the ability to produce gametes, the ones who have the ability to produce gametes, because nama vandu sexual reproduction da we are studying about. So adanale sexual reproduction la vandu gametes patti da nama pesa porom. Full chapter is going to be about that. So and the gametes form pandra organs are going to be called the essential part. Nama life la essential parts vandu nama parents da, nama parents and nama siblings. Okay, non essential part na maybe every day-to-day things we do not tell this person, or every day-to-day things we do not want to see this person, but we want to see our parents and our siblings, right? But they are also going to help us in many things in our life, like our friends, colleagues, okay, or relatives, or our neighbors. Avrella vandu nama non-essential part. So non-essential means students corolla and calyx. They are not going to be directly involved in sexual reproduction, but they will help. Like example, corolla means petals. Petals vandu it is going to help in insect to get attracted. So and the help பண்ணுவே. Calyx vandu protection க்கு help பண்ணுவே. So அது மாதிரி it does not get directly involved in sexual reproduction, but it is helping us. So essential and non-essential parts. Okay, understood? Family, yes, family becomes the essential part. Now look here, events in reproduction. Nama reproduction event, that means entire chapter le 3 events are there. Okay, very important chapter, listen to me carefully. 3 events are only there, students. First is called as pre-fertilization events. Ipo NEET preparation le in the pre-stage la tha we are all there. Okay, we are all in the pre-stage. That means you are actually preparing for the examination. Okay, that is called your preparatory phase. So pre-fertilization phase la enna nadakum? Ma'am, formation of gametes nadakum, ma'am, because if fertilization has to happen, நம்ம வந்து gametes form பண்ணியே ஆகணும். So that is going to be called the pre-fertilization stage. அதுக்கப்புறம் then fertilization will come. So pre-fertilization stage la formation of gametes happens with microsporogenesis and megasporogenesis. எப்படி நடக்கும் students? Microsporogenesis, அதுக்கப்புறம் megasporogenesis. Microsporogenesis na நான் வந்து male gamete form பண்ண process தான். Okay, what am I forming? The male gamete I am forming. Okay, here I am forming the female gamete. Megasporogenesis, I am forming the female gamete. Okay, megasporogenesis and microsporogenesis. So these are the 2 things that is happening. Clear? Now look here, the fertilization. Okay, fertilization, students, it is going to happen with the help of what? Pollination. Yes or no? Pollination, self-pollination, cross-pollination, yeah, 𝐴𝐻 So this kind of pollination has to happen. Otherwise, how will the gamete meet together? So adukada water is there, insect is there, wind is there. So using these 3 mediums, we can do pollination. That means gametes, male and female gametes can come together. Okay, come together. Adukapram enna irukum students? Post-fertilization. Nama preparation ku appuram. Okay, in the fertilization stage only is your exam date. Okay, adukapram post-fertilization, where we wait for results, where we wait for, you know, government college, which government college will I get. So there only your post-fertilization events are coming, where we will form the seed, adukapram fruit. Is this clear to all of you? Understood so far? Understood so far? Okay, okay, good. Understood, right? Awesome. Okay, now students look here. Androecium. Androecium na enna, ma'am? See, androecium, students, androecium la vanda main what is going to be there? Ma'am, androecium, mainly your stamens are the units you told, right? I told stamens are the unit. So stamens la vanda enna irukum, students? Stamens la vanda 2 categories will be there. Enna irukum? Oru anther irukum and adukapram oru filament irukum. anther irukum, adukapra oru filament irukum. So this is what the stamen is going to have. Yes ma'am, correct. Okay, yes ma'am, correct. So students, all these notes will be available in the description box. After that, it will also be available on the Telegram channel. Okay, and you can also follow me on Instagram and Telegram with my name. Just search Gopika Biology, you will be able to find it. Okay, in next students, see, here you have your thalamus. Okay, this is your thalamus and this is your pedicel. Correct or not? Yes ma'am. Now you tell me, this box, okay, this box is so close here, so close to me. So can I call this proximal end? Yes ma'am, you can say that the box is at the proximal end. Okay, close by proximal. Now this is here. Now you tell me, is it at the proximal end or is it at the distal end? Quickly in the chats. Tell me. Is it at the proximal or the distal region? So we say no, in excretion chapter la vanda nama proximal convoluted tubule, distal convoluted tubule, edhukku appadi proximal na close ah irukum, distal na far ah irukum. So idu vanda proximal. Ipo idu proximal because it's very closer to me. Ana inge irukumbodhu idu distal ah irukum. So nama Androecium students, Androecium la nama filament and stamen to the thalamus, is it at the proximal end or at the distal end? One of the years PYQ has come. So here you can see, this is your thalamus. Thalamus proximal end. Okay, thalamus proximal end, you will have your filament. Yes or no? Yes ma'am, very correct. Now at the distal end, okay, very far, the distal end you will have the anther. At the distal end you will have the anther. Very good. Okay, now look here. When I take a cross section, okay, when I just remove the filament and anther, I will see the filament is there, which is a long and slender structure, and then you have anther. Students, when you look close to the anther, it just looks like this. That means it has two lobes. Correct, Rana? Just look at it. It has two lobes. This is one lobe, this is one lobe. Okay, this is one lobe, this is one lobe. So can I call this bilobed? Can I call this bilobed? Yes ma'am, you call it bilobed. Okay, can I call it bilobed? Yes ma'am, you can call it bilobed because here one lobe is there, here one lobe is there. Okay, va. So idu vandu students bilobed structure. Okay, bilobe. So in the anther vandu bilobed irukum because it has two lobes. Okay, ipo now vandu if I take that and if I take a cross section of it, so imagine two lobes are here, two lobes are here. And the cross section paakumbodhu idu madri irukuma students? Yes ma'am, it is going to look something like this. Correct, no? It will be like this if I take a cross section. Understood, right? So remember, in the attachment, students, see, this is the attachment. Okay, this is the attachment. This line which you are seeing here, this is called the line of dehiscence. Yenna ma'am, dehiscence na? Students, dehiscence means here if the anther is like this. Now imagine inside this pollen grains are there. If in the pollen grains release aganume, if the pollen grains are not released, then how will fertilization happen? Because pollen grain is the male gametophyte. Inside that the male gamete is there. So adanale, this is the line of dehiscence that will allow the anther to rupture and release the pollen grain. Okay, release the pollen grain. Got it? Yes, got it? Understood? People have to respond, yeah. Okay, people have to respond. Constantly, I'll keep asking you people to respond. Yeah, I used to be a student who used to always write and learn because now I'm a slow learner in the church. So, I used to write and I used to make notes. But I don't know where's all those notes. Okay. I have my master notes, like post-graduation notes I have. Okay. Okay. Now students, I am going to do this. Okay. Just look at this. This is going to be like this. No. Now imagine you do like this. It will look like a butterfly shape. Yes or no? Yes ma'am. It is going to. So just imagine like this, students. Like this. Okay. Like this. It will look something like this. Yes or no? Yes ma'am. It will look something like a butterfly shaped. Okay. So here I am going to take a cross section of the anther and try and understand. Okay, cross section of the anther, try and understand. Now look here, you have one lobe, two lobe, three lobe, fourth lobe. Okay, four lobes are there. So just look here, students, how many lobes are there? So I have taken a cross section, and in the cross section, I am able to see a structure like this. Now how many lobes can you see? 1, 2, 3, 4 lobes are there. Now we understood that the anther is bilobed. Correct, ma'am? But yenna ma'am, 𝐶𝐸? dithekas. Enna ma'am in the words? So students, imagine பண்ணிக்கோங்க students, நம்ம இந்த processல நம்ம வந்து ஒரு space, okay, we are building a space. So just imagine this is your microsporangia. Okay, I hope all of you are marking your attention. Students, marking your attendance. Okay, marking your attention. Okay. Oh, that is new. Anime fan. Sham. Should never ask all those. But I was a topper in biology. That much I know. I used to do really well in chemistry, biology and physics also. I think all of it, I think bio I had 90 plus. Chemistry, I think I had 86 or 85. And bio I had, no, physics I had 87, 86, something like that. Maths I used to not like. Okay, maths I used to be very weak in maths. Okay, so students, imagine இதுதான் ஒரு ஸ்பேஸ். இந்த ஸ்பேஸ்ல வந்து நம்ம ஒரு மைக்ரோஸ்பொராஞ்சியா we are keeping. எதுக்கு மேம் இந்த மைக்ரோஸ்பொராஞ்சியா மேம்? Students, inside the microsporangia only your microsporangia, microsporogenesis will happen, and அதுக்கப்புறம் நமக்கு வந்து pollen grains form ஆகும். So can I call this space? Can I call this space as theca? Okay, can I call that space as theca? Yes ma'am, you can call that space as theca because அங்க வந்து நம்ம ஒரு லோபுக்கு அப்புறம் inside there is a space, அந்த ஸ்பேஸுக்கு தான் theca. அதுக்கு what do you say inside? What do you say inside in Tamil? Okay, அதுக்கு உள்ளே, okay, அதுக்கு உள்ளே வந்து நம்ம microsporangium will be there. Okay, microsporangium will be there. Okay, you people told me you'll teach Tamil, now I'm learning it by myself. Okay, yeah, so look here, this is a theca. Okay, this is one theca. இதுதான் ஒரு ஸ்பேஸ், இது ஒரு ஸ்பேஸ். Okay, இது ஒரு ஸ்பேஸ், இது ஒரு ஸ்பேஸ். So இது எல்லாமே theca தான் students, and inside this what is going to be there? This structure which you are seeing is the microsporangium. Okay, it's going to be the microsporangium. So just look at the structure. We will go one by one. Don't worry. Okay, don't worry. Okay, yes, that's mean chemical coordination will come. So here we understood that this is bilobed, this is one. So look in my hand. Okay, look in my hand. Idu vandu one lobe, this is the two lobes. Okay, in the rendu lobume attach aganame students. To attach that we have something called as a connective. What do we have? Something called as a connective, and the terminology one that it is used for connection. So adanala connective, it's not very difficult to remember. Now I told you this is the theca, this is the theca. So in one lobe how many theca will be there? Okay, in one lobe how many theca will be there? Thank you Yashika Bhaskar, so sweet. Okay, yes, come on, who is going to be the class leader today? Someone should be there who will put the target today. Come on, let's see who is going to put the target today. நம்ம வந்து target achieve ஆகணும். Yes, very good. Ma'am, in one lobe. So students, இது வந்து இது ஒரு lobe. Okay, my hand. This is one lobe. இந்த lobeல இது ஒரு theca, இது ஒரு theca. So two spaces are there. So I can call it dithecus. Yes ma'am, you can call it dithecus. So now I understood two words. Anther is bilobed. அதுக்கப்புறம் I got to know anther is dithecus. Okay. Now look at how many microsporangiums do I have. How many microsporangia I have, students? Count and tell me. How many microsporangia I have? Ma'am, when I count, I'm seeing 1, 2, 3, 4. 4 microsporangia. So when 4 microsporangia is there, can I call it tetrasporangiate? Yes ma'am, tetra means 4. Sporangiums are there. So tetrasporangiate. Okay? Clear? No, Priya, in one lobe, how many theca? This is one lobe, this is one lobe. So in one lobe, 2 theca means dithecus. Totally 4 thecas are there. But in one lobe, only 2 thecas are there, no? So that is how the question is framed. Okay, wow. So now students, imagine the entire anther, okay, cross section of the anther, there are layers. Okay, there are protective layers. So how do I remember this? Elephant eats my tomato. Okay, so imagine I'm a farmer, I have a tomato cultivation, elephant eats my tomato. Now what do I mean by that? E for epidermis. Okay, E for epidermis. Okay, that is the outermost layer, single layer. Outermost layer, single layer. Clear? Outermost single layer. Then we have endothelium. Endothelium is the second layer, eats. Okay, that is again a single layer. Okay, adukapram students, we have the middle layer. In the middle layer, the 1 to 3 layer irukkalam. Okay, it can be 1 to 3 layers. So it is like a multi-layered structure. So here you can see multi-layers. I have put more pink color. Adukapram, one of the most important layers, which is called your tapetum. Most important layer, which is called your tapetum. Now understand this. This epidermis is going to line the entire thing. This endothecium is going to line the entire thing. But your middle layer is going to be specific for each of the microsporangia. Okay, each of the microsporangia. Clear? No, you cannot. You have to call it tetrasporangiate only. Now look here. This is the middle layer. This is the tapetum. And students, ma'am, what is right in the center, ma'am, and the blue color circle? Enna ma'am, paakadhukku nama frog egg madri irukku. But students, that is actually something called as a special type of cell. Okay, they are the special type of cell which are called as sporogenous tissue. They are compactly arranged, homogeneous tissues which are present right at the center, which are called as sporogenous tissue, which have so much of function. Okay, so much of function. Clear? No, it is 1 to 3. Okay, it is usually 2 layered. Okay, we can say 1 to 3. It will be more than 1, less than 3. Okay, clear? Now, look here all of you. Tapetum vanda students, mainly it helps for nourishment. It is binucleate and it releases 𝐘𝐨𝐻 enzyme. I want all of you to remember this and make a note in your NCERT. Tapetum vanda, it helps for nourishment. Nourishment na, ma'am, it will provide all the nourishment for the growing microspores or whatever. For nourishment, it will grow. Adhikapran students, this is binucleate. This question has come in one of the, I think, AIP-MT level, Ippa students before. Okay, before it has come. So here it is binucleate. I'll tell you why it is binucleate also. And remember that it releases 𝐘𝐨𝐻 enzyme. What is this 𝐘𝐨𝐻 enzyme, ma'am? My God, don't worry, you will understand in the flow. Now look here, epidermis, endothecium and middle layer is going to help for dehiscence and protection. Enna dehiscence in the, idu open pannumbodhu students, this is going to open, no, this is dehiscence. Now what will happen? Pollen grains will get released. Okay, so it is going to do dehiscence and it is also going to do protection. It is also going to do protection. Okay, done. Understood all of you? Can I see some thumbs up in the chat box? Okay. Yes. And who is going to be the class leader? No one is the class leader. Huh? Yes, Nagaraj is the class leader. Nagaraj is from Karnataka. Okay. Yes. So here you can see, now I have taken I have zoomed that. Okay. I have taken the microsporangia and the microsporangia enlarged structure. Okay, students, this is middle layer irukum, adhikapram tapetum irukum. Right at the center, they are going to be compactly packed tissues which are called as sporogenous tissues. And that sporogenous tissue is going to become what? It is going to form the pollen mother cell or the microspore mother cell. Majority of the time, the pollen mother cell we will call it, because PMC. Okay, we usually use the word PMC. We will not use the word MMC. Why? Because there also megaspore mother cell is there. So we will stick to the word microspore mother cell. Now we are going to learn, just like how we have oogenesis, spermatogenesis, same way here also we are going to study the process. Ana plants la vandu konja primitive irukku, so adanala vandu konja easy irukku, but oogenesis, spermatogenesis vandu konja complicated irukku. Okay va? So this is understood. So the sporogenous tissue has the ability to become, or it is potentially, it can become a pollen mother cell or a microspore mother cell. It can undergo meiotic division and it can continue the entire thing. Okay, clear? Yes, I will come to that, Priya. I'll come to that, don't worry. So look here, this is the layers. Okay, can you see? Connective is there, epidermis is there. Students, epidermis paakumbodhu enna irukku? Full layer ah irukku thane. See, like this, full it is covering. Adhikapram endothecium also you see full it's covering. But when you look at the middle layer, you will notice that the middle layer is like this, little specific to this person. And then you have tapetum, again very specific to the microspore. Okay. Now if I take a magnified image of it, again epidermis is a single layer, endothecium is a single layer, Adhikapram middle layer is there, microspore mother cell, tapetum. Okay, microspore mother cell which will be right at the center, Adhikapram tapetum is going to be there. Is this understood? Understood? Now shall we go to the main part, microsporogenesis. What is this microsporogenesis? Everyone talks about microsporogenesis, very easy concept. Okay, so students, look here, formation of microspores. from microspore mother cell or pollen mother cell through meiosis is called as microsporogenesis. Enna nadakudhu students? Formation of nama microspores from microspore mother cell or pollen mother cell is called as microsporogenesis. Okay va? Okay va? Now, yes. So here you can see PMC illana microspore mother cell. Adu vandu first meiosis undergo. First meiosis. It will undergo the first meiosis. Adukapram students, we are going to get microspore tetrad. What will we get? After the pollen mother cell or the microspore mother cell undergo meiosis, we are going to get microspore tetrad. Tetrad means 4. Correct? No. But can you see this tetrad is all attached with each other? They're like, I'm not going to leave you, I'm not going to leave you. Okay, like that, they're attached to each other. Nama vanda the break pannaname, because if we don't break, adu vanda eppadi pollen grains formation agum. So adanala nama vanda adu break pannana. Okay, we are very bad people. Okay, so we have to break it. Yes. So can you see this entire microspore tetrad is attached to each other, and it is like close to each other with the help of callose. Okay, who is keeping them together, students? Callose. So adanala da nama tapetum vanda it released 𝐘𝐻 enzyme. Why? Because it can degrade this and the microspores can be free. Microspores can be free. Now you can see microspore 1, microspore 2, microspore 3, microspore 4. Okay, microspore 4. Now all the microspores are separated? Yes, ma'am. Now each of them, students, they will undergo mitosis and they can form the pollen grains. Each of them, they can undergo mitosis and form the pollen grains. And when there is a sac of pollen grain, number one, adukku pollen sac, we call it pollen sac. Okay, clear? Don't worry, Nandini, you can watch me here. Okay, understood? Understood? Yes. So this is very important. Okay, very important. This so much so far understood, right? Okay, so PMC or the microspore mother cell is there that will undergo meiosis and it will form the tetrad, and this microspore tetrad are going to be attached together with the help of callose. That callose will be broken down with the enzyme that is called as 𝐘𝐻 enzyme, which is released by the tapetum. So tapetum vande enna jobs ella pannuvanga students? Nourishment. And if they see, always remember, if they are going to do nourishment, avarku vande cytoplasm vande dense ah irukum. Okay, dense cytoplasm irukum, lot of food reserve will be there because they have to do the nourishment. So when I teach you pollen grains also, similar thing you are going to come across, dense cytoplasm and food reserve. So that words you have to keep in mind that, okay, if I am going to do nourishment, if I am going to, let's say, provide food for someone, I should have enough food. So adu madri da. So nourishment, and this tapetum is going to do nourishment. It is binucleated. Why it is binucleated? Because here, students, remember, karyokinesis will happen, then cytokinesis will not happen. Karyokinesis na enna, and the nucleus division nadakum, but ana adukapram cell wall formation nadakada. So adanale ore cell la vanda two nucleus will be present. Okay, two nucleus will be present. Ma'am, if you don't mind, can you say that again? Which one, dear? Yes, tapetum has dense cytoplasm. Yes, yes. So here, this one. So the microspore tetrads will be together because of the accumulation or the deposition of your callose. Aduvanda it will be broken down by the 𝐘𝐻 enzyme, which is released by the tapetum. That will lead to the separation of your microspore tetrads. Students, what is this NN I have written? That NN that I have written is actually the ploidy level. Okay, this will be diploid. This will be diploid, but as the meiosis happen, because meiosis is one the reductional division. Okay, all of you know this, no? Next I'll take cell also. Oh, sorry. Okay, meiosis is a reductional division. So reductional division means you should remember that automatically the number is going to be by half. Okay, so idu vanda 2n na, idu vanda n. That means this is going to be all haploid. Okay, this is all going to be haploid because meiosis happened. Okay, now students, each of them will undergo mitosis and form a pollen grain. Okay, pollen grain. So students, a sac of pollen grain is called as a pollen sac. Imagine in this bottle. Okay, in this bottle, instead of water, I have juice. Imagine in this bottle, instead of water, I have juice. If I have juice inside this, I will call it a juice bottle. Okay, let's say if I have water inside it, I'll call it water bottle. So if the sac has pollen grains, what will you call it? Pollen sac. If the sac has microspore, what will you call it? Microsporangium. Okay, wow. Understood that basic thing? Understood that basic thing? Yes or no? Okay, understood, right? Yes, ma'am, understood. Why is the attendance not there, guys? Come on. Quickly mark your attendance and subscribe to the channel. Okay, we are going to be 11K very soon. I hope by the end of this class, we should be becoming 11K. Come on. Done? Okay? Yes. Understood, understood. Okay, next topic. See, now students, each of the microspore, okay, all are haploid, 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 haploid. They are going to undergo mitosis and form a beautiful pollen grain. Okay, they are going to form a pollen grain. But on the pollen grain formation, two other steps will happen, like vacuolization will happen, spindle fiber formation will happen. In the spindle fiber vacuolization, எல்லாமே வந்து நம்ம cell chapterல தான் இருக்கும். So அதனால there I will detail tell you, but understand this is a space. Vacuole is like a vacuum na என்ன ஒரு space, ஒரு void. அது மாதிரி தான் vacuole formation நடக்கும். அதுக்கப்புறம் spindle fiber formation நடக்கும். அதுக்கப்புறம் தான் நம்ம pollen grains formation will happen. Okay, pollen grain formation will happen. So just look at the pollen grain. It's a beautiful pollen grain. இப்போ இந்த pollen grainல வந்து important ஆன கொஞ்சம் layers இருக்கு. Okay, now look here students, all of you. Slowly, learn it slowly, aram se, try to understand. Okay, so first you will see one porcupine like layer. No, and the layer is called as the 𝐂𝐴𝐿. Okay, it is the outermost layer. It is called the 𝐂𝐴𝐿. Ana ma'am, and the 𝐂𝐿 la vandu nama space kanupe ma'am. That space is called as the germ pore. என்ன ஸ்டூடெண்ட்ஸ், அந்த ஸ்பேஸுக்கு நம்ம என்ன கூப்பிடுவா? germ pore னு கூப்பிடுவா. Okay, so here we have this 𝐂𝐴𝐿. This 𝐂𝐴𝐿 will have certain space which is called the germ pore. But எதுக்கு ma'am in the space? Wait, I'll tell you. Students, this 𝐂𝐴𝐿 is made up of sporopollenin. 𝐂𝐴𝐿 is made up of who? Sporopollenin. But in the sporopollenin has a problem. Sporopollenin has a problem. Enna ma'am, and the problem? Sporopollenin is one of the hardest, most resistant substance to be known. அது வந்து it will not get degraded by acid, alkali or temperature or any enzyme. So அதனால imagine one outer layer is so strong. Do you think pollen tube formation will happen? No ma'am, pollen tube formation will not happen. So அதனால தான் students in the space இருக்கு. Just look at how beautiful nature is. How beautiful, how thought process nature has. So this is the 𝐂𝐴𝐿. Okay, this is the 𝐂𝐴𝐿, and this 𝐂𝐴𝐿 is made up of sporopollenin, and there are spaces in between which are called the germ pore. Is this understood? Now ma'am, what is this inner layer? So students, the inner layer is going to be intine. This layer which you are seeing here is the intine, which is made up of cellulose and pectin. Which is made up of what? Cellulose and pectin. So 𝐂𝐴𝐿 is made up of sporopollenin, intine is made up of cellulose and pectin. PYQ has come, so make sure that you remember this. Now here you can see the nucleus. Here also can you see dense cytoplasm? Dense cytoplasm, and here you can see one cell which is floating in the cytoplasm of this cell. So two cells are there students in the pollen grain. One cell is called the vegetative cell, which is this large cell. Okay, this is called the vegetative cell, and then we have a small cell which is called the generative cell. Ma'am, but where is the male gamete, ma'am? Yes, this generative cell will have the male gamete. Okay, generative cell will have the male gamete. Remember, this generative cell will float in the cytoplasm of the vegetative cell. Because in the vegetative cell, it is going to provide nourishment for the growing male gamete. Okay, so adanala in the vegetative cell la dense cytoplasm irukum, food reserve irukum, so as to provide the generative cell. Is this clear? Is this clear to all of you? And students, the nucleus on the vegetative cell will be round. Here it is going to be more of spindle shaped structure. Spindle shaped na enna? Idu maadhiri sharp. Okay, idu maadhiri irukum. Spindle shaped structure. Okay. No, the layer, the portion where sporopollenin is absent, that is what is called the germ layer. Okay, sorry, germ pore. Okay, germ pore. Clear? Understood? So vegetative cell will provide all the nourishment to the generative cell. Generative cell will be floating in the cytoplasm of the vegetative cell. In the generative cell will have the male gamete. So in the pollen grain the students, I want you to understand, in the pollen grain, okay, yeah, this is the answer. Pollen grain is actually the male gametophyte. Pollen grain is what? The male gametophyte. Okay? Pollen grain is the male gametophyte, not the male gamete. Like example, imagine, this is the pollen grain. Okay, this is bottle is the pollen grain. In the bottle, I keep forgetting the word inside. Okay. So inside this bottle, what is there? Water is there. So imagine inside this bottle, male gamete is there. So this becomes the male gametophyte, and inside this, the male gamete is going to be present. So students, pollen grain is the male gametophyte, not the male gamete. Okay, inside this, there will be the male gamete, that is inside the generative cell. Clear? Okay, understood? Understood this difference? Many students don't know this. Blindly you people will study and then you will realize, oh my god, this much is there. Ulle, ha, inside ulle. I know the word, but ana, and the word vandu ஞாபகம் வெச்சிருக்கிறது ரொம்ப கஷ்டம். Okay, yes, come on. Now, why are tapetal cells binucleated? Why are tapetal cells binucleated? I told you students, tapetal cells வந்து first karyokinesis நடக்கும். Karyokinesis na enna? Nucleus will get divided. ஆனா அதுக்கப்புறம் cell wall formation நடக்கணுமே, அது நடக்காது. So அதனால from karyokinesis nucleus will divide and two nucleus will be formed, that is binucleated, leading to the tapetal cells becoming binucleated. Okay, and students, after that cytokinesis will not happen. Because of them, the tapetum will stay binucleated. Is that clear to all of you? This question is there, NCERT in-text question this is. Okay, that's why I'm told you have to watch this lecture, everything will be there. Okay, everything will be there. You don't have to watch 10 lectures for in-text, 10 lectures for detailed revision. Okay, NCERT in-text question it is. Understood this? Okay, just look here, whatever we studied so far, it's underlined in the NCERT. So here students, look here. So epidermis, endothelium, middle layer and tapetum are the four wall layers. Okay, outer three layers perform the function of protection and help in dehiscence of the anther and release of the pollen grain. Innermost wall layer is called tapetum. It nourishes the pollen grains and generally is having more than one nucleus. That's why it's binucleated. Can you think of how tapetal cells becomes binucleated? In the answer one, because it undergoes karyokinesis, does not follow up by cytokinesis. When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue will be present right at the center. And the cells one, it will undergo meiotic division and it will form the, sporogenous tissue undergo meiotic division and form the microspore tetrad, or we should say other words, pollen mother cell will undergo division and it will form microspore tetrads. Clear? Oh, target achieved. Very good. Now, look here. As each cell of the sporogenous tissue is capable of giving rise to microspore tetrad, each one is a potential pollen or a microspore mother cell. The process of formation of your microspore tetrad with the help of your microspore or pollen mother cell through meiosis is called as microsporogenesis. The microspores, as they are formed, are arranged in clusters. See? And the cluster word is important, because in the cluster word that callose is not allowing them to leave each other. That is when tapetum will release 𝐘𝐻 enzyme and 𝐘𝐻 will be like, bro, move that side. Okay, in the complete a break pannanam. So under release this cluster word is used. Okay, cluster of 4 cells, the microspore tetrad. As the anther mature and dehydrates, the microspore will dissociate from each other and they will develop the pollen grains. And students, here what will happen? Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. So that's why I told you, inside this, if microspores are there, I will call this a microsporangium. Inside this, if pollen grains are there, this is the pollen sac agum. And inside this male gamete irundhuchana, this is the male gametophyte agum. Okay va? Okay, understood? Yes or no? Okay, your energy is too less. Okay, energy is too less. I want more energy. I have so much energy, you should also have more energy. But yes ma'am, yes, we are going to do it. Okay, Okay, now look here. This is what we understood right now, that the pollen grains have an exine layer which is made up of sporopollen, which is one of the most resistant organic matter. In the resistivity, one the students is so high that acid, alkali, temperature, enzyme, whatever you put, nothing is going to get affected. Okay, no enzyme can degrade sporopollen. Why? First of all, you just think of it. Ma'am, why sporopollen should be there? Yes, awesome energy. Nagaraj, Deepika, Joshua, awesome. So, on the energy I want to see throughout the class. Okay, new students also don't hesitate. Be active in the class, because understand it's a live session. It will be like I'm talking to the camera. I want you people to feel that, okay, ma'am is not separated by a screen. I should feel that energy should come to you. Okay, because if it is a long marathon, I want that energy from you to come to me. So, look here. And that no enzyme degrades the sporopollen. First of all, why should we have sporopollen? Why such a hard substance should be there? Because friends, in the pollen grains, when the transfer agum, when these pollen grains are getting transported, there are so many chances that the pollen grains will not reach the anywhere. It would have, reached halfway. It would have gone into water, temperature, many things it can fall into. So, on the yella condition mein, other Taranam panida pananam. So, adinalada sporopollen is one of the most resistant substance. Okay, because of which pollen grain will be able to reach to the different locations. Clear? Understood? Awesome. Awesome. Yes, Vaishnavi. Yes. Okay. So, look here, students. Where the sporopollen is absent, that place is called the germ pore. Okay. The inner wall of the pollen grain is called intine, which is made up of cellulose and pectin. Everything what is important, I have already drawn the diagram. Okay. I have already drawn the diagram. Okay. Yes, it is haploid. Okay, now look here. The cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it will contain two cells, one vegetative cell, one generative cell. The vegetative cell is bigger, abundant with food reserve, irregular shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It has a spindle shaped structure with dense cytoplasm. Okay, and a nucleus. Here students, in over, see look here, in over 60% of the angiosperm, okay, in over 60% of the angiosperm, enna irukum? One vegetative cell will be there, one generative cell will be there. That means basically in 60% of the angiosperm, the entire thing is going to be two-celled. Right, the entire thing is going to be two-celled. That means one vegetative cell, one generative cell. And what about the rest, ma'am? 60% is two cell. That means one vegetative cell is there, one generative cell is there. What about the others? Right. So, students, and the rest of them is going to be three-celled. Ma'am, in the three-celled, students, one vegetative cell, two generative cell. Two generative cell? Yes. Generative cell on the halfway will decide that, okay, I want to do one more mitotic division, and then we will get two generative cells. So, here you can see two-celled stage is for over 60% of the angiosperm. In remaining species, the generative cell will divide mitotically to give rise to two male gametes before the pollen grains are shed. So, that means basically instead of two, GC, we can just write GC that will divide mitotically and it will form two male gametes. Okay, it will form two male gametes. Okay, understood this? Yes, this number is important. 60%, 40%, 20%, everything is very important. Okay, now look here. How many mitosis and meiosis in angiosperm male gamete development? How many mitosis and meiosis has to happen in a angiosperm male gamete development? Just look here and tell me. So, students, very simple. In a two-celled stage, number two-celled angiosperm, 60% angiosperm or more than 60% it is going to be two-cell stage. So, adanale or pollen grain or pollen mother cell it will undergo meiosis, form a tetrad. Each of them will undergo mitosis and form a pollen grain. So, basically, one mitosis and one meiosis. So, one meiosis and one mitosis will happen in a two-celled stage. Correct? Okay? Correct? Clear? Clear? Yes? Okay. Now look here. Three-celled stage. Okay. Three-celled stage, students. 40% of the angiosperm. Three-cell stage. Rest of the 40% of the angiosperm. One pollen mother cell will be there. It will undergo meiosis and it will form microspore tetrad. Yes, ma'am, it will form microspore tetrad. Each microspore will undergo mitosis. Yes, ma'am, it will form the pollen grain. Ana, one of the generative cell will be like, no bro, I don't want to stop here. எனக்கு வந்து இன்னொரு mitosis பண்ணணும். So அதனால one more mitosis it will do, leading to the formation of 2 male gametes. So here what is going to happen? It is going to be 1 meiosis and 2 mitosis. Okay, I want all of you to take a screenshot or write it in your NCERT because it's very, very important. It's very, very important. So please make sure that you write it. Okay, this question will come. So if such kind of questions come, it will be beneficial for you. Okay, because you already studied. Understood? All of you? Yes, ma'am. No, ma'am. Everything understood, ma'am? Okay, let me just see your attendance. Okay, a lot of you are there, but only very few are responding. Okay, don't be shy. Come on. All of you respond. Okay, respond and subscribe. Okay, yes. Come on. Okay, this is clear. Awesome. Now, students, look here. Pollen grain allergy. So, due to pollen dust, okay, due to pollen dust, you can have sneezing. Okay. Okay, okay, so due to pollen dust, many of them will have pollen dust. So please make sure that if you are giving flowers to someone, you should know that if that person has pollen allergy or not. Okay, you will dance and go telling that, oh, I will give flower today, and when you go and give the flower to that person will sneeze on your face. So and the madri story katam. Okay, so adhanala students, you should ask if people have pollen dust. Namakku vanda food allergy irukum illa, walnut allergy, peanut allergy, illana some wheat, atta, maida, adu madri lot of allergies will be there. So adu madri da one allergy is called as pollen dust allergy. That means once the person inhales the pollen grains, they will start having sneezing. So sneezing can happen, bronchial issues can happen. In extreme cases, rashes can happen. Rashes is actually an indication of allergy. Okay, normally if I come also, like rashes are an indication of allergy. Okay. Yes, don't worry Pratap, you can still continue. Okay, now students, example here they have given is Parthenium or carrot grass. Okay, example, this is how it will look like. I think all of you would have seen. Nama vanda, let's say if you're walking near an empty site, okay, empty site. Bangalore la vanda empty site romba kadumeya irukku, like it's very less, but ana in Tamil Nadu side and all of that, you would have seen lot of empty sites. And the empty site la ellame students, you will start seeing Parthenium or carrot grass. Focus, notice properly, you will start seeing that white flower. It has a different kind of smell as well. Okay, it has a different kind of smell as well. That is the example there. Understood? Next topic is viability. So far everything is good, no? Microsporogenesis you understood, pollen grains you understood, okay? Pollen grains you have understood, and then now we are going to the next topic, viability. I want to see all of your green signal. Okay, quickly give me a green signal that everything is super duper okay, ma'am. Yes, come on, fast, quickly. Next, viability we are going to understand. Okay, will be... No, bro. Your name is bro only, yeah? Okay. Okay, let's go. So students, yenna ma'am in the viability. Normally students, nama vanda Swiggy la Zomato la food order pannume. How many of you order food? Tell me, how many of you order food? Raise your hands. Ma'am, I order food, ma'am. So I order food. Okay, when I order food, I have noticed that Swiggy sends me or Zomato sends me an OTP. One-time passcode. Okay, and the one-time password la oru number irukku. Okay, 5431. Okay, oru number irukku. That number, within that 5 minutes, if I don't enter, and the OTP வந்து invalid ஆயிருக்கும். நம்ம bank proceduresல வந்து students அந்த OTP வந்து only 1 minute validity will be there. பட் ஆனால் நம்ம ஸ்விக்கி ஜொமேட்டோ ஷாப்பிங் மிந்த்ரா அது இதுலலாம் 5 minutes 10 minutes வந்து லாங்காக தான் இருக்கும். Now just like OTP and the time limit இருக்குமே, that expiry date will be there, no, that expiry time will be there. அதே மாதிரி தான் நம்ம pollen grains, let's say it has been released from the anther. Anther has got dehydrated, dehiscence has happened. If dehiscence has happened, pollen grain வந்து release ஆகுமே. Yes Fatima, this is live. When that pollen grains are released students, within a certain time period, the pollen grain should land on the stigma. இல்லைனா அந்த pollen grains வந்து it is non-viable. It is not going to germinate. It is not going to do fertilization. So, அந்த மாதிரி ஒரு expiry date இருக்கும். So, look here. Viability means students, in it now see, OTP can also depend on our internet speed, okay, our service, everything, right? So, adu madri tha, here you should remember, in the viability one, there are two external factors that will depend on it. One is called as humidity, next one is called as temperature. Based on temperature and humidity, nama viability vanda change agum. So, viability concept is clear to all of you. Once the pollen grain has been released from the anther, then it has to go and fall on the stigma and fertilization should happen in a certain time period for certain species. Example vanda, nama cereals illa, nama and cereal illa students, okay, here this cereal, okay, and the cereals la rice and wheat, okay, rice and wheat, clear? So, rice and wheat vanda students 30 minutes da viability, okay, thank you SSC, okay, viability. Okay, correct? Now, families, some families illa vanda students, rosole, families la, like rosole. Remember like this trick, okay? Rosole. Rosole na, rosaceae, so for solanaceae, le for leguminaceae. In the families la, viability will be for few months or for months. Okay? Yes, Deepak. More target we should achieve. Come on. So, viability depends on external factors like humidity, temperature. Cereals like rice and wheat will take 30 minutes. Viability and the time period வந்து 30 minutes இருக்கும். Families like rosole and the viability வந்து for months இருக்கும். But Anna ma'am, I am a farmer ma'am. I want to do I want to do the fertilization. I want to take the pollen grains. Just like how blood bank is there, seed bank is there. I want pollen bank also. Then I cannot tell no because you are asking. So, அதனால நம்ம வந்து என்ன பண்ணணும்? We have to preserve the pollen grains in minus 196 degrees Celsius in liquid nitrogen. We have to do what students? We have to preserve the pollen grain in minus 196 degrees Celsius in liquid nitrogen so as to, you know, conserve it, to preserve it, okay, to preserve it. Clear? Understood this much? This is your viability concept. Now here I am giving you an NCERT sniff that talks about the entire haploid diploid situation. Because students ikka usual ana confusion the haploid and diploid. Adanala this will help you. Look here. Haploid structures means these are formed after meiosis. Okay. That contain only one set of chromosome like microspore. Produced by meiosis, so it will be haploid. Pollen grain. Then vegetative cell, generative cell, gamete. Diploid structures, pollen mother cell, sporogenous tissue, walls of the anther. So, all this will be diploid. Clear? Understood? Shanti, you can check out my video. Thank you, Shailaja. Okay, you can check out my video. Telegram la illa Deepak, description box la notes irukum. Adukapram I will make a sheet and I will put all the notes together. Okay, description box la notes irukum. Sanjay, day 4 question வந்து 7:30 க்கு upload ஆகும். Okay, every day 7:30 challenge will be uploaded. Okay, every day 7:30. I love all of your energy. You people are asking me every day, ma'am, when is the question, ma'am? Okay, yesterday also I saw in between the class, many students were like, ma'am, when is day 3 coming? So, most of you are, you know, waiting for it. That is awesome. Okay, awesome. And I hope all of you get lot of gifts on, you know, like 7th. So, one set of team will be there. They will be selecting the students who are going to get some free gifts. Okay, so, adanala make sure that you come on Tuesday sharp at 7 p.m. Okay, va? Clear? Yes. Okay, done. Very good. So this is understood? Understood, ma'am. Now, look here students, whatever we studied so far, it's in the NCERT, okay? That is, pollen grains have land on the stigma before they lose their viability. They have to bring about fertilization. I told you the same thing, number OTP, normala, you should think that, okay? That is students, they will have viability, highly variable. That is depends on temperature and humidity. Viability for 30 minutes for cereals, Rosaceae, Leguminaceae, Solanaceae family for months. And if you want to preserve it, you have to preserve it in liquid nitrogen in minus 196 degrees Celsius, just like how you have seed bank, blood bank, everything like that. Okay, seed bank, blood bank, everything like that. Okay, understood? Yes. This AC, no, it is like it has mood swings. Okay, it has mood swings. Now it will be happy. Temperature on the correct set pannanga. Adukapram oru 1 hour 45 minutes adukapram he'll feel mood swings. He'll be like, what is this teacher telling? From that time she is, eating my ears. So adanala temperature automatically change agum. Okay, now whom do I complain, students? You people tell me. Okay, when AC is fighting with me, imagine. Okay, ready? Ready. Okay, we are going to do pistil and carpel. Okay, I think 2 hours maybe we will take for one chapter with question practice. So this way we will be able to complete all the chapters very fast. Okay, pistil or carpel, don't worry, I'll give you breaks also. I'll give you enough breaks also. So that is why make sure that you are having the energy. Yes, Deepak. Okay, yes. So start pannalama students. So idha nama stigma, idukapram style irukku, ovary, ovules irukku. To connect this ovary to the stigma, in the structure irukum students. This structure is present to connect the ovary to the stigma. Okay, to connect the ovary to the stigma only, this structure style is present. Okay, so stigma is the pollen grain landing region. Landing na nama helicopter ella land agume airport la adu madri da nama pollen grains vanda enga land agum in the stigma mele da land agum. Okay, in the stigma mele da land agum. So adanale this landing portion is called as the stigma. Adukapram style and then ovary. Inside the ovary, students, ovules will be there. Ovule is usually in a placental region. Okay, we say no placentation. Okay, marginal placentation, axial placentation, morphology chapter you would have studied, no? Placentation. So on the placentation, your ovules will be there. It can be single, multi-ovules. Okay, many ovules can be there. Now look here. This entire structure, stigma, style, ovary, together we will call it what? We call it carpel. What do we call it? Carpel. Carpel lavanda. Okay, carpel lavanda. Okay, carpel can be monocarpellary or multicarpellary. Yes or no? We all can have single friends or complete friends. Correct? No students. My English. Single friend or many friends, not complete friend, okay? Single friend or many friends, okay? So, single friend means monocarpellary. That means this carpel is like, bro, I don't want to talk to anyone, I want to be alone. Okay, some moody people will be there. No, adanala vanda, it will be single, okay? So, monocarpellary, that is single carpel. Other couple of students, the carpels can be multicarpellary. The carpels can be in a multicarpellary, many carpels. Okay. Now, this many carpels have multiple options. See, see, this bottle is alone. Okay, this is alone. This fellow doesn't have any other option. He has to be alone like this only. But imagine, multiple peoples are there. Okay, no multiple people are there in my hand. Okay, just imagine like this. Okay, so look here, like this. Okay, this is multi. Now they can decide, they want to be like, bro, I want to be together, bro. So fused. Okay, fused irukala, illena separate irukala. Correct? No. See, if many people are there, it is our wish. Should we sit with them? Should we stand? Should we be separated? But single person will not have that many options. He has to stand like that only. So, multicarpellary le fused irukkum, illena free irukkum. Fused condition. Okay, multicarpellary fused condition, we call it what? Syncarpus. Nama phone vandu sync pannume. PAN card, Aadhaar card sync pannume. So, fused means sync word you should remember. Okay, fused means sync word you should remember. So, fused na syncarpus, free na apocarpus. Okay, va? Okay, fused means syncarpus, free na apocarpus. Now, you have to remember an example. Okay, you have to remember example here. This is the diagram given in your NCERT. Konja adhiga brighter da irukum. Okay, this is your stigma, style, ovary, thalamus. Okay. Now, students, look here. 2 examples you have to remember. Apocarpus. Ayyapo, ma'am, how much you will teach, ma'am? So, apo means you remember me. Okay, you remember ma'am. Okay, I am self-damaging myself for you to remember the examples. Okay, so that is why. Make sure that you are I think another 10 more students and then we will be 11K. Okay, that's awesome, right? It's awesome. Imagine I have 11,000 students with me. That's like super duper exciting. Okay, yes. So students, inga vanda apocarpus. Apocarpus na ma'am. Apocarpus na ma'am. You apo ma'am. How much you will teach ma'am? Adu madri. Apocarpus na ma'am is the example. Okay, ma'am is the example. Clear? Now look here. M for multicarpellary. A for apocarpus, M for micellia. Example enna irukum students? Ma'am. M for multicarpellary, A for apocarpus, M for micellia. Okay, clear? Yes or no? Yes. Tansy, correct. Okay? Correct. Okay? Yes, I think many of them will have school and tuition. So after 6 o'clock, 7 o'clock, they might join. Okay, yeah. So look here, this is understood. Next one, the students, syncarpus. Syncarpus, okay. So that is going to be MSP. Okay, MSP. This is not a big trick or anything. I just attached the word. M for multicarpellary, S for syncarpus, P for papaver somniferum. MSP, okay, MSP, M for multicarpellary, S for syncarpus, P for papaver somniferum. Okay, va? Understood this much? Yes or no? Example is important, students. Question will come. Okay, question will come. So, adinala in the tricks ellame NCERT la please write and keep. Ippo you will be nodding like this. Oh, yes ma'am, understood. Because I am putting so much energy. Because of that energy, because of that voice modulation, nama brain lo automatically attach avum. But ana adukapram we will forget it. So, adinala I am requesting you, please idu vanda write it down in your NCERT and keep. Okay, MSP and your ma'am. Okay, MSP and your ma'am. Good evening. Some of you are joining little late. Okay. Yes. Now we will do anatropous ovule. What are we going to do? Anatropous ovule. Anatropous ovule. Now students, actually there are many types of ovule. Okay. Anatropous ovule. Then we have circumference ovule. So there are different types of ovules. This ovules here. Anatropous ovule. Na, if you look here students, nama chalazal, sorry, micropyla, chalazal end and funicle is not in a straight line. Right. If imagine. If they are all, sorry, if they are all in the straight line, it will be a different type of an ovule. Inga vanda nama funicle vanda in the side la acha, chalazal and micropyle is in a straight line. Okay. Now students, this is called as an ovule. Ovule means what? It will be inside the ovary. Yes ma'am, it will be inside the ovary. You are so smart ma'am. Okay. So look here. Now students, can you see the stalk-like structure? The stalk-like structure, and the stalk-like structure is called as the funicle. Okay, and the stalk-like structure is called as what? Funicle. Okay, adukapram students, this look here carefully. In the integuments and the funicle, okay, this, or let's say the funicle is attached to the ovule at a notch or a place. That place is called as the hilum. That place is called as the hilum. How many of you can tell me where have we studied the word hilum before? How many of you can tell me where have we studied the word hilum before? Okay, tell me quickly. You can draw this ovule this way also. Okay, you can draw it this way also. Okay, no problem. But I have drawn it this way. That's it. Okay, you can draw it this way or the other way around. Okay, clear? Okay, that's up to you, dear, to think like that, bro. No problem. You can think like that. Okay, I told a genuine answer. I didn't think about art books or anything like that, but it is a genuine answer. Okay, it's up to you how you want to complicate it and think about it. Okay. Okay, so here what is happening, students? In the position where the ovule is attached to the funicle is called as your hilum. Adukapra in the space, can you see the space? Before I tell about the space, you will have seen this yellow color and orange color structures. They are actually protective layers. Okay, they are what, students? They are protective layers of the ovule which are called as outer integument, and this is called as inner integument. So there are two protective layers which are called as outer integument and inner integument. Correct? Yes, kidney. Very good. Kidney. Okay. So outer integument and inner integument which are going to help in protection. Okay. Adukapra students, in the integument, okay, inside the integument, you will see a set of cells. Okay, a set of cells. These cells are called the nucleus cells. What are they called? nucleus cells, okay, not nonsense cell, nucleus cells. In the nucleus cells also ultimately will do one job, I'll tell you that. So student, the place where the integuments are absent, the place where the integuments are absent and the place we call micropyle end, we call it, okay, micropylar end, okay, micropylar end. Clear? Now look here, nucleus, okay, nucleus has the potential to become the megaspore mother cell. That is, students, one of the nucleus will be like, have you seen in your class also there will be one student, okay, yes, who's that? Shanti, yes, okay, not Shailaja, yes. So look here, in your class also there will be one student who is always like, I want to become the class leader, I want to become this, I want to do that, I want to do this. Adamadi continuous, he'll be raising hand for everything or she'll be raising hand for everything. Nucleus, one of the cells will be like, bro, I want to become the megaspore mother cell, I will only become the megaspore mother cell. Adamadri overact pani, it will come in the front and it will become the megaspore mother cell. So nucleus also helps with nourishment. Nucleus also helps with nourishment, and students, inside this what you are seeing is the embryo sac, which is the female gametophyte. That means the embryo sac will have what? Egg it will have the female gamete. Okay, it will have the female gamete. Clear? Understood? Then students, this end, the end opposite to the micropylar end is called as the chalazel end. Any doubt in the ovule? Because now we are going to learn megasporogenesis. Too zoomed it is, no? 1 second. Okay. Megasporogenesis. Ready all of you? Okay. Yes, let's start. So students, I told you, one of the nucleus cells will be like, I want to become the megaspore mother cell, I will become the megaspore mother cell. It will jump and come. This nucleus cells can be towards the micropylar end, okay. And the nucleus cells, when the students, it will become the megaspore mother cell, which is again diploid structures, because mother cells are all going to be diploid structures. Megaspore mother cell will become, it is diploid, it will undergo meiosis and it will form same like the microsporogenesis. It will form megaspore tetrad. It will form what? It will form megaspore tetrad. That is 4 of them it will form. Now we will be like, wait a minute, but there is a problem. The problem is, students, the 3 of them will degenerate. Out of the 4, 3 of them will degenerate. They will be like, yeah, I don't want to do this. So adanale, and the 3 of them will degenerate, and only 1 which is towards the chalazel end will become the functional megaspore. Enna nadakum? And the 4 of them, adla 3 of them will degenerate and only 1 which is towards the chalazel end will continue to the next step. Okay, super energy in the chat box. That is called as the functional megaspore. Students, in the madri or development okay, in the madri or development we can call it monosporic. We can call it what? Monosporic development. Okay, all these notes you will get it in the description box, okay, later after the class. We can call this kind of a development as monosporic development. Wow. Chat box is full. Okay, chat box is full. This is called as monosporic development because one of the spores are completely forming and it is going. Okay, now students, look here. This kind of a development is called as monosporic development. Now this monosporic development, after this, what will this fellow do? This fellow will be like, yeah, I will go and divide. Okay, enakku vandu divide pannam. So adanal na vandu mitosis panni, okay, karyokinesis, that is nuclear division panni, I will keep dividing. So from 1 stage to 2 cell, 2 cell to 4 cell, 4 cell to 8 nucleated. Sorry, not celled, 8 nucleated, because karyokinesis is happening, only the nucleus is dividing. Adanal 1 nucleus, 2 nucleus, 4 nucleus, 8 nucleate. So we will ultimately get a 8 nucleated embryo sac. 8 nucleated embryo sac. Okay. So look here, students, again, megaspore mother cell will undergo meiosis. Out of the 4 of them, 1 becomes functional, that is called the one towards the chalazel end. That kind of a development is called as monosporic development. And the single megaspore one, it will start dividing. Okay, it will start, the nucleus will start dividing. So we will get 1 to 2, 2 to 4, 4 to 8. So this will lead to 8 nucleated stage. Understood this much? Podu energy. Okay, thank you. Understood? Yes, yes. Embryo sac is the female gametophyte. Inside that, the egg is going to be there. Okay, inside that, the egg is going to be there. Okay, clear? Embryo sac is the female gametophyte, and inside that, the egg is going to be there. So, egg is going to be the gamete. Okay, understood? Okay. Now, look here, students. On the embryo sac, it is going to develop properly. It is going to form the entire thing. So, look here. Out of the 8 nucleated embryo sac, you will notice that 3 of them, so in the nucleus ellame move agum. So 3 of them will go to one pole, one end, one pole. The rest of the 3 will go to another pole. Okay. So here we have 3 of them that we call it as antipodals, which are towards the chalazal end. So adanala inga vandu in the trick use pananam. AC. Okay. AC na? AC. Okay. Antipodals in the chalazal end. And then 3 of them will move to the micropylar end. That is your synergids. Egg. So this total we will call it as egg apparatus. We will call it as what? Egg apparatus. Egg apparatus. Okay. What will be there? Egg will be there. 2 synergids will be there. Egg will be there. 2 synergids will be there. And students, on the synergids, there will be some finger-like structures which are called as filiform apparatus. And the filiform apparatus vandu nama pollen tube na guide panadukku romba helpful ah irukum. Okay. And students, right at the center namakku enna irukum? Polar nuclei will be present, and this is the central cell. So embryo sac structure is clear. 3 will move to the chalazal end, that is called as antipodal cells. 3 will move to the micropylar end, which is called the synergids, plus the egg apparatus. And right at the center you will have polar nuclei, and then there is a central cell. Okay, on the synergids there are filiform apparatus. Same thing what I taught you, it is given here. Okay. Same thing here. So the process of formation of megaspore from megaspore mother cell is called as Microsporogenesis. Ovules generally differentiate a single megaspore mother cell in a micropylar region. Okay. Yes, your drawing is nice, ma'am. Thank you. Okay. So students, if this is the diagram given in NCERT, look here. Nucellus towards the micropylar end will become the megaspore mother cell, telling that I want to become the megaspore mother cell. Adukapram adu vandu megaspore dyad agum, then megaspore tetrad. In the megaspore tetrad only 1 will become functional, rest 3 will go. Okay, and then they will go from 1 to 2, 2 to 3, 3 to 4, and then ultimately form the embryo sac. So in inge vandu students, there is actually more mitotic division than in before, right? See, look here what I have written here. 3 mitotic divisions. How many mitotic divisions are there in microsporogenesis? All of you tell me in the chat box. Let me ask you some questions as well. Okay. Let me ask you some questions as well. Very good. Yes, I asked the same question. Answer the question. How many divisions will happen? Quickly. 1. Very good. So I told you in 3-celled stage. Okay, remember this. Okay, remember this, students. In 2-celled stage. Okay, in the 2-celled stage, it is going to be, okay, it is going to be 1 meiosis and 1 mitosis. Okay, and then remember in a 3-celled stage, okay, in 3-celled stage, it will be 1 meiosis and 2 mitosis. Now what is going to happen in this scenario? In this scenario, students, it is going to be only 1 stage is there, okay? So for female gamete formation or embryo sac formation, we will go through 1 meiosis and 3 mitosis. Okay, 3 mitosis. So this is what will happen. Okay, understood? 1 meiosis and 3 mitosis, because this 3 mitosis is going to be like this. Okay, 2-celled or 2 nucleated stage, then 4-celled, then 8-celled. Okay, so like this difference would have happened. That is why we have 3 mitoses. Okay, 3 mitoses will be there. Understood? So here you can see that 7-cell. So students, this has come as a PYQ. Many a times it has come. Remember this. 7-celled, 8 nucleated. Just count here. 1, 2. First we will count the cell. Okay. 1, 2, 3, 4, 5, 6, 7. 7 cells are there. Okay. 7 cells are there. Evlo nucleus irukum? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated. So 7-celled, 8 nucleated stage because நம்ம polar nuclei வந்து ஒரு single cell consider பண்ணியாச்சு. So அதனால தான் ஒரு cell வந்து கம்மியா தான் இருக்கு. ஓகேவா? ஓகேவா? Understood? Can we go to pollination? Can we go to pollination? Okay, let's go. So students, pollination, we have microsporogenesis and megasporogenesis that has ultimately given us the formation of male gamete and egg. Okay, these two should do fertilization. For fertilization to happen, they should meet each other. If they don't meet each other, how will fertilization happen? So adanale, remember that here we have the flower. The pollen grain has to fall on the stigma. For that to fall on the stigma, we need to have a honeybee or maybe any insect for that matter. So this is the honeybee. Okay, this is the honeybee. Clear? Now students, what is going to happen? In the pollination one, it can happen with agents. Agents, okay. That is biotic agents and abiotic agents. Biotic na living agents. Living agents na you have insect to study. Entomophily, okay. Abiotic na wind irukum, water irukum. Hydrophily. Okay, anemophily, wind irukum, water irukum. So biotic means living. Abiotic na non-living. This much you people know. Living and non-living you know. Yes or no? Okay. Now, look here students. Kinds of pollination. Nama pollination vanda mainly 3 categories le divide aachu nama NCERT le. First one students, it is called as autogamy. Second is called as 𝐴𝐻𝐻𝑚𝑦. Third is called as xenogamy. Auto na enna? Auto. And auto kediyadhu. So autogamy na students, nama auto mode nu solluvaanga. Auto mode na enna? Nama phone le auto mode set paniya contraction, exposure, brightness ellame automatically phone will adjust. Adu madri da self it is. Auto na self. So autogamy na self pollination. Okay, that means transfer of pollen grains from anther to stigma of the same flower. Okay, same flower la nadakadha namma. Okay, yes. Okay, so autogamy na transfer of pollen grains from anther to stigma of the same flower. Okay, now students, in autogamy they have given 2 types of flowers. Like in one plant 2 types of flowers, chasmogamous and clistogamous. Chasmogamous na open flowers, adula exposed anther and stigma irukum. Clistogamous flower na adu open agadu. It will never open. So adanala adu vandu autogamy da pannanum. There is no other option. Okay, clistogamous flowers. Now students, look here. 𝐵𝐻𝐻𝑚𝐦. 𝐵𝐻𝐻𝑚𝐦 na enna? Mama ponnu, mama paiyan. So when you marry, okay, when you marry your mama ponnu or mama paiyan, that means what? That means what? When you marry your mama ponnu or mama paiyan, that means you're still marrying inside the family only. So adanale nama genetic disorders, whatever issues are there in the family, it will continue in the same bloodline. Adu madri da 𝐵𝐻𝐻𝑚𝐦. That is transfer of anther, pollen grains from anther to stigma of different flowers of the same plant. This is a flower, this is a flower, this is a flower. Ana ella flower me ore plant illa da. Ella flower me ore plant illa da. So adu vandu different variation kediyadu students, because ore family le mama ponnu mama paiya, if you get married also adu vandu same family da. So adu madri da from this flower to this flower it is falling, so it is still in the same plant. So adanala variation irukadu. No variation. Okay. Then students, xenogamy வந்து complete cross pollination. You're not marrying your mama ponnu or mama paiya, you're marrying from someone else's family. That will be called as xenogamy, cross pollination. That means from one flower to another flower. So anther, transfer of pollen grains from anther to stigma of different flower, complete variation will happen. Clear? Yes or no? Diploid. Megaspore haploid. Okay. megaspore haploid, nucellus diploid, MMC diploid. Okay, understood? Understood this? Yes or no? Okay, now we are going to understand pollination by wind. Only the words that I have underlined you have to look into. Only the words I have underlined you have to look into. Clear? So look here. Pollen. So in wind pollinated flowers, pollen grains are light. Okay. Pollen grains are what, students? They are light. Okay. They are light. And they have to be non-sticky, because if they are not light and non-sticky, wind will not be able to carry it and go. No. Wind will not be able to carry it and go. No. So that will be difficult. Okay, hi Ratis. Okay, these are my old students. Okay, so pollen grains are light and non-sticky, so they can be transported in the wind currents. They often possesses well exposed stamen. So stamen வந்து கொஞ்சம் exposed இருக்கும், because if it is not exposed, நம்ம wind வரும்போது it will not be able to transport. So அதனால pollen grains வந்து லைட்டா இருக்கும், non-sticky இருக்கும், and the exposed stamens will be present. Okay, அதுக்கப்புறம் students, often feathery stigma இருக்கும். Why? So as to accept the pollen grains. Often feathery stigma இருக்கும், so as to accept the pollen grains. Okay, then students, wind pollinated flowers will usually have single ovule in each ovary and a numerous flowers in an inflorescence. Inflorescence na ஒரு packet of flowers மாதிரி. Okay, inflorescence. Clear? Then here students, corn cob, the ears you see are nothing but the stigma and style. இது வந்து நம்ம old NCERT ல tassel னு கொடுத்துட்டா, but this year they have changed it to ears. Okay, they have changed it to ears. Clear? So the corn cob, the ears you see is nothing but the stigma and style. Okay, which wave in the wind and trap the pollen grains. Wind pollination is quite common in grasses. So only the words I have underlined you have to study. Well exposed, light, non-sticky, exposed stamen, feathery stigma, single ovule, inflorescence, common in grasses. Okay, common in grasses. Clear? Oh, thank you, Sri. You're the reason for my 170 out of 180 in locomotion. That is awesome. This is why you should watch my lectures. Understood? See, students are getting full marks. Okay, next one, students, pollination by water. Namakku speed la poga. Okay, speed la poga. So pollination by water is quite rare. Okay, quite rare. So in flowering plants, it is limited to only 30 genera, mostly monocots. Okay, as against this, you would recall that water is a regular mode of transport in lower plants. Nama bryophyte vandu we call it the amphibian of the plant kingdom. Edukku ma'am? Because bryophyte life cycle complete பண்றதுக்கு it needs water and it also needs sometimes land. So அதனால நம்ம வந்து amphibian of the plant kingdom we call it. Okay, so here you can see students, நம்ம waterல வந்து here Vallisneria and Hydrilla, Zostera, all aquatic plants use water for pollination. Ana, this water hyacinth, okay, water hyacinth and water lily. Okay, this flower is what they will do, students. இதுதான் நம்ம water. Okay, இதுதான் நம்ம water. So this flowers will come like this on the top. Okay, you can see my hand. Wait, okay, this is the flower. Okay, this pen is the flower. So இதுதான் water. These flowers will come on the top of the surface and they will wait for wind or insect to pollinate them, because they are not confident that water pollination will happen enough. So adanala they will be like, it's okay, I will do one thing, I will do insect or wind pollination. This has come in 2023 PYQ. Okay, so insect or wind pollination. So look here, the flowers will emerge above the level of the water and they get pollinated by insects or wind. Okay, so it is very confusing. Why it is very confusing? Because on the question varumbodhu automatically you will see the word water and you will be like, oh, busy water hyacinth and water lily vanda, it will get pollinated by water. What a question this NEET people are asking. But Anna, that question is twisted. Why? Because even though they are in water, they will come above the surface of water and wait for insect and wind to pollinate them. Okay, adukapra students, in Vallisneria, okay, in Vallisneria, the female flowers reach the surface of water by a long stalk and the male flower or the pollen grains are released onto the surface of water. Slowly, passive current illa, passive and the water slow move agule, because of the movement in the pollen grains vanda move agum. Okay, and the students, they will reach the female flower. They will reach the female flower. Clear? Understood? Understood. See, look here, like this. So here you can see, pollen grains have come. The pollen, what is this? Pollen grains have come and they are coming here. Pistillated flower, okay, pistillated flower means flower which has only the pistil, okay. Pistillated flower will accept this pollen grains and then pollination will happen. Inga vandu staminated flowers irukku. Staminated flowers na male flower, pistillated flower na female flower. Okay va? Okay kanna, understood? Yes or no? Students, in the pollen grains la vandu oru mucilaginous covering irukum so as to not அது வந்து sink ஆகக்கூடாது. ஸோ அதனால தான் ஒரு கோட்டிங் இருக்கும், ஒரு mucilaginous covering இருக்கும் on the top of the pollen grain so that they don't sink. Okay, they don't sing, no, they don't sink. Okay. How many of you saw the movie? How many of you saw the movie? Jananayagan, come on, tell me. Students, I want to see students. You people are not studying, that's why I'm not seeing. Okay, I have decided that I will only go and watch a movie until my students are, you know, every day attending my class. So, adanale. Never give up. I did not give up, so that's why I'm taking classes here. Students, how many of you saw Jananayagan? I only saw Talapati Kacheri, tanana, 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 tanana. Only that much I saw. That's the only thing, okay? Release, it didn't release, it got released, no. No one saw the movie, huh? All of us are the same. I'm teaching you, you're studying. We'll all have to go see the movie. Okay, it's our last Talapati's movie, students. We have to go and see. Okay, that's a tribute that we can give it to him. Okay, I'm gonna cry. Not now, later. Okay, now look here. Okay, in the female flower, this is the male flower. Adu vandu ipdi float agum water la, and by passive current they will reach the female flower. In the female flower will accept the male. Okay, clear? Movie is not yet released. Only the song is released. Then why are people going to the theater? Why are people going to theater? I am confused. Why people are going to theater then? Students, I don't even know the release date, students. I thought song came, so movie also came. Okay. Thank you. See, I thought, I kept thinking movie got released and I kept thinking that I am missing out. Okay. I kept thinking that I am missing out. Should tell all this. No, see, no knowledge only I have. Wow, just 5 more students and we are 11,000 team. Okay, we are a group, or we are a family of 11,000 people. So who are the 5 people who have not subscribed? All Kanjus in my chat box. Okay. Okay, awesome. Oh, that is why I am very confused. Okay, students, I don't know much about movies. It's not like I don't watch, but on a release date, a song release, adalama I don't understand. Okay, I'm little slow in all this. I am only fast in teaching you people. So people should forgive me. Okay, ready? Done? Tired? No, right? We have long chapters. Okay, these 2 chapters, if you complete, you will really feel so accomplished. Okay, you will feel so accomplished. Trust me. Okay, I'll give you a break. Don't feel bad. Okay. Oh, that is why. Yeah. Okay. Yes. Next one. Okay. Now, students, insect. Insect is very easy, students. Insect is attracted. Now, you imagine you are a honeybee. Okay, you are flying like this. If a fly banamode, you see a very boring flower. Color illa, onnume illa, summa epdi it is looking. You will not feel attracted. Okay, you are an insect. So imagine you are flying as an insect. Suddenly you see a very bright flower like this. Obviously you will get attracted. So on the job vandu nama corolla illa petals will take care. That is, insects need colorful flowers. Okay, colorful flowers. So students, this is a PYQ. Okay, this is a PYQ. Common pollinating agents. Bees are the dominating biotic pollinating agents. Other organisms also are there, like primates, lemurs, tree dwellers, rodents, or even reptiles. All of these people also are going to do pollination, but nama bees the maximum dominating pollinating agents. Okay, now students, this is my honeybee. Okay, this is my honeybee. I don't know what honeybee is this. We will name it. Okay, we will name it by ourselves. All of you tell me. Okay, give different names for this honeybee. The best name will be selected. Okay, you will get gift. Okay, now look here. So students, நம்ம insect pollinated flowers வந்து large ஆயிருக்கணும், colorful ஆயிருக்கணும், fragrant ஆயிருக்கணும், rich in nectar ஆயிருக்கணும், because insects have too much demands. Insects is like, see, I will not come if you are very boring. So அதனால colorful flowers இருக்கணும், nectar should be there, and also they should have plenty of pollen grains as well. So see, animals attracted to flowers by color or fragrance, the flowers pollinated by bees and beetles. Okay, நம்ம flies illa students, house flies and beetles, they get attracted to foul smell. Okay, foul smell, don't worry, never give up, majority is English only, you can understand. Okay, they get attracted to foul odor. Okay, nama Rafflesia flower illa students, and the Rafflesia and all has a foul odor, and the flowers also, this flies and beetles will get attracted. So this foul odor also is a very important thing to sustain the animal visit. That means, let's say I want you people to come back to my class. What will I do? I will teach very well, and I will make sure that you are understanding everything. Okay, we can understand, you people can understand everything, so you will come back to my class. Adu madri sustain pandradhukku, animal visit sustain pandradhukku, we have to give them reward. Namalla flowers have to give them reward. What are the rewards that they have to give? Nectar and pollen grains. Okay, nectar and pollen grains are few of the usual rewards that is given by the flowers. Clear to all of you? So students, nama animal body vanda, it will attach the pollen grains, it will go and sit on the next flower, pollen grain dusting will happen. Okay, okay, you are sitting in Varshan's class, huh? Okay, now here two examples I have given, very important. Symbiotic association. Okay, symbiotic association it is. We can say symbiotic, or mutualism also we can say, association. Okay, in the symbiotic association, students, we have to remember the example of amorphophallus flower. In the amorphophallus flower vanda, it is going to be this flower. Okay, this is nama amorphophallus flower. This flower is going to allow the flies, okay, to lay their eggs in the ovary, ana. Okay, ana. What is going to happen? This flies will be like, okay, since you are taking care of my eggs, since you are taking care of my eggs, I will do pollination for you. Adu madri or help. So you give me a hand, I will give you a helping hand. Adu madri da. So this amorphophallus flower will allow these flies or beetles to lay their eggs in the ovary, and the flies and beetles will always come and pollinate the flower. Same thing students, Yucca moth. Yucca plus Pronuba moth, what they will do? Yucca will protect the moth eggs, and moth will do pollination for the Pronuba. Pollination for the Yucca. Clear to all of you? This is the amorphophallus. So the fly will lay egg in the flower, and this will do the protection. Instead, the fly will always do what? Pollination. Fly will always do pollination. So amorphophallus flower doesn't have to take tension, saying that who will pollinate and all that. Then students, this is your Pronuba moth. Pronuba moth, and this is your Yucca plant. Okay, Pronuba moths are white color moths. Here you saw, no? This is a real image. That white color moth is the Pronuba moth. So Yucca moth, we can also call Yucca moth, pollinates the flowers and lays eggs in the ovary. Is this clear to all of you? Okay. Done. Okay. Okay, yes, we are almost there. Okay, we will complete the chapter very soon. Okay, now students, listen here. Continued self-pollination. Now imagine, you also imagine, you are sitting in a room every day. Okay, not talking to anyone, you do not watch phone, you do not listen to music. Every day you are just sitting and looking out of the window. Do I call that depression? Do I call it depression? Ma'am, because I am sitting alone, I am starting to have depression, you can tell me. So, continued self-pollination. That means, imagine, by yourself you are sitting every single day, you will obviously go to depression. Adu madrita, nama plants students, if continued self-pollination happens, it will lead to inbreeding depression. It will lead to what? Inbreeding depression. So on the inbreeding depression avoid pandradhukku, plant itself is like, relax bro, I don't want any outsiders. Na vanda outbreeding devices create pandren, and the outbreeding devices vanda 4 of them are there. Okay, okay, clear? Oh, is it? Okay. Cosmic Ideas will be thinking, ma'am, I watched all the 3 parts, ma'am, you're doing one shot here. So students, first of all, Continued self-pollination leads to inbreeding depression, and to avoid inbreeding depression, we have outbreeding devices. We have outbreeding devices. First outbreeding device வந்து என்ன இருக்கும் students? Asynchronized receptivity. Now normally என்ன நடக்கும்? Anther and stigma will mature at the same time. So when the pollen grains fall on the stigma, stigma will be like yes, let's do fertilization. அந்த மாதிரி ஒரு synchronized நடக்கும். ஆனா இது வந்து asynchronized receptivity. Asynchronized na the pollen grains are going to fall on the stigma, right? Now both of them are matured. In this scenario both are matured. ஆனா இந்த scenario la stigma வந்து mature ஆகல students. Anther has got matured first. Stigma has not got matured, anther has got matured first. So இப்போ when the pollen grains fall on the stigma, will fertilization happen or not? Yes or no question. Will fertilization happen or not? Answer quickly. Will fertilization happen or will fertilization not happen? What do you think? Fertilization will happen or fertilization will not happen? Easy question da, answer. Yes. Question has answered. Who else? Fast. What is this? What is this? Slow you people are doing. Huh? Very slow. No. Yes. So here pollination will not happen. Sorry, fertilization will not happen. Why? Because pollen grains that has fallen here will not get fertilized. Stigma will not accept. Compatibility will not happen. Inga vanda students, anther has not matured and stigma mature acha. So again here also nothing will happen. Okay. Now look here. Second scenario vanda changing the style. That is, style means the position. Heterostyly we call this condition as. That is changing the positioning of the anther and the, that is stigma. Okay. So normal scenario la vanda anther vanda it will be on the top, so the pollen grains can fall in the stigma. Ana heterostyly la enna nadakkum students? Anther length or the filament length will be reduced. Ipo vanda reverse pollen pogadhukku chance romba kammiya irukku. So adanal heterostyly. Inga paakumbodhu stigma vanda height la irukku, anther vanda is a small. So adanal here also this will not happen. Okay, clear to all of you? Heterostyly. Okay, now asynchronized receptivity, two types are there, protogyny and protoandry. So one is maturation of the stigma, one is maturation of the anther. Our next one, the students, unisexual flowers. So in one plant male flowers, one plant female flowers. So self-pollination will not happen because one male flower, one female flower, separate it is. Okay. Example, papaya. Change in position of anther and stigma, which is your heterostyly, and self-incompatibility. Nama plants, it will tell it to itself, telling that bro, if same pollen is coming, you reject off. Self-incompatibility. Okay. So protogyny, stigma maturation, protandry, anther maturation, dichogyny, and then your self-incompatibility. Clear to all of you? Understood this? Quickly answer. All sleepy heads have come to my class. Okay, all sleepy people. All sleepy people. Come on. Okay. Understood? Understood? Done? Yes or no? Okay. Yes. Okay. Okay, done? Okay, done. Now look here. This is understood. This is the what is given in NCERT, okay? So make sure that you're remembering it, okay? See students here, can you see pollen grains are here? This circle is the pollen grains. They will come and fall, okay? They will come and fall on the stigma, and from the stigma, can you see small stretching is happening? That is your pollen tube germination. So this is how the entire pollen tube and stigma, this will happen. But before that, you have to know pollen-pistil interaction. What should you know? Pollen-pistil interaction. Enna ma'am in the pollen-pistil interaction. So 2-cell stage, 3-cell stage is understood, no? Now students, imagine after the pollen grain has fallen on the stigma. Stigma-க்கு decision எடுக்கலாம். என்ன decision எடுக்கலாம் ma'am? If it should accept the pollen or reject the pollen. என்ன நடக்கும்? If it should accept the pollen or reject the pollen. So here you should remember, one second, where is that? Oh no, I think that went off students somewhere. Okay, nothing to worry. Nothing to worry. Okay, yeah. So what happens students here, basically pollen-pistil interaction where the stigma will take a decision if it should accept the pollen grain or not. Okay, if it should accept the pollen grain or not. So let's say on the stigma the pollen grain has come and fallen. Okay, pollen grain has come and fallen. Now which pollen will get accepted? That totally depends on the stigma, because stigma will be releasing some chemicals. Okay, now students, 3 pollens have come and fallen on the stigma. Okay, இப்போ இந்த 3 pollensல imagine பண்ணிக்கோங்க. Okay, let's consider this is as a marriage proposal. In the marriage proposal, students, one is a doctor, one is an engineer, one is a bank manager. So 3 boys also have come and 3 boys are also telling this girl that, you know what, I will take you for vacation, I will make sure that your parents and everyone is healthy. நான் வந்து இது பாத்துக்கிறேன் அது பாத்துக்கிறேன். So girl is like confused. Okay, so on the first interaction between a pollen and a stigma is going to be called as a chemical dialogue. Okay, or a chemical interaction, because inga vanda students chemical will be released and that will lead to chemical dialogues, exchange of chemical dialogues, illana chemical interaction we say. And the chemical interaction ku apparam nama girl vanda she will select. Okay, the girl will select if she wants a doctor, engineer or a bank manager. So let's say the girl has got convinced that, okay, I want to get married to a doctor. So and the doctor will be allowed to enter inside her life, right? Yes, enter inside the life means here pollen tube germination is going to happen. So here what is happening students? Here once the stigma accepts it, the pollen tube germination will start. Pollen tube germinate. Oh, wait. Pollen tube germination begins. Okay, இப்போ என்ன நடக்குது? Now the pollen tube will start germinating and it will all the way go till the embryo sac. Okay, it will start going towards the embryo sac. So the girl has accepted the doctor. Now the doctor is allowed to enter inside her home. Okay, and finally he is going to meet the parents. Okay, adu madri da. So pollen tube germination. Okay, or we can say pollen tube growth. If students in the pollen tube grow achu, now inside this what will be there? One tube nucleus will be there which is going to lead the way, and then your generative cell male gamete is going to be there. Ana imagine பண்ணிக்கோங்க இது 2-cell stage. Ana generative cell வந்து decide பண்ணிட்டா, oh no, I want to do mitosis. Then the scenario will be having 2 male gametes. Okay, 2 male gametes. How it seems like I'm telling in a very funny way. What is happening students? Okay, what is happening? Basically imagine the brother has come for the marriage. Okay, and one more younger brother is there. Now that brother will be like, no, even I want to get married. So adu madri tha last minute la vanda innoru male gamete form agum. So one male gamete can go and fuse with the egg, ana innoru male gamete will fuse where? So adanal oru confusion irukum, right? Oru confusion irukum. So this is called as pollen-pistil interaction, where the stigma will accept the pollen and after that only germination will happen. So students, on the pollen tube it will move through the style, go to the ovary, then to the ovule, then to the embryo sac. From the embryo sac, it will enter the cytoplasm of the synergist, then it will enter the egg. In the synergist, near the synergist only filiform apparatus will be there, who will be like, come inside, this way, in the way, they'll tell. Okay, but so style to ovary, ovary to ovule, ovule to embryo sac to cytoplasm of synergist, and then the egg. So now the pollen tube has gotten entered. Okay, now pollen tube has got entered. Okay, ana inga male gamete irukku, oru male gamete can fuse with the egg, problem kedaiyadhu, because adha nama initial plan. Okay, Yes, initial plan. But ana halfway la vanda நம்ம generator cell has decided to do a mitotic division, then ஒண்ணுமே பண்ண முடியல students, அந்த mitotic division நடக்கும், 2 male gametes will be formed. இன்னொரு male gamete எப்ப போய் fuse ஆகும், எங்க போய் fuse ஆகும், we will understand here. So look here, this will lead to the next topic called as double fertilization. So students, இது male gamete will fuse with the egg. அதுதான் நம்ம initial plan. Elder brother will get married to a bride. அதுதான் நம்ம initial plan. ஆனா plan வந்து flop ஆச்சு because அந்த younger brother will like the sister and he'll be like, I want to get married now only. So அது மாதிரி தான். So suddenly ஒரே இதுல மண்டபத்துல 2 marriages will happen. Okay, I'm telling all this so you remember this lesson. If someone else listen, they are going to sit and cry. Okay, so students, in the male gamete will fuse with the egg and it will form zygote. And the zygote வந்து diploid ஆயிருக்கும். Okay, n plus n diploid it will form, and this zygote, this male gamete plus egg formation is called as syngamy. Male gamete plus egg formation is called as what? Syngamy. Okay, இதுதான் நம்ம actual scenario. In 2 cell stage also syngamy will happen. அதுக்கப்புறம் male gamete plus polar nuclei, and the second male gamete இருக்குல்ல students, அது வந்து it will go and fuse with the polar nuclei. It will go and fuse where? It will go and fuse with the polar nuclei. So that will lead to the formation of primary endosperm cell. இல்லைன்னா primary endosperm nucleus ன்னு கூப்பிடலாம், PEN. Okay, so students, இங்க வந்து n இருக்கு, இங்க வந்து 2n இருக்கு. So ultimately நம்ம primary endosperm nucleus வந்து 3n இருக்கும். So இது வந்து triploid இருக்கும். So அதனால இந்த conditionக்கு we call it as triple fusion. So when syngamy and triple fusion happens together, we call that process as double fertilization. Okay, clear? Yes or no? Thank you, Nandakumar. So students, male gamete plus egg, zygote, syngamy. இதுதான் நம்ம actual marriage, actual plan இதுதான். Male gamete வந்து egg கிட்ட fuse ஆகி it should form zygote. ஆனா இங்க வந்து ஒரு twist நடந்துச்சு. Male gamete will now fuse with the polar nuclei and it will form primary endosperm nucleus. Okay, so primary endosperm cell or primary endosperm nucleus which will form 3n. Why? Because we know polar nuclei has 2 nucleus. It is considered as a single cell. ஆனா 2 nucleus இருக்கும். So n plus 2n will form 3n. Okay va? Wow, nice energy. Yes, yes, go for. Okay, so students, primary endosperm cell, primary endosperm nucleus, ultimately it will form endosperm. Ultimately it will form endosperm, and this endosperm will provide nourishment for the growing embryo. Is this clear to all of you? So syngamy plus triple fusion is called as double fertilization. This will happen in 3-celled angiosperm. 2-celled angiosperm, okay, sorry. 2-celled angiosperm only syngamy will happen. Only syngamy will happen. புரிஞ்சுதா? Understood? Okay, next topic வந்து artificial hybridization. Okay, let's take 1 minute breathing break because my voice is going. Okay, my voice is going to rest. Okay. Yeah, physics second class is going to happen here. Last week வந்து கொஞ்சம் problems இருந்துச்சு, so அதனால தான். So his sir's class will happen. Okay, sir's class will happen. Yes, all of you quickly let me know if you understood. Understood, right? Okay. Okay, done. Okay, next topic one, the students, artificial hybridization. Artificial hybridization na enna ma'am? Artificial hybridization na imagine பண்ணிக்கோங்க students, I'm a farmer. Okay, I do cultivation and everything. I also have flower farming and other things. Okay, இப்போ suddenly வந்து I'm thinking that, oh no, I want to make this flower because you know Ambani's wedding is coming, so adanala vandu inda flowers pannanam, commercial value la adu increase pannanam. So அந்த டைம்ல வந்து நம்ம artificial hybridization பண்ணலாம். What is artificial hybridization? Basically students, நம்ம self pollination, for self pollination to happen we should not give any chance. Okay, because if self pollination happens, then my whole plan will go. Okay, I can't go for Ambani's wedding. Okay, so here you should see no self pollination. No self-pollination. So what am I going to do? I am going to remove the anther. I am going to do what? I am going to remove the anther with the help of a forceps slowly, and I am going to bag that plant and keep, bag the flower and keep. Okay? Clear? Yes, yes, I'll give you a break. Okay, I'll give you a break. So here students, இங்க வந்து செல்ஃப் பாலினேஷன் நடக்காது. இந்த ஆந்தர் வந்து நம்ம ரிமூவ் பண்ணிட்டா அதுக்கப்புறம் நம்ம அதை என்ன பண்ணணும் ஸ்டூடெண்ட்ஸ், அந்த ஃப்ளவர் வந்து நம்ம பேக் பண்ணணும் with the use of a butter paper. We cover the emasculated flower with a butter paper. Emasculated. Now that means what? Anther is not there. Emasculated flower. So we have like we have removed the anther from this flower and we have bagged it. That process is called as, so this process where we remove the anther from the flower is called as emasculation. See, in my notes everything is there. In my notes everything is there. Okay, Gopika Ma'am Armida, wow wow, nice nice. Okay, so removal of anther from the flower is called as emasculation. Adukapra students, emasculation appuram we have to, like, see, if I don't cover the flower, any other pollen grains can come and fall on it, right? I want the desired flower that I am thinking of. So adanala nama on the flower la we will put butter paper and we'll cover it. That is called as bagging. Okay, adukapram let's say I want to make a flower of my own. Okay, yes, this Josh is more than enough. Yes, so students, adukapram I will dust the pollen grain that I want to fall on this stigma, that is called as desired pollen grains, and then I will rebag it. Rebagging na idukapram one more bagging I'll do. Okay, that is called as rebagging. Okay va? Understood? 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 Wow. Wow, wow, wow. We just need 3 more Gopika Ma'am armies and then we are a group of 11,000 students. Okay, so I'm super happy that we will become 11,000 in this class only. Okay, just 3 more students and we are 11,000. Okay, this is awesome. Yes or no? Okay, so from small we started, now we are growing. Okay, 3 more Gopika Ma'am armies and then doom, we'll become big. Okay, yes. Okay, students, like this we should be able to do 1 million, okay, 100k. Idu madri we should display it on the board and do it, right? Okay. Okay, yes. So students, understood this? So artificial hybridization, 2 steps are there: emasculation and bagging. Emasculation na nama anther remove pandra process, emasculization. Okay, adukapram bagging na nama butter paper vechi cover pandra process. Okay, cover pandra process. Okay. Who is the 3 conjuice? Even I don't know. Okay, yes, this is understood. Yes, we are coming to post-fertilization events, so I'll give you a 10 minutes break. Okay, post-fertilization. Actually, I wanted to finish the chapter, but the problem is after post-fertilization events, I have to make you do questions. So, adanala, I'll give you the first break here. Okay, first break I'll give you here. Adukapram, I'll give you one more break. Don't run away, come back here. Okay, you people ask me for break. Adanala, don't run away. Okay, this much is there. Little more is there, students. Another one maybe 20 minutes. Okay, then we will do questions and then human reproduction. Okay, so much is there. Another one, 10 more slides are there. Then we will do human reproduction. Human reproduction also is a big chapter, so we need energy before that. Okay. Okay. So students, what's the time? 6:20. Okay. Oh, sorry. Okay. Yes, we will definitely. We have to cut the cake, no? Okay. Okay. Yes. Quickly run and come back. Okay, I'll also fill water and come back. Hello, Basingla. Yes. How are you? Yeah, tea was very hot, cold. So, Adinala, I'm very upset. Okay, there's no tea. I'll order coffee after some time. Okay, you people remind me. Ma'am, drink coffee. Okay. Tea was so cold because it was made at 4 o'clock. So, Adinala, I'm very upset. Okay. Yes. So, let's finish off this. Okay, then we have to start human reproduction. Reproductive health will take 30 minutes. Okay, max to max reproductive health வந்து max to max 30 minutes. அவ்வளோதான். Okay. New friend ah? எங்கேடா? Oh, Shailaja. Okay. நம்ம Telangana friend it is. Okay, she has come from Telangana or AP. நமக்கு வந்து full mixed friends. Hybrid நம்ம channel வந்து. Okay. Done. Okay. Organism and population. இருக்கு. Yes, definitely, Karishma. Today I'll do it. Thank you so much. Okay, start பண்ணலாமா students? Post-fertilization events. Post-fertilization events na என்ன? So, pre-fertilization events la வந்து நம்ம gamete formation ஆச்சு. Fertilization la வந்து fertilization ஆச்சு. Post-fertilization la வந்து நம்ம ovule and seed formation பண்ணணும். Okay வா? So, here you see PEN undergoes successive nuclear division to give rise to free nuclei. Don't worry about this. I will come back in the this one is enough for you to understand the whole thing. That is the importance of notes. நம்ம NCERT பார்க்கும்போது நமக்கு இது என்ன important தெரியவே மாட்டேன். Like just look at it. You will not understand which is important, which is not important. But ஆனா இந்த slideல you will understand. Okay, something is happening. So here students look here. PEC and PEN. Okay, PEC and PEN is there. In the PEC na primary endosperm cell. PEN na primary endosperm nucleus. Okay. And then you will notice that slowly inga vandu zygote formation start achu. Okay, zygote has to become now embryo. Okay, zygote has to become now embryo. So, zygote will slowly start developing. Okay. Slowly start developing. Karyokinesis will happen. No cytokinesis. So, first vandu nama PEC illana PEN vandu only free nuclear division pannum. Okay, it will do what? Only free nuclear division. Okay. So, inga students nucleus. So, only karyokinesis is happening. So, ultimately, nama karyokinesis complete acha. We have got free nuclear division. Now, example vandu students nama tender coconut water. Tender coconut water, what do you say in Tamil? Tell me quickly. Yelneer, no? So, Yelneer, so that tender coconut water is actually a free nuclear division of your endosperm. Endosperm will undergo successive nuclear division leading to the formation of free nuclear division. They do not have any restriction. தானே அந்த ரெஸ்ட்ரிக்ஷன் எதுவுமே கிடையாது. ஸோ அதனால தான் இது ஃப்ரீ நியூக்ளியர் டிவிஷன். பை தென் ஸ்டூடெண்ட் ஸ்லோலி எம்ப்ரியோ ஹேஸ் ஃபார்ம்ட். ஓகேவா? அதுக்கப்புறம் ஆஃப்டர் த ஃப்ரீ நியூக்ளியர் எண்டோஸ்பர்ம் ஸ்லோலி cytokinesis start ஆகும். That means cell wall formation start ஆகி see here like this cell wall formation will happen. Understood? This cellular endosperm is the white kernel that you eat. Okay? Yellow-neer. Yeah, yellow-neer. Okay, that white kernel that you eat, no? What do you call that? The white color thing inside the tender coconut. அது வந்து சாப்பிடும்போது that is actually a cellular endosperm. The water that you drink is a free nuclear endosperm. So, next time you go to a tender coconut person, you should say, I want only free nuclear endosperm in my Yelneer. I want only the white kernel, cellular endosperm in my Yelneer. He will throw that on your head. Okay. He will throw that on your head. Okay. And don't come crying. I don't know. I will tell many things. Okay. If you follow that, I don't know. I'll tell like that and I will. Tengai. Okay. Same yeah. Malayalam is tenga. Here it is tengai. Okay. Understood? Next time you should go and ask like that. Okay. And then tell me what is the response. Okay. Okay, that is understood. So, here you can see students, cytokinesis agum, cell wall formation, developing embryo, endosperm, nourishment for the developing embryo. So, always remember that first Thank you, Kaitishri. Okay. So students, what is going to happen? Please understand. Endosperm will be 3N. Always remember. Now, if I have to provide nourishment, I should be formed first. Then only I can provide nourishment. Adu madrita, endosperm will precede the embryo development. Endosperm will precede the embryo development. That means endosperm should be formed first. Adukapram the embryo formation will happen. Okay, va? Okay. Now, students, see here. Embryo formation happens in stage to stage situation. Okay. That is, first from zygote, you will have a pro-embryo stage. Adukapram, you will see globular embryo. Okay. You will see globular embryo. Adukapram, you will see heart-shaped embryo. Adukapram, the complete structure will be formed where you will start seeing radicle, which will again form the root. Okay. This is the basic part of it. So, look here. Zygote, pro-embryo, globular embryo, heart-shaped, and finally mature embryo, which will have plumule, radicle, cotyledon, all of it. Okay, cotyledon, all of it. Clear? Now look here. Mature dicot embryo, students, there will be a root cap. Root cap edukku ma'am for protection, because adu vandu hard rock, illana road, illana number soil vandu students stones and all will be there, no? So the root tip shouldn't get damaged and the root meristem shouldn't get damaged. Adanal a cap like structure will be present. Okay, cap like structure will be present. Are you all understanding? Stop talking to each other. Root cap, radicle, hypocotyle, cotyledon and plumule will be there. So please remember here, look here, a typical dicotyledon embryo will have an embryonal axis, 2 cotyledons, okay, and the portion of the embryonal axis above the cotyledon will be called as epicotyle, below the cotyledon will be called as hypocotyle. Okay, this and all you can do quick recap. Students, in the question vandu nama test series la irukum. So when I made the test series, no, in the this question I have added. So make sure that you are studying. Okay, clear? Suresh Babu, it is live. Okay, done. Okay, now look here, students. Here you have this scutellum. Then you have the coleoptile, shoot apex, epiblast. Epiblast is just a portion, okay? And then radicle, root cap, coleorhiza. In the coleorhiza vandu in the root region la irukum just as a protective covering. In the coleoptile vandu shoot region la irukum again as a protective sheath. Okay? Okay. So here you can see embryos of monocot possesses only one cotyledon. In the grass family, the cotyledon is called as scutellum. The cotyledon will be called as what? Scutellum. That is situated towards one side, lateral. I told you like these things you have to focus, because NEET 2026 one the difficult they will take lateral, ventral, dorsal, all that side they will take and they will make you confused. So adanala this and all please underline in your NCERT now itself. Yes, Nitya. Thank you, Dr. Dikshita. All of you should comment, okay? All of you should comment and only go. Students, one side lateral of the embryonal axis. At its lower end, the embryonal axis has a radicle and a root cap enclosed in an undifferentiated sheath called as the coleorhiza. The portion of the embryonal axis above the cotyledon or above the attachment is called as epicotyle. Below will be called as hypocotyle. Same thing, but instead here we are using the word scutellum. Understood this? Yes. Now we are coming to types of seed. Almost we are ending the chapter. Okay. Coming to an end of the chapter. Okay. So, types of seed one the students. First one the, let's imagine. Okay. I gave you one treasure. Yes. Okay. I gave you one treasure. Okay. Let's say I gave Deepak and Nafal around 25,000 rupees. Okay. Both of them I gave 25,000 rupees. Deepak vanda and the 25,000 rupees, he has taken all of you people for trip and he has enjoyed, he has spent full money. Not even 1 rupee he has. Okay. And then Naufal vanda and the 25,000, he spent 10,000. Me the 55,000 he has saved and kept it. Telling that I'll use it later. So, Adamadita, I am giving you endosperm. The plant is giving you endosperm, telling that use this for nourishment. If you use up the entire endosperm, then you are called as a non-albuminous seed. Anna, let's say some of the endosperm you have kept it back. You have not utilized it completely. You are called as albuminous seed. Okay, clear? So students, in the situation, this is Deepak. Okay, he will have no money, no endosperm. So the seed has utilized the entire endosperm. Deepak has utilized the entire endosperm. Okay, clear? Okay, va? Yes, I will remember all of your name. Okay, Deekshita, Shailaja, Naufal, Karishma, Deepak, all of your names I'll remember. Okay, all of your names I'll remember. So he has no money. Why? Because he has utilized the entire money that I've given him. So adanale it becomes a non-albuminous seed. Endosperm irukka, the students complete endosperm வந்து utilize ஆச்சு. In the example, ஞாபகம் வச்சிருக்க this word doesn't come, students. I don't know why. Okay, some words doesn't come for me at all. Okay, some language problem I have. Okay. So it is called as non-albuminous seed. To remember this example, you have to remember PUBG. You have to remember the game PUBG. What is the PUBG game? That is PUBG. P for P, U is chuma. Okay, dummy it is. Okay, B for beans, G for groundnut. Okay, I have told this in 2 videos now. Okay, 2 videos, I'll become famous now. Okay, I'll become famous. So now look here. Okay, yeah, Rathas and all knows that how many times my students, not only in Tamil, Malayalam, English, Kannada, every language I will have one word that will slip out of my tongue. Okay. Oh, don't know PUBG, ma'am. Thank you, Aradhana. It's such a sweet name, yeah, Aradhana. Okay. P, beans, groundnut is the example of non-albuminous seed. Ana albuminous seed la now fall. He has saved around 15,000, which I will later take it from him. Okay, which I will take it later from him. So he has preserved some endosperm. So endosperm retains, seed doesn't utilize the entire endosperm. Okay, va? Seed doesn't utilize the entire endosperm. And the seed ku nama enna solluva? Albuminous seed. Example one enna irukkum, students? Castor, barley, wheat. Castor, barley, wheat. Clear? Understood? Some trick you can make, you please make. Okay? BMW you can make, no, BCW. That one word is not there only. BW. JW Marriott, students, like that BW will make of. BWC. CWB? No, CWB is what? Okay, something, okay? Thank you, Jagdish. Okay, next question. Okay, this is done. So, non-albuminous can also be called as X-albuminous and albuminous, which will retain it. Okay, students, inga vandu or example irukku, BB house. BB house in a Big Boss house. W. Students, example, trick is unlocked, new trick unlocked. Okay, so you won't believe, one of the sirs came to tell this trick till here. Nama class vanda avlo interesting irukku. Okay, sir is watching biology. Sir vanda physics teacher. WBC. Okay, W for wheat, B for barley. All of you tell thanks in the chat box. Come on. Tell thanks to sir. Quickly. Okay, tell thank you, sir. He came running to give a trick. Okay. Oh, World Biology Conference, it seems. You people don't know conference spelling. Now I have to write conference spelling. Huh? Oh, you told it too. I did not see. So sorry. Okay, you people all told. I missed it. Yes, thanks, sir. Very good. Vijay, sir, video will be next week. Monday only. Monday. Okay. Yes. Very good. Awesome. Okay. So, students, WBC is the example. So, PUBG, WBC. Okay. PUBG, WBC. Now, PUBG culture, WBC will reduce. Okay. Here. BB house, Big Boss house. How many of you watch Big Boss house? You are not supposed to say. Okay. If your parents see, they are going to scold you. I am not responsible. So, students, BB house, Big Boss house na continuous எப்பவுமே Sunday da. Okay, not that Sunday, fight. Okay. Perisperm, persistent nucellus. When persistent nonsense or fight will happen, that house is called as BB house, Big Boss house. So, adu madri persistent nucellus, that is called as perisperm. Okay, persistent nucellus is called as what? Perisperm. Okay, va? So, example one, the black pepper and beet. Adanala da BB house I told. Black pepper and beet. Okay, understood? Okay. Okay, this is done. Students, everything in the seed, okay, has a ultimate job. Okay, that is your nucleus cells is there. If they are going to be persistent, we call that perisperm. Okay, integuments are going to become the seed coat, testa and tegmen. Then we have micropyle end, okay, which will be still an opening in the seed so that germination, oxygen, water transport can be facilitated. Adukapra students, here you see, if nucleus is persistent, we call it BB house, BB house, beet and black pepper, beet and black pepper. Clear? Okay. Okay. Beetroot. Understood? Okay, this is done. This is the ultimate goal of everyone. Okay, fertilized ovule on the seed agum, ovary on the fruit agum, ovule on the seed agum. Simultaneously, it is going to happen. It's not like first one will form then. So imagine, okay, imagine Aradhana is eating apple. Okay, first one the fruit formation acha. Adukapram as Aradhana is eating and eating, seed formation aguma illa. Both will happen simultaneously. So when you are buying a fruit, seed also will be there, fruit also will be there. Okay, yes. Okay. Okay, next one, integuments is the seed coat. Wall of the ovary will form the pericarp. Wall of the ovary will form the pericarp. Okay, va? Okay. Types of fruit, students, false fruit and true fruits. True fruits are true students madri. Who will subscribe, who will support, idu madri, they will be, always entertaining, talking, and the energy when they maintain bunny complete class la, they'll be thinking, ayo papa, my teacher is standing and teaching for this much time, I should also be energetic. You are true students. Some are false students who will just sit like this, they will not like, they will not subscribe, they will not talk, they are false students. Okay, so adu madrita, true fruits ovary will be developed from the so basically ovary should develop to the fruit. That is why ovary wall is forming the fruit wall. In some scenarios la vandu enna nadakkum students, nama thalamus illana other parts of the fruits like other parts will become the fruits, like apple. That is called as false fruit. Clear? Integuments provide protection for the ovule, and the integuments it will become seed coat, testa and tegmen in the seed. Okay, and it will protect the seed later. Okay, va? Thank you. Thank you, Dikshita. Okay, done. Drupe fruit, again you will have to study here. This is understood. This is understood. Students, I told you micropyle will remain open so the oxygen and water can enter inside. Otherwise, the seed will germinate. How? Have you noticed in some scenarios, the seed will lose 10 to 15% of their water content and they will be sleeping like this. They will go to dormancy. And when the condition becomes favorable, they will start, giving rise. Okay, they will start giving rise. Clear? That is called as dormancy. Okay, that's called as dormancy. Last topic, parthenocarpy. Fruit developed without fertilization. Such fruits are called as parthenocarpic fruit. Banana is one example. That means without fertilization, if I have to form a fruit, and the fruit is parthenocarpic fruit. Okay, parthenocarpic fruit. Clear? Hey, stop talking, da. Listen to the class. Now I'll scold you people. Then don't cry. Okay, see, although seeds in general are products of fertilization, a few plants, such as in some species of Asteraceae grasses, they have evolved a special mechanism to produce seeds without fertilization. This is called as apomixis. Okay, so number one, the fruit without fertilization, parthenocarpy. Seed without fertilization is called as apomixis. Okay, va? What is there asking? Apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg formed will be going without reduction division. That means, students, the egg cell that we saw, no? It will not undergo meiotic division. It will directly form a seed. So, obviously, that will be without fertilization. Okay? Without fertilization. Yes. Oh, yeah. Coffee. Yeah, I should order. Okay? Here, Polyembryony students, polyembryony na normally one embryo will be there, but in some scenarios many embryos will be there. How this many embryos will be formed? The nucleus cells which are on the outer, no, they will start protruding inside, telling that I want to be, they will protrude inside and they will form many embryos. That condition is called as polyembryony, which you see in citrus fruits. So I want all of you to do one thing. Once you finish everything, now make sure that you're reading the NCERT and you're making a small box with all the example. Parthenocarpic example, false fruit, true fruits, apomictic example. Ella example mein you have to write it in a box and keep. Yes, Sanjay, yes. Okay, students. Panchan Maheshwari 1904 to 1966 was the author here. So please remember, last time you remember, no, scientist question had come. He worked on embryological aspect, embryological characters in taxonomy. He also worked on artificial culture of immature embryos. He also have a fellowship of Royal Society of London, FRS. Okay, FRS. Remember his name. Okay, remember his name. Okay, now ready for questions? Okay, after this human reproduction I'll do. Okay, human reproduction chapter next we will start. First you do questions. Let me see how many of you listen to the class. Okay, I want quick answers. Okay, no time to waste. Quick answers. Then only we will go to the next chapter. Otherwise, I'll have to sleep in the office. Okay, where is the questions? Okay, start. First question. Which of the following is diploid? Which of the following is diploid? Okay, think and answer. Which of the following is diploid? He's tired of telling his name to everyone. He has taught me also. He gave me imposition. Still, I didn't study. Okay, we have a lot of students. Amazing. Okay, amazing. Make sure you are part of the channel as well. Okay. Come on, yeah, we didn't reach the target. So poor. So poor. Yes, answer. Ready? Okay, let's go. Yes. Good boy. Okay. Answer is going to be A. What is secondary nucleus? What is secondary nucleus? Nucellus. Nucellus, da. What is secondary nucleus? Everyone are telling C. Everyone are telling C. Did I use the word secondary nucleus anywhere? Have I used the word secondary nucleus anywhere? I have used the word nucellus. One person told C. Everyone copy pasted C. I know you people copied. You people copied. Okay? It's okay. Next one. Stop copying and do your own thing. Mature embryo sac in angiosperm is. Without copying you do. I know you people know the answer, but you don't want to copy paste. Just copy paste. I just now told you nucellus will be 2. That is why that fellow can become the megaspore mother cell. Correct? No? Answer this properly. Very easy. Yes. Like this you should answer, not copy paste ulta pulta. Okay, 7-celled, 8 nucleated. Okay, 7-celled, 8 nucleated. Next question. Synergids have a special structure. Synergids have a special structure to guide the pollen tube. What is the answer? Synergids have a special structure to guide the pollen tube. What is the answer? Yes, I can see the answers here also. Yes, good job. The answer is going to be option number A, filiform apparatus. Very good. Next question. The fusion of male gamete with a secondary nucleus forms. Okay. Secondary. The fusion of male gametes with secondary nucleus forms. Okay. Here we should write polar nuclei. Okay. The secondary nucleus, polar nuclei. Polar nuclei. Yes, easy question. A or C? Why are you giving me options? I am giving you options means you are giving me options. Huh? Male gamete will fuse with the polar nuclei. What will be the answer? What will be the answer? Now I understood. Oh, you people are having confusion here. Ma'am, secondary nucleus is a polar nuclei. It is diploid. No, it is not diploid. It is actually one haploid and one haploid. So we cannot technically call it diploid because fusion has not happened. So adanale nucleus is the correct answer. This is haploid. This is haploid. This is combination of haploid and haploid cell. So together we cannot call them diploid. At one shot you cannot call them diploid. So they are not typical diploid. Okay, they are not typically diploid. Okay, wow. I understood this confusion will come now. Not typically diploid. They are haploid cells which are staying together as a single cell. So adanala nucleus only will be the answer. Okay, nucleus only will be the answer. Okay, now you come back here. The fusion of male gamete with the secondary nucleus forms, obviously what will it form? Endosperm, right? That is called as what? Triple fusion. This process is called as triple fusion. Okay, triple fusion. Next question. Which layer develops into the pericarp? Which layer develops into the pericarp? Just now I taught you. You people are sitting and talking that time. Just now I taught you. Which layer develops into a pericarp? No, think properly. Don't make mistake. Think properly. Okay, think. What did I tell you? Integuments form the seed coat. Integuments when the seed coat formation nadakum, students. Okay, yes, very good. Ovary wall is the answer. Integuments when the seed coat formation nadakum, students. Seed coat formation. Nucleus will continue, that is perisperm, that is pericarp. In the perisperm, endosperm and the nourishment. So ovary wall. Next question. Which of the following statement is incorrect with respect to microsporogenesis? Microspore mother cell undergo meiosis to form microspores, which is incorrect. Microspores are haploid and arranged in a tetrad. Tapetum directly give rises to pollen grains. Pollen grains mature with microsporangia. Okay, fast. Can you believe it's almost 2 hours 45 minutes and we didn't even feel it, right? Did you feel like it was so long? I don't think so. I didn't feel it at all. I thought like only 1.5 hours got over. So that is the thing. Okay, next we will do human reproduction. We will learn it so nicely. Okay, we will learn it so nicely. Yes, no ma'am. See, I told you. Didn't feel it. Yes, very good. Tapetum does not directly give rise to any pollen grains. Adu vandu nourishment kudupanga. So adu the end of the story. Okay, very good. Next question, students. Oh, big question. Which statement correctly describes the double fertilization? Describes double fertilization. One male gamete fuses with the egg to form a zygote. One male gamete fuses with two polar nuclei to form a triploid primary endosperm nucleus. Double fertilization is observed in both angiosperm and gymnosperm. Triple fusion involves 3 male gametes. Don't spam. You can put the options once. Okay. Ilana, you will only not understand. Okay. Yes. One male gamete fuses with the egg to form a zygote. Okay. One male gamete fuses with the two polar nuclei to form triploid primary endosperm. Double fertilization is observed in both angiosperm and gymnosperm. Triple fusion involves 3 male gametes. Very easy question actually. Question long da irukku, but ana answer kandu pudikadukku easy. Which statement correct? Which statement is correct, you should tell. Yes, yeah. Who is that? I can't read your name. Soumya. Yes. Organism and population is there. Environment is not there. Environmental issue is deleted. Yes, very good. The answer is going to be 1 and 2. Okay, because these two are correct. These two are wrong. These two are wrong. Okay, next question. Identify the incorrect match. These are all questions made from NCERT line. So I put the NCERT line and then I made the question using Ved. Okay, I used Ved to make the questions. Okay, when Ved is there, why to worry? Okay, yes. Identify the incorrect match. Chalazal, basal part of the ovule, funicle connects the ovule to the placenta, hilum, junction of ovule and embryo, micropyle, entry of the pollen tube. Identify the incorrect match. Yes, correct. Junction of ovule and embryo, incorrect match. Hilum is not a junction of, it is funicle and ovule. Okay, very good, very good. Okay, next question. Assertion reason. Wind pollinated flowers produce large showy petals. Reason, they rely on insect for pollen transfer. So funny question. Hema, then what you have to do is you just scroll down and then see YouTube option is there. Because students, if you're part of any batch, test series, pro batch, prime batch, you will get all the 4 features for free. Anna, if you are a YouTube students, you will get only lecture lens. Lecture lens only will be visible for you. So, adanala and the Ved app download panni, you scroll it down, you will see YouTube. And the time nama channel select pannita, any part of the video you have doubts, you want practice questions, you can ask. Okay. What is the answer? Yes, both A and R are false. Wind pollinated flowers do not produce large showy petals. And why will wind pollinated flowers rely on insect? What? Okay. Yes, sure. Yes, sure. Aradhana, you can go dear. Okay. Yes. Which one of the following correctly describes polyembryony? Okay. Last question. After this, we will start the next chapter. Okay. Which one of the following correctly describes polyembryony? Okay. Aradhana, come back and later comment. Okay. What happened? Terminal batch. Oh, okay. Yes, very good. Development of multiple embryos in a single ovule. In the multiple embryos, students, eppadi form agum because of a nucellus. Nama nucellus vandu oru nuisance achu, and the nucellus vandu it will protrude inside and it will form multiple embryos. Okay, va? Okay, this is the last question. Human reproduction, lot of questions are there. I'll make you solve later. Okay, so many questions are there. Around 300 slides are there. Okay, we will start with human reproduction. Okay, all of you stretch for 1 minute. Okay, stretch for 1 minute and then we will start, because again a very big chapter, but don't worry, we will learn it very simple and easy. Okay, we will not complicate it, we will keep it very simple. Simple, humble, dimple. Okay. Ready? Yes, come on. Ready? I want to see the ready in the chat box. Okay, ready in the chat box. Yes, ma'am. On your marks, get set, go. The madri irukanam. On your marks, get set, go. Yes, definitely. OVR, definitely. Because no, sometimes we look at NCERT, but Anna, we don't understand which is important. So when you write side notes, it will always work. Okay. Okay. I hope all of you have marked your attendance. Okay, that's how you mark your attendance, and then also subscribe to the channel. Okay, yes, start Banlama students. So look here, nama reproductive events in humans include formation of gametes and the process nama enna koopruva gametogenesis. And sperms in males and ovum in females, transfer of sperms into female to the genital tract. And the process is insemination, we call it. Okay. And fusion of male and female gamete is called as fertilization, leading to the formation of zygote. This is followed by the formation and development of the blastocyst and its attachment to the uterine wall, which is called as implantation. Embryonic development, that means the 9 months period when the baby is developing or the fetus is developing, and the process gestation we call it. And the delivery of the baby, delivery we say casually, and the delivery of the baby we say what? We call it as parturition. Okay, we call it as parturition. Okay. Now, male reproductive system. Nama male reproductive system la vanda primary sex organ irukku, secondary sex organ irukku. Primary na enna students? Primary na enna? Romba important ana oru velai pandra aalu. Primary. Okay, nama primary sex organ vandu students, it is going to do two main jobs. Enna ma'am? Gametogenesis and production of the sex hormone. Gametogenesis na enna? Production of the sperm, and it is also going to release the sex hormone. Okay va? Then secondary sex organs na, it will not involve in gametogenesis, ana side by side whatever helps is required, other things it will be helping. So it is accessory ducts, accessory glands and external genitalia. So they are going to all help for the fertilization, insemination to happen properly. Okay va? Yes? Okay. Now look here. Testis. Listen to me carefully. From here I want full concentration. We will do this chapter very clearly. Okay va? So students, see here. Testis vanda, it is an extra abdominal structure. Extra abdominal. Ana, it's not inside the body, it is outside the body. In a pouch. And the pouch ku enna koopiduvanga? Scrotum. எதுக்கு மேம் இது வந்து extra abdominal இருக்கு? Why is it not present inside like ovary? Because students, in the scrotum, this testis, for it to function, for it to form the sperm, it has to be present extra abdominal in a pouch, because it requires a body temperature that is 2 to 2.5 degree less than the actual body temperature. So அதனாலதா இந்த testis வந்து extra abdominal இருக்கு. It is present outside the body in a pouch called as scrotum, which decreases the temperature by 2 to 2.5 degrees Celsius. Okay va? Now, in the scrotum one, the students, it is slightly pigmented compared to the other body parts in male. Okay. Now, in some animals, it is intra-abdominal. In some animals, what is happening? Testis one, it will not be outside, it will be inside. Like in elephants, dolphins, whales, all of this. Okay. Elephants, dolphins, whales, it will be intra-abdominal. Now, scrotum is not just helping in decreasing the temperature. They also help in students providing favorable condition, also helping in maturation, storage, production and survival of the sperm. Clear this much? Okay, yes, 10 more, very nice. Now students, if you look, let's say, look at the scrotum, you will notice that the scrotum is divided into like two structures like this. That means it has a median ridge right at the center. Okay, or a median ridge which divides it into two, like or a single pouch da, ana that single pouch la or a division which is the median ridge, which will divide it into two pouches. Okay, two pouches. Okay va? Understood this much? Now, main part. Ma'am, testis when the starting line, it is extra abdominal. The answer to that question is no, it was not. First, number testis when the students, when it was formed in a male, like how we do know gender, boy and a girl baby. So, testis is formed in the 7th to 8th week of the pregnancy. So, pregnancy period le, and the 7th to 8th week le vanda nama testis formation nadakum. Okay, testis formation nadakum. Ana, adukapram students, and the testis one, it is going to be inside. Okay, it is going to be intra-abdominal. Okay. Now, during the 7th to 8th week, 8th month, 7th or the 8th month, we can say 7th month also. During the 7th to or 8th month gap, students, and the testis one, it will descend from the abdomen to the scrotum. Enna nadakum students? Namma intra. Thank you, butterscotch. Huh? Okay, the testis வந்து intra-abdominal இருந்துச்சு. That was formed during the 7th and the 8th week of the pregnancy. Ana, during the 7th and the 8th month of the pregnancy, in the testis வந்து it will descend down to the scrotum. Understood? From intra it is becoming extra. Okay, it will descend down to the scrotum during the 7th or let's say 8th month of the pregnancy. Okay, now you should be thinking, now the testis is going to be extra abdominal. Ipa testis on the students, extra abdominal. Now you will be thinking, ma'am, ma'am, it will descend. How will it come through the abdomen to the scrotum? Students, there is going to be a canal which is called as the inguinal canal. And the inguinal canal the reason where the scrotum on the descend agum to the, testis on the descend agum to the scrotum. Ma'am, what will happen to the inguinal canal after that? In the scrotum and already descend ache, now it has become extra abdominal. What will happen to the inguinal canal? Students, through this inguinal canal, your spermatic cord will come. In the spermatic cord is the reason why you have the vas deferens. Okay, all these structures will be formed again through the spermatic cord. So here the spermatic cord formation will happen after this is, that means it will become completely vascularized. Blood vessels will all start coming and it will start having the spermatic cord. So that inguinal canal will later lead for that. Is this clear to all of you? Is this clear to all of you? This much is done and dusted? Okay, understood? Share the video with all your friends. Ask them to join if they have not studied the chapter. Okay, okay, done ma'am. Okay, now students, testis will be connected to the scrotum with a gubernaculum. Okay, testis will be connected to scrotum with the help of a gubernaculum. If you can remember, good. If not, it's okay. Okay, now this condition is very important. Cryptorchidism. Cryptorchidism is a condition, students, where the testis would have not descended from the abdomen to the scrotum. In the condition it is normal. Okay, in the normal situation na, in babies, when the doctor notices this, the doctor will tell, let's wait for an year or so, or let's wait for few months, maybe the testis will descend. In case the testis doesn't descend, then we'll have to go for other medical surgeries and other things. Okay, clear? Surgeries and other things. Testis is connected to the scrotum with a gubernaculum. Okay, understood? Connection, the connection, testis is connected to the scrotum with gubernaculum. Understood? Cryptorchidism, testis has not descended from the abdomen to the scrotum. That condition is called as cryptorchidism. Gubernaculum. Okay? Understood? Yes, you want to copy, you can copy. This dartos muscle, I'll tell you right now. I'll tell you right now. Okay? Okay, like don't like break your head. Just understand, like now imagine abdomen, from the abdomen testis is coming towards the scrotum. So in the gubernaculum vandu oru madri guide rope madri, students. It will like guide. Okay, it will be like a rope. It will guide the testis to come till the scrotum. So that is the gubernaculum. Okay, gubernaculum. Okay, gubernaculum. Okay, cryptorchidism, testis have not descended from the abdomen to the scrotum. Mostly the doctors will tell wait for some time. In case that doesn't happen, they will go for surgeries. Okay, next one, students, two types of muscles are present. Two types of very important muscles are present. First one is called as dartos muscle. This is present in the wall of the scrotum. Idu paakumbodhu, see, it's present in the wall of the scrotum. Okay, va? Then just beneath the skin, smooth muscle it will be. What does it do, students? It causes wrinkling of the scrotum skin, helps to regulate the temperature of the testis by reducing the surface area when it is cold. So under regulation in the dartos muscle vande help agum. Both dartos muscle and the cremaster muscle both are going to help in the regulation of the temperature. Okay. Second one, students, cremaster muscle, which surrounds each spermatic cord and testis. That spermatic cord I told you, no? Now, raises or lowers the testis for temperature regulation and protection. You can just remember the name of the muscles, dartos muscle and cremaster muscles. Okay. Now, just look at the structure, you will understand it. So, students, here you can see epididymis, okay, testicles, tunica, albuginea, vaginalis, we are going to come see. Spermatic, cremaster muscle, can you see cremaster muscle? Then you will have dartos muscle. Dartos muscle is here, cremaster muscles is here. Okay, and then the scrotum skin is there. Scrotum skin is there. So this is the spermatic cord. Can you see this is the spermatic cord through which this only the testes have descended. Okay, testis has descended. First it was called as inguinal canal, then the spermatic cord formation happened. Okay, clear to all of you? Pamniform plexus is a group of veins. Okay, group of veins. Okay, now this you understood. Now here two questions can come. What is the length of the testis? What is the width of the testis? Length of the testis is 4 to 5 centimeter. Width of the testis is 2 to 3 centimeters. Okay, understood? Oh, be careful. Okay. Now students, நம்ம வந்து cross section of testis பார்க்கலாம். Okay. Cross section of testis வந்து ரொம்ப easy students, don't stress. Okay. Okay, understood? Understanding gone to peak it seems. You should get me also. Deekshita, this is not fair. Students, Deekshita is eating alone. I am hungry too. Okay, even I want. Okay, even I should order coffee. Yes or no? She's eating alone. All of you are eating alone. I feel bad. Okay. Okay, we'll order during the break. Let's complete. Okay, look here. Nama cross section of testis. Okay, we are taking a cross section of testis. Okay, ba. And the cross section students, first layer வந்து நம்ம tunica vaginalis. எதுக்கு மேம் இந்த இது எல்லாம் மிக்ஸ் ஆச்சு மேம்? Yes, tunica vaginalis நம்ம testisல தான் இருக்கு. Okay, so this is the tunica vaginalis. Okay, tunica vaginalis. And அதுக்கப்புறம் students second layer வந்து tunica albuginea. Tunica என்ன? Albuginea. In the tunica albuginea வந்து slow it will move inside and it will start forming இது மாதிரி septum students. It will start forming என்ன form ஆகும்? Like this septum will be formed. Septum will be formed. In the septum formation நால we are going to see what? We are going to see compartments. Testicular lobules. Testicular lobules are going to be formed, or else we can say compartments. Okay, compartments. Testicular lobules or compartments. If students look here, tunica vaginalis, tunica albuginea, and then again septum formation will happen. That septum formation will lead to compartments which are called as testicular lobules. 250 compartments will be there in one testis. How many will be there? 250 compartments. 250 compartments. Okay, clear or not? Okay, yes. Now look here, in each of the compartments students, around 1 to 3 seminiferous tubules are going to be present. What is going to be present? Seminiferous tubules. In the seminiferous tubules, the main person who is going to do sperm formation. Main person who is going to do sperm formation. Okay, va? Seminiferous tubules. If students, from the seminiferous tubules, in the tubes irukla, in the tubes, and the tubes tube na கூப்பிடணும். So we will call these tubes as tubuli recti. Okay, no talking. Listen to me carefully. Listen to me carefully. Okay. Thank you, Suresh. Thank you, Pushpalata. So sweet of you guys. Okay, look here. Students, this is our seminiferous tubules. Seminiferous tubules in there will be extension. This madri. Okay, there will be extension, and the extension ka nama tubuli recti na கூப்பிடுவா. Okay, now this tubuli recti will all open into one larger portion which is called the rete testis. Larger portion which is called what? Rete testis. And from the rete testis there will be tubes which are arising. Tubes which are arising which can be called as vasa efferentia or efferent ductiles. Okay, now they will all open into a tube-like structure which is called the epididymis. What do you look there? Epididymis. Clear? Okay, understood or not? Epididymis. Now students, in the epididymis la vanda 3 regions are going to be there. Nama young students cap wear pannum head la thane. So the head region of the epididymis is called as the caput. Okay, caput. And then students, body nama newspaper la we see corp dead. Okay, corp dead na dead body na artham. Okay, so corpus na body, adukapram cauda na tail. Okay, caudal tail, like that is then a cauda na tail. So what has happened? Testicular lobules are there, inside the seminiferous tubules are there. Seminiferous tubules will open with tubuli recti to rete testis, rete testis to vasa efferentia, vasa efferentia to a tubular structure which is called the epididymis, epididymis to vas deferens. Is this clear? This kind of diagram you need to understand. Okay, that beautiful. I have drawn the diagram. If you don't understand, then what is the use of my diagram? Huh? What is the use of my diagram? I'm like, okay, ma'am is sitting and coloring, but I'm not understanding. Did you all understand this much? Don't distract her, guys. It seems, let her complete. I am your student or you people are my student, I doubt. Understood? Yeah, no talking about food for next 1 hour. Whoever talks about food is a cockroach. Like that we will tell. Okay? We play, in smaller classes like that. Okay, done. Now students, idhukku oru flow chart irukku. After the sperm formation, and the sperm vandu eppadi ma'am, it will move. Okay, it has to come till the external genitalia. How it is going to be moved? So understand, formation vandu seminiferous tubules la nadakum. Okay, from seminiferous tubules it will come to tubuli recti. From tubuli recti it will come to rete testis. From rete testis it will come to efferent ductiles. From efferent ductile it will come to epididymis. Epididymis to vas deferens. How will I remember this, ma'am? You will remember Shraddha Kapoor's new movie is there, no? Stree, and the horror movie. So like that, will you remember? STREE. Okay, Stree. That is seminiferous tubule, tubuli recti, rete testis, efferent ductiles, epididymis, and V. Okay, Stree V. Clear? Okay, understood this? So what is the trick that you will remember? Stree. Streena and a woman. Streena and a woman. Okay, avladha. Stree. So seminiferous tubules, tubuli recti, rete testis. So some of the teachers will skip this and they will tell the rest also. That is also fine. But since I made a flowchart, I told you everything. Okay, I told you everything. Okay, see, look here, students. Same thing here. Vas deferens is there, epididymis is there. Can you see? This is the head region, this is the body, this is the tail. Okay, and then you have, this is the testis, this is the lobules, you can see. Okay, inside the seminiferous tubules are there, and this is the testis part. Okay, efferent ductiles. Okay, efferent ductiles. Here, this one, efferent ductiles is also called as in some books it is vas efferentia. Okay, vas efferentia. Okay, clear? Thank you. Okay, now moving forward. That is clear, no? Testis understood, cross section understood. Very easy. Tunica vaginalis, tunica albuginea, septum, testicular lobules, 250 compartments, seminiferous tubules, tubuli recti, rete testis, epididymis, vasa efferentia or efferent ductiles, epididymis, then vas deferens. Okay, vas deferens. Now students, this is the structure given in your NCERT. Now this and all we will learn, then I will come back to the structure. Okay, this we will learn, then we will come back. Now, if I take a cross section of the seminiferous tubule, what are the things that is going to be there? Students, if I take a cross section of the seminiferous tubules, 2 types of cells are going to be there in the seminiferous tubules. Which type of cells, ma'am? In the cells kata, namma vanda spermatogonia cells we will call. Adu vanda namma male germ cell. Microsporogenesis we saw how important this pollen mother cell is. This is also very important. This is the spermatogonia, which is the male germ cell. Okay. Between the spermatogonia, students, there are a set of cells which are called as Sertoli cells. And obviously these are cells. Another space irukume. Okay, cells, when they are there, space irukum. That space is called what? Interstitial space. And the interstitial space also there will be cells, students. That cells are called as interstitial cells or Leydig cells. Interstitial cells or Leydig cells. Clear? Crystal clear. Awesome. So, spermatogonia, male germ cell, Sertoli cells is nurse cell. Who is it? Nurse cell. Okay, so this Sertoli cell, student, has a set of functions. Yenna ma'am functions. Sertoli cell vanda nourishment it does. Okay, it does nourishment. Obviously nurse cell na nourishment panniye agana, so nourishment. Adukapram it releases inhibin. It releases what? Inhibin. It will inhibit the FSH. Yedukku ma'am, FSH shouldn't do overstimulation. So, because it shouldn't do overstimulation, adinalada it will release inhibin, telling that inhibit the FSH. Because hormones control pananam, students. If you don't control the hormones, then it is a problem. So, inhibin will inhibit the FSH, and the Sertoli cells releases ABP, androgen binding protein. Okay, I'll tell you the use of it, and it also releases anti-Mullerian factor. for like boys and girls, the characteristics have to be there, no? So Mullerian duct is going to help in the secondary sexual characters of female. So adu vandu nama opposite ah pananam. So anti. Okay, anti-Mullerian factors. Now students, what is this ABP? Androgen binding protein. Nama Leydig cells vanda, they are actually endocrine in activity. Endocrine in activity means what? They are going to release hormones, ma'am. Correct? No. So which hormones they are going to release? They are going to release testosterone. In the testosterone vanda nama interstitial space la irukku. In the interstitial space la irukra testosterone eppadi ma'am nama seminiferous tube like inside, how do we add it? So adukku da nama ABP, this androgen binding protein will concentrate, okay, take the testosterone and it will concentrate the testosterone in the center, because that is where spermatogenesis should happen. Clear? Thank you, Spurti. Understood? Understood or not? Okay. Yes. Understood? All of you? Yes, understood? This much is clear, right? Any doubts, ask me now. Okay, any doubts, ask me now. I don't think any doubt will be there. This is super duper understood, I believe. Okay. ABP again, ma'am. Thank you, Muppidi. Okay, so many guys have come, but I don't know. All of you are not regular students, so I don't know all of your names. I'm learning your names, okay? ABP na students, androgen binding protein. Our one the main job enna in the testosterone one the interstitial space la வந்து release ஆகும், because in the Leydig cells வேலை என்னன்னா in the endocrine activity. So அதனால அது வந்து testosterone it will be releasing in the interstitial space. Now what is the use of keeping it in the interstitial space? I need it here because this is where spermatogenesis will happen. So அதனால I have to concentrate this testosterone here. For that your ABP will be doing the job. It will do high concentration of testosterone. Understood? Okay? Understood? So it will increase the testosterone. It has to take the testosterone, pick it up from here and put it in the center, because then only concentration of ABP, that is testosterone, will happen, because testosterone is required for spermatogenesis. Clear to all of you? This is understood ABP? Again you need explanation. So students, this is the diagram. Just look at it. Here this is the interstitial space. Here Leydig cells will be there. These Leydig cells are going to release testosterone, which has to be taken right in the center. Sertoli cells, it will release inhibin. It will have anti-Mullerian factors. Actually Sertoli cells have at least 5 to 6 functions, but ana adu konja adhigam, so adanala teachers do not teach you that much, okay? But these 3 functions are important: inhibin, ABP and anti-Mullerian hormone, okay, factor. Okay, so this is the entire flow chart. So you can revise using this notes. I have already shared this notes on Telegram channel. We Need Tamil Nadu. You can join the Telegram channel. Okay, okay. We Need Tamil Nadu. Clear? Leydig cells or interstitial cells. I told you endocrine portion of the testis present in the seminiferous tubule. Avar vandu enna பண்ணுவாங்க students? Androgens. Okay, they will release the testosterone. Seminiferous tubules highly coiled, present in the testicular lobules. Sertoli cells and male germ cells are present in them. Okay, here you can see. Sertoli cells or substenticular cells found in between the germinal epithelium cells. Single elongated cell, they provide nourishment. They secrete androgen binding proteins and concentrate the testosterone in seminiferous tubules. Whatever I told, thank you Arati, whatever I told, it is in this one box. Whatever I taught you so far, it is in this one box. Okay, so when you are revising, this will help you. This is also again just to help you, because many students will get a confusion between Sertoli cells and Leydig cells. When I was in your class, like when I was in your age, I used to get confused with Sertoli cells and Leydig cells, and trust me, I did not have a teacher who used to explain it like this or draw like this and teach me. So adanale I had no clarity on the chapter. Okay, but now I want to make it better. I want to correct the mistakes that my teachers could not do because of any reason. I want to be a better teacher for all of you. So adanale students, always remember, yes vanda Sertoli cells na support பண்றாங்க, nurse பண்றாங்க, support பண்றாங்க. L na Leydig cells lead பண்றாங்க. Okay, and the testosterone release பண்ணி lead பண்றாங்க. Okay, clear? Ayo Pushpalata, I am studying dear. Okay, I am studying. It will take a lot of time. Okay, so inside the seminiferous tubule, FSH, follicle stimulating hormone, it will be stimulated by main function, support, nourishment, regulation, inhibin, androgen binding protein, anti-Mullerian hormone. Other functions also are there. Inga vanda testosterone release students, and here also other functions are there. Okay, understood? Okay, understood. Now look here. This is what I just made you understand. Okay, this is what I just taught you. See, rete testis, vasa efferentia, epididymis, vas deferens are the accessory ducts. The seminiferous tubules of the testis will open into vas efferentia through the rete testis. The flow chart and the three one you have to remember. Okay, now look here. The epididymis leads to vas deferens that ascends to the abdomen and loops over the urinary bladder. It receives the ducts from the seminal vesicle, opens into the urethra as the ejaculatory duct. These ducts store and transport the sperm. So, number one, the next we have to study about accessory glands. Okay, accessory glands. This is a, what is that? This is the easy way of studying the diagram, drawing the diagram. Okay. Okay, so urinary bladder, seminal vesicle. So இதுதான் நம்ம seminal vesicle. இது வந்து paired இருக்கும் students. இது வந்து prostate gland. Okay, அதுக்கப்புறம் bulbourethral gland. Okay, bulbourethral gland. இது வந்து நம்ம urethra, penis, testis, epididymis, foreskin, glans penis. I will teach you all of that. Okay, slowly I will teach you. Okay, understood this? Okay, next வந்து நம்ம accessory ducts and accessory glands. இது ரொம்ப important. Listen to me carefully. Accessory ducts in our students, நம்ம rete testis, vasa efferentia, epididymis, vas deferens. இதுல வந்து you don't have to break your head. இது வந்து ரொம்ப easy. Ma'am, I already studied 3, ma'am. So from this 3, I have to remove the tree and then it will become the, okay, then it will become the accessory ducts. Okay, இங்க வரும்போது தான் important. Accessory glands. நம்ம sperm வந்து, it is going to be formed. But ஆனா அந்த sperm வந்து lubricate ஆகணும். And the sperm வந்து until it reaches that female tract, it should be able to move. So அதனால நம்ம வந்து certain secretion add பண்ணணுமே. அதுதான் இது. So those accessory glands are going to help in the secretion. That is, seminal vesicle one, the students, 60 to 70% it was going to. So basically, that lubrication which I spoke about is called as the seminal plasma. That lubrication that I spoke about. Okay. Thank you. Okay, so accessory glands one, the students, seminal plasma, and the lubrication one, it's called as the seminal plasma. And the seminal plasma composition le 60 to 70% one, the seminal vesicle is only adding. 60 to 70% when the seminal vesicle only is adding. Okay, so in 60 to 70% is added by the seminal vesicle. Then it is going to also have fructose. It is going to have what? Fructose in it. Okay, inositol also is going to be there. Many things are there, and also prostaglandins and fibrinogen. All these are going to be there in the seminal vesicle secretion. Clear to all of you? Seminal vesicle secretion. Fructose is going to be there. Prostaglandins is going to be there. Fibrinogen is going to be there. Understood? Okay, understood, right? So students, this prostaglandins is actually someone who is going to help the sperm motility, sperm to move better. Okay, it will help in the sperm, like it will, let's say, like it will increase the efficiency of sperm motility, fertilizing capacity, adu madri. Like mucus coating and all, it will work, the prostaglandin will work. Because once this seminal plasma enter into the female, this prostaglandin is going to do the job of uterine contraction, help the sperm to pass through it. So it basically increases the sperm motility, okay, enhances the sperm motility. Clear? The arrow mark indicates sperm motility. Adukapram students, nama prostaglandins, okay. So now you should remember prostaglandin is the only single fellow among all the accessory glands. Other two are paired. Other two are paired. They can ask you question also, which is paired, which is unpaired. So seminal plasma vandu paired ah irukku, prostate gland vandu single ah da irukku. Prostate gland vandu students 20 to 30% of the secretion add panniranga. Ana and the secretion mainly edukku ma'am, alkaline pH maintain pandradhukku, because nama males la vandu there is urinogenital duct. The urethra is going to act as a urinogenital duct, where the urine also is going to come, sperm is also going to come. Urine is acidic, so other neutralize pandradhukku da your prostate gland is going to release alkaline, so maintain the pH, okay, and also it is going to release some calciums as well. Now ma'am, what will bulbourethral do? 30% here happens, 60% here happens, 100% is over. Students, bulbourethral or the Cowper's gland is also paired, pea-sized gland. It will help in lubrication and again alkaline pH for neutralization. Okay, next level Josh. Okay, next level Josh. Clear? Understood this much? Bulbourethral, Cowper's gland. Okay, now we will go back to the diagram and we will see, because NCERT diagram is important. They can ask you questions. Oh, why is this moving? Students, something happened. I don't know where I am. I am here. Is the notes good? Is the notes good? So here you have ureter. Here you have seminal vesicle, okay, it looks like this, okay, seminal vesicle. Then you have urinary bladder, okay, vast difference is there. Prostate gland, look at it students, prostate gland is like this, one gland it is, okay. Penis is there, urethra is there, glans, penis, foreskin, I'll teach you. Ejaculatory duct, rectum, anus, bulbourethral gland, also called as Cowper's gland. Cowper's gland is only this much, very small pea-sized gland, okay, very small pea-sized gland. In the diagram this person has drawn also, you can see it very clearly. Bulbourethral is a pea-sized gland, prostate gland is one gland like this, and then we have seminal vesicle, okay. Okay, seminal vesicle. Now coming back, this also you understood. Rich in fructose, calcium and certain enzymes, I told you. Now students, inga paakumbodha, the penis is a male external genitalia. All of you know this. It is made up of a special tissue that help in erection of the penis to facilitate insemination. The penis when it enters the vagina, insemination nadakanume, that means sperm has to be released. So for that, the penis should have the erection, okay, that means the sperm has to come out of the penis. For that, they have to have the special type of tissues. Now the enlarged end of the penis, okay, the enlarged end of the penis is called as the glans penis, which is covered by a loose fold of skin, which is called the foreskin, okay. I was about to say sunscreen, but then yeah. Okay, so this is the foreskin students, this is the skin, this is the glans penis, okay. The enlarged portion is called the glans penis, the side the skin is there, that is called the foreskin. Okay, now look here. In the male reproductive system also, there is ampulla. Wherever ampulla word is there, remember that it is wider. It is wider. Okay. Now look here, students. This your urethra actually has three parts. Okay. The first one is called as the prostatic part. This is the part where the prostate gland placement is there. Then you have a membranous part. Then we have the penile part, which is bulbous. Okay. That is the largest part. Now, enlarged portion in the penis is called glans penis. Retractable fold of skin is called as the foreskin. Is this understood? So the entire male reproductive system is here. Okay. Yes, foreskin also can be removed for surgery, like it can be removed using surgery, and certain religious people do that, like they remove the foreskin as a part of their religious thing. Okay, like that many kind of operations can be done, surgeries can be done. So students, entire whatever I have taught you is in one page. So whenever you are doing quick revision, use this page to revise the whole thing. Trust me, nothing is missed. Actually, everything that I have written is here. Okay, because I've read this also. I've read your NCERT, I've read this, I've watched, you know, all the animation videos, there are doctors who teach us. I am nerd like that. Doctors are there. So those videos also I have watched. So make sure that you read through this. Okay. Clear? Yes. Okay. Now next one, students, we are going to the female reproductive system. Okay. Just give me 1 minute. I will just order something hot because my throat is aching too much. Okay. I will just get something and then we will continue. Okay. Any doubts you have, please let me know. Oh my God. Yes, any doubts you have so far? Okay. Yes, ma'am, run. I'm not running anywhere. I'll stay here only. But then I just want to get something. Students, everything is cold. Cold, cold, cold. Okay. Done, done, done. Okay. Let's continue. So sorry to keep you all waiting for 1 minute. Okay. Okay. Continue. Yes, ma'am. Okay. Now look here. The female reproductive system, students, it has a pair of ovary, it has oviduct, uterus, cervix, vagina and an external genitalia. Okay, now here you should understand that the structural and functional. So other than that, there is also mammary gland. Mammary gland also, students, they are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth and childcare. Now, whenever a woman, okay, whenever a woman is going through her menstrual cycle, she feels a pain in the mammary glands, okay, or the breast region. Why is it? Because it is to indicate that the menstruation is happening, ovulation is happening, fertilization, because everything is a connected process, right? If the baby is born, then there should be colostrum in the mammary glands, so milk production should happen. For this, everything has to be connected with each other. Clear? Connected with each other. Any doubts you have in the male reproductive system, please let me know, okay? Now students, look here. Each ovary is about 2 to 4 centimeter in length. That means very smaller than that of the testis. Testis is 4 to 5 centimeter. And students, they are attached to the pelvic wall and the uterus by ligament. There are ovarian ligaments, suspensor ligaments. All these ligaments are going to help in attachment. Okay, all these ligaments are going to help in attachment. How will I give you guys treat? You people are all, I'll come to Chennai, that time I'll give you treat. Or anywhere in Tamil Nadu, I'll come, that time I'll give you treat. Okay, look here. Female reproductive system, there is primary sex organ and secondary sex organ. Same thing. Primary sex organ is going to help in oogenesis, right? Primary sex organ is going to help in oogenesis and also release of ovarian hormones like estrogen and progesterone. Same thing, gametogenesis, production of sex hormones, okay? Now, secondary sex organ, students, fallopian tube, oviduct, uterus, cervix, vagina. Okay, fallopian tube, oviduct, uterus, cervix, vagina. These are all the secondary sex organs. With that, we have external genitalia, which is the vulva, and mammary gland. Mammary gland. Okay, ovary is around 2 to 4 centimeter, attached to the body by ovarian ligaments. Okay, attached to the fallopian tube with ovarian ligaments. Okay, clear? Okay. Now let's. Okay, now students, we are going to understand how is the structure of the entire female reproductive organ. So here you look here, this is the structure of it, students. Here you can see you have tube like structure here, which is your fallopian tube, and then here you have a swollen region, which is again corpus of the entire the uterus or the womb. Now this uterus has 3 regions. This region is called the fundus. then we have the corpus, and then we have the cervix. Now students, the tube, the fallopian tube also, has 3 regions, okay, 3 regions, clear? Now you people are seeing, okay. No, nothing like that, nothing like that. All of you saw this one now, okay. So here, what is happening? How does the unique spiral artery, I can't read the rest, artery stay without, okay, stay without ischemic injury? Because you know the spiral arteries are there, no students, it is very coiled and it has the ability to uncoil, like what do you say, it can extend and it can come back because of that, okay. And second thing, it is mainly because it is under progesterone control. So because of that, without injury, these people can do it. That is the answer, okay, because they have the ability to do rapid regeneration even during the endometrium tearing without causing any ischemic injury. I think Sarvesh, that is your question, I believe, okay. They have a quick, we say reperfusion, that means they can extend and come back, so without much, you know, without much damage to be caused, okay. I hope that is clear. And since it is attached to the basal layer, no, the shedding will not happen in that layer, no, so they are safe to go, okay. Now students, look here, this is your fallopian tube, okay, this is your entire uterus, you have fundus, you have corpus, you have cervix. Now the tube region has mainly 3 parts, okay. What is it? It is the infundibulum, ampulla, isthmus. The fallopian tube has what, students? Infundibulum, ampulla, isthmus. Now infundibulum, just below the infundibulum, you will notice what? You will start seeing fimbriae, finger-like structures which will hold, okay, which will hold this ovary in place. Like that it will look like, the fimbriae will be there. So this ovary will be there, fimbriae will be there. Then you have infundibulum, ampulla, isthmus, which is present in the fallopian tube. Clear? Yes or no? Understood? Now look here. Here students, you can see the ovarian ligaments, okay. Ovarian ligaments will be present, which is helping the ovary to attach to the fallopian tube. Here you have the cervix and the vagina. Now the cervical canal plus the vagina, we can call it as the birth canal, okay, birth canal. Now students, this question has come in one of the years, that is from the ovary, okay, what is the fallopian tube? You have to remember infundibulum, ampulla and isthmus. That is from ovary, infundibulum, ampulla, isthmus. Which is the region closer to the ovary? The question answer will be infundibulum. Infundibulum, clear? Okay, and which is the region closer to the uterus? If the question comes, what will your answer be? Isthmus, ampulla, infundibulum. This question, please, just make a note in the NCERT right now itself. Please make a note in the NCERT right now itself, otherwise you will forget. You will forget. Okay, make sure that you are, oh God, my throat, yeah. My throat is crying. Understood? Understood. Party for 11K. My God, you people. Yeah, I'll give you party when I come there. Okay, when it becomes a little more bigger, I'll give you. Don't worry. Okay, yes. Done? Okay. Now, look here. If I take a cross section of the ovary, okay, if I take a cross section of the ovary, then what is going to be the situation? Students, if I take a cross section of the ovary will have two regions, one cortex region and one medulla region. In the cortex region la vandu nama follicles ella present aayirukkum. Ana and the medulla region la vandu nama blood, vascular nerves adella vandu ready aayirukkum. So this is the cortex region, this is the medulla region. This cortex region will have the follicles, medulla region will have the blood vessels and everything. Okay, clear? Now look here. This is the follicles. This is the follicle region where the cortex region is the follicle region, and here we have the blood vessels and nerves, all of it running. Now you can see primary follicle, secondary follicle, tertiary follicle, graphene follicles, corpus luteum, corpus albicans. These are the layers of the follicle. Now students, nama ovary la nama secondary oocyte vandu release agume. That has to be taken. No, that will be taken with the help of abdominal ostium, which is present in the fimbriae, near the fimbriae region. Okay, yes or no? Clear? So the secondary oocyte will take it through the ostium. That ostium is called as abdominal ostium. Abdominal ostium. Clear to all of you? So the graphene follicle will rupture, it will release the secondary oocyte, and that is when ovulation will happen. Pathway involved in, ma'am, ignored. What did I ignore? Students, that message is gone. Pathway is there. Wait. Order I should teach, no? What chuma I'll teach or what? Order I should teach. Wait, Madi. Okay? Wait. All of you wait. Yes. This you understood? All of you? Okay, let me just go and see your... Okay, what Nandini, you are asking me, ma'am, what if the ovary has water bubbles? Will insemination not happen? Yes, there are chances, because this water bubbles that you are saying, no, that is actually follicles. Okay, those are the follicles which have not matured properly. Any other issues have come because of that. So this mainly happens when a person is going through PCOS, that is when a woman is going through PCOS. So adanala there are chances that ovulation does not happen, because no release of egg will happen. So yes, the ovary, if it has multiple water bubbles, then there is chances that they do not ovulate, fertilization does not happen. That can happen. Okay, my God, I had to go back to the chat and see the questions. Okay, now students, see here, each fallopian tube is around 10 to 12 centimeter long, and from the periphery of each ovary to the uterus, the closer part of the ovary is called as the infundibulum. This question will come, which is closer to the ovary, which is closer to the uterus. Okay, this also we did. Now students, remember the layers. Okay, the layers of your fallopian tube. First one, you have the endometrium, then you have the myometrium, then we have the perimetrium. In this, your endometrium is the shedding layer. Sorry, first perimetrium, then myometrium, then endometrium. This is the layer that is shedding continuously during every menstruation, where it is building, then shedding. Where it is building, then it is shedding. Okay? Building and shedding. And this also, fallopian tube parts, infundibulum, ampulla and isthmus. Fimbriae is the finger-like structure. Cervix is there. Cervical canal plus vagina is called the birth canal. Okay, birth canal. Clear? Okay. Here also students, the uterus is a single, also called as womb. This is the shape. So remember, ovary is almond shape. Uterus is inverted pear shape. Inverted pear shape. Okay, wow. The uterus is single and it is also called the womb. This is called as an inverted pear shape. In the uterus, students, it opens into the vagina through a narrow cervix. Adu vandu open agum through a narrow cervix, and the cervix is called as a cervical canal. The cavity is called as a cervical canal. Along with the vagina, it forms the birth canal. External thin membranous perimetrium is there, a thick myometrium is there, and a glandular endometrium is there. And the endometrium shed eye continuously, your menstruation will happen. Okay, myometrium students, they are going to exhibit strong contraction during the delivery of the baby. Is this clear? Any doubts so far here? Okay, now external genitalia. External genitalia. Okay, Podala. No students. Okay. Okay. Now look here. This is the external genitalia, the vulva. Okay, the vulva. So students, this region which you are seeing here, this is your mons pubis. On the mons pubis, there is going to be hair-like structure which is called the pubic hair. Okay. Now, this is the external genitalia. What you're seeing here, you have two main like lips-like structures we say, okay, upper lip and lower lip, like that only we say. Like it is just because the structure framing is like that, okay. So, like that two layers are there. First one is called as labia majora. This is the labia majora, and inside you have labia minora, okay. Two things will be there, students. Labia majora, this one, and this is the labia minora. In this labia minora, you will be able to see that there are certain openings. Like first you have the urethral opening, adukapram you have the vaginal opening, and then students there is a gland called as vestibular gland or Bartholin gland, which actually helps in lubrication. Okay. What happened, Deepak? You people are texting something, I can't read, students. Okay. And here, students, we will have the anal opening. So, here you have the urethral opening, okay, for the urine to come out. Inga vandu nama vaginal opening irukka. Inga vandu nama Bartholin gland irukka, which is going to help in lubrication. And here you have a special structure which is the clitoris, which is actually having the erectile sensitivity. Just like in males, the females also will feel certain like that emotional or sexual behaviors. And the time clitoris is going to help. Okay, clitoris is going to help. So this is the structure of external genitalia. Any doubts you have, please let me know right now. Okay, right now. So here you can see labia majora, a fleshy fold of tissue which extends down from the mons pubis, surrounds the vaginal opening. Adukapram students, labia minora, which are paired fold of tissues under the labia majora. Opening of the vagina is often covered partially by a membrane called as hymen. In the hymen, the controversial topic. Edukku ma'am? In the hymen is considered as a skin which is actually telling about the virginality or the virgin, if the woman is a virgin or not. But in the hymen one, the students, because of any reasons like horse riding, cycling, or tampons, or active participation in some sports, because of any of this hymen can be torn. Okay, if the hymen tears, that does not indicate the like sexuality or the virginality of the woman. Okay, so it is a completely wrong misconception that it is an indicator of virginality, which is not true. In the hymen, like in some women at a very younger age only it will be broken. In some women, because of the exercise that we do, because of wearing menstrual cup, tampons, it can tear. Okay, it can tear. Okay. Okay, break you people want, I'll give you. Break, I'll give you. Okay. Okay, any doubts so far here? This you understood, okay. Vulva you understood, labia majora, labia minora. Next one, the students, nama mammary glands tha. Okay, mammary glands also, it's again a part of this. Yes or no, it's a part of this. Correct? Yes, ma'am. So, okay, after a break you will do. Okay, fine. Then you take a break here. Just now we finished a break, no? No, we took it at 6, na? So fast time is going, yeah? We'll take okay, be back in 15 minutes. Okay, or should I finish it off? Yeah, PYQs are there. Yes, Bartholin gland or vestibular gland. Okay, 10 minutes is enough. Okay, 10 minutes, like by the time I go down, it will become 15 minutes. So you can resume back in 15 minutes. After coming back, we have to do oogenesis. Okay, spermatogenesis. Okay, folliculogenesis, everything is there. So much is left. Okay, but we'll finish. Fourth day question did not come. How come? Told the team to do it. Wait, let me check. Okay. Hello. Yeah, this Nandini is one big problem. Huh? She's like, ma'am, come, ma'am, come, ma'am. You don't drink water, ma'am. Water and all you can drink tomorrow. Okay, mammary glands, let's start. Okay, mammary glands. Okay, mammary glands here. See, students, first of all, this mammary glands, when you see this region, this region, you will start seeing the fat. Okay, both the regions, fat will be there. Right at the center is where you will have the mammary ducts and everything. Okay, mammary ducts and everything. So remember here, your glandular tissue, okay, glandular tissue will be present, which will have around 15 to 20 mammary lobes. Okay, whatever you are seeing here, no, these are the lobes. These are the lobes. Inside these lobes, what will be there? Your alveoli will be there. So in the alveoli, where the milk production will happen. So here, alveoli, where the milk formation will happen, the cells of the alveoli, milk formation will happen. Okay. So here, alveoli, okay, from there the milk production will happen and then it has to come. So then you have mammary ducts, then you have an enlarged portion called ampulla, then you have lactiferous duct, and ultimately the milk will come out through the nipple. So all doctors are loyal is the trick to remember. Okay, all doctors are loyal. A for alveoli, D for doctors, that is ducts, mammary ducts, A for ampulla, L for lactiferous. All doctors are loyal. All of you also are loyal, no? Okay. All doctors are loyal. Time up, where that time up? Which time up? You're playing game or something or what? Like some competition is happening. Okay. Students, day 4 question I have posted, okay? My team people forgot. So, like something happened. They had some other work, so that's why. So, because of that, okay? Hi, Suvra. Okay. So, look here. All doctors are loyal. So, alveoli, ducts, ampulla and lactiferous tubules, and then it will come out through the nipple. Okay, it will come out through the nipple. So, this is where the formation happens. It will form in the cells of the alveoli. Okay, it will form in the cells of the alveoli. One glandular tissue, okay, in the breast will have divided into 15 to 20 mammary lobes containing cluster of cells called alveoli. Milk formation will happen. The cells of the alveoli will secrete the milk, which is stored in the cavities or the lumen of the alveoli. The alveoli open into mammary tubules. The tubules will open into mammary ducts, then a wider ampulla region, then lactiferous ducts, and then nipple. Okay. No VLC class. Yeah, this is not a trick that I found. This is a trick, it is already there. Okay, some tricks are just already there, students. You don't have to waste energy on that. Okay, we don't have to waste energy on that. Yes, whatever I taught you so far, everything is right here. Okay, this will be more visible in the PDF. Ipa vandu idu visible ah irukada because idu pixelate aachu. Ana nama PDF la vandu idu clear ah. Okay, it will be visible. Okay, next topic bolama. Next topic bolama. We'll be able to finish this chapter in an hour, I believe, with question practice. After that, we will be doing reproductive health in half an hour. Okay, reproductive health, we don't have to stress so much. Only question point of view questions we will be doing. Okay, 1 second, students. Okay. So, next is gametogenesis. Okay, gametogenesis. Namakku vandu spermatogenesis irukku. Adukapra oogenesis is there. The primary sex organ will produce gamete and then process is called gametogenesis. The process of producing sperm is called as spermatogenesis that begins at puberty. My voice is gone. Spermatogonia present on inside the walls of the seminiferous tubules will multiply mitotic division to increase in number. Each spermatogonium is diploid and it contains 46 chromosomes. Okay. Now, primary spermatocytes periodically will undergo meiosis. Now, this whole thing you will not understand if I read. Okay, you will not understand if I read. So, that's why we will write a flow chart. Okay, this is the flow chart. Yes. So, what is exactly happening? Namakku spermatogenesis nadakra site vandu nama seminiferous tubules. Okay, site enna irukum seminiferous tubules. When does it start? It starts at puberty. When does it start? It starts at puberty. Number spermatogonia cells one, the students. First it will undergo meiosis 1, which is a reductional division. Other couple it will undergo meiosis 2, which is an equational division. So in the reductional division la nama diploid வந்து haploid agum. In the equational division la and the haploid nama maintain pannum. Okay va? So here you see spermatogonia, which is 2n, will undergo the meiotic division, and first you will get type A spermatogonia and type B spermatogonia. Ana type A vandu pause agum, it will not continue. Type B one, the students, it will form the primary spermatocyte. What will it form? Primary spermatocyte. So inge vandu no reductional division has happened, only one mitotic division has happened. Okay, clear? Yes, yes, please write the flowchart in the corner of NCERT. I would be more than happy. I would be more than happy that you people are all writing. Okay, writing. Clear? Please write it. Please write it. Okay. The more you write flowcharts, it will always help. Okay, it will always help. Yes. Not clearer. Write it, write it. Okay, yeah. Look here. Spermatogonia cells. Okay, spermatogonia cells vanda, it will undergo meiotic division. Okay, because inge vanda we cannot do reductional division yet. We can only do reductional division later. They will do mitotic division and they will form 2 spermatogonia. Some places you will also see people only writing spermatogonia. That is also fine. Type A and type B is there, but then people write spermatogonium and just stop it there. So students, they will form type B, and the type B vandu continue agum. Type A vandu pause agum. Type A will not continue. So type B will continue. If you are getting confused here, just write one formation and just write one only. Okay, one only. Clear? Ma'am, today's question is awesome, ma'am. Yes, we have super duper questions, no, in the test series. Even I like it, okay. I have asked them to add some more biology questions as well, but then I think like they are, I don't know, they will add that as well. Okay, they'll add that as well. Clear? 1 second, students. I will just refresh this. Okay, yeah, cool. Okay, yeah. So look here, in case it's too confusing, you let it go. Otherwise, I'll tell you, students, nama oogenesis la vanda polar body, secondary polar body to maintain the chromosome number, adu madrita. This type A is actually a stem, like it's a stem cell pool. Okay, we say stem cell pool for the cells. Why so? Because these people will maintain and continue the mitotic division, and some of them will become the type B and they will continue the entire spermatogenesis. If it is too confusing, you leave it. You just remember that from the spermatogonia cells, we are getting a type B spermatogonium. From there you see, from here it is important. Okay, here it is important. First primary spermatocyte will be formed. It is not a reductional division. Adanala 2n and 2n will be maintained. Adukapram students, the first meiosis 1 will happen. Meiosis 1 the reductional division. So adanala primary spermatocyte is going to form secondary spermatocyte which has haploid. Okay, which has haploid. Okay. Then the secondary spermatocyte will undergo meiosis 2 and it will form spermatids. What will it form? Spermatids. Okay, from the spermatids, next step is going to be the spermatozoa, or else you can say sperm itself. Last ultimate formation, which is going to be haploid. Very, very simple. Spermatogonia cells, spermatogonium, primary spermatocyte, secondary spermatocyte, spermatids and spermatozoa. That's it. That's it. Okay. Now here, look here. Spermatogonia cells are embedded in Sertoli cells until maturation. Why? Because Sertoli cell is nurse cell. Nama vandu nurse cell kit or nurse, when we go to a nurse, they will want to take care of us. So Sertoli cells will not be ready to leave it. So Sertoli cells will take care of the spermatids and spermatozoa until maturation. So spermatogonia cells are going to be embedded. Idu madri it will be embedded, and then the spermatozoa also will be, the head of the spermatozoa also will be embedded in the Sertoli cells. Then they will have to leave from there. If they don't leave from there, they cannot reach the external genitalia. So they have to leave from there. Okay. So this Sertoli cells will be helping like for nursing. It will want to nurse. Okay. Now look here students, important thing. From your spermatogonia till your secondary spermatocytes, we call that process as spermatocytogenesis. What we call it as? Spermatocytogenesis. Okay, write it in your NCERT and keep. And when the spermatozoa is leaving, okay, when the spermatozoa is leaving, we call it spermiation. That means from your spermatid to spermatozoa, we call it what? Spermiation. Is this clear to all of you? Is this clear to all of you? Yes or no? Quickly in the chats. I want all of you to write this and keep, and otherwise, gone. You will forget. You will forget. Okay? Spermiation is the process by which your spermatozoa will leave the Sertoli cells. Okay? They are ready to leave here. That is your spermiation. Spermiation. Then spermatids to sperm. Okay? Spermatids to sperm is your spermiogenesis. Okay? Students, this actually, the writing, the way I have written is wrong. So, this will be this. Okay? This we can write it as spermiogenesis. Wait. The way I have written. Okay, spermiogenesis. Okay, spermatids to sperm, spermiogenesis, and when these people are leaving the Sertoli cells, we can call spermiation. Correct? Yes or no? Okay, you're writing. Okay, right. Okay. Okay, understood? Understood? So, spermatogonia. Then your type B spermatogonium, primary spermatocyte, secondary spermatocyte, this entire process is called as spermatocytogenesis. Then we have this spermatids to sperm, adavanda nama spermiogenesis, and when the spermatozoa leaves the Sertoli, nama vanda spermiation. Okay, spermiation. So this is the three terminologies that you have to remember. Okay, adukapra students, for this gametogenesis to happen, நம்ம வந்து hormones use பண்ணி, we have to use certain hormones. Okay, we have to use certain hormones to do the entire process. Like example, hypothalamus will actually stimulate the GnRH, that is gonadotropin releasing hormone, that will actually tell the anterior pituitary that please activate this FSH and LH. In the FSH one, the students, it is going to already inhibin. You already know, no? It will help in spermatogenesis. Follicular stimulating hormone. Now, this inhibin will send a negative signal. Inhibin's job to inhibit the FSH. Okay, so inhibin will send a negative feedback mechanism. Don't worry about that. LH, luteinizing hormone, it will release the testosterone, which is very, very necessary for spermatogenesis. Clear? Clear? Nandini, I have already taught this Leydig cells and Sertoli cell difference. I had given a box. I think you joined late. Okay, you check it out once I share the notes. Okay, once I share the notes. Any doubts here? Any doubts here? So here you can see finally released from the seminiferous tubule. So after spermiogenesis, sperm heads become embedded in Sertoli cells and are finally released from seminiferous tubules, which is called as spermiation. And here the spermatids are transformed into spermatozoa by the process called as spermiogenesis. So three processes: spermatocytogenesis, spermiogenesis, and spermiation. No genesis is happening. That means it is leaving. Okay, tata bye bye, it is telling and leaving. Spermocytogenesis, it is forming something. Spermiogenesis, forming something. Some genesis is there. Spermiation, leaving. Okay, spermiation is leaving. So here students, what is happening? Where is it? Here. ICHI is interstitial cells. Okay, interstitial stimulating hormones. Here your Leydig cells are there. No, luteinizing hormones are there. No, this luteinizing hormone, one more name is this ICSH. I had made you write last time. No, haven't I made you write? I told you last time also. ICSH you have to write. Okay, understood? Okay, now here, any doubts here? Students, the sperm plus the seminal plasma together, we call it as what? We call it as the semen. One ejaculation will have 2.5 to 3 million. One ejaculation will have 2.5 to 3 million. That is around 200 to 300 million sperms. And the 1 million of 1 ml la vanda 100 million sperms irukkanam. Okay, normally 1 ml la 100 million sperms irukkanam. Ana 1 ml la 20 million illana adukkum kammiya irunduchena, then that person has oligospermia that will lead to infertility. That will lead to what? Infertility. Now look here, at least now how do I know? Like how do I know this whole thing happens? The entire process takes around 74 days. Anna students, please remember, this is your seminiferous tubule. Every part of the seminiferous tubule will be forming the sperm. So adanale, if this is already formed and released, this will go through the entire cycle again. So that is why that 74 days you don't have to you don't have to worry. Okay, you don't have to worry. Clear? You want to write it? You want to write it? Interstitial cell stimulating hormone. You want me to write it here? Students, nothing. It is basically another term for Leydig cells. It has come in one of the 2023 or 22 paper. That is why we have written it here. Okay. It is called as interstitial stimulating hormone. Okay, Leydig cell, other term. Why? Because it is present in the interstitial space. So adanale namakku vande interstitial cell stimulating hormone. This is the other name for Leydig cell. That's it. You don't have to worry. Okay. Interstitial cell stimulating hormone, another term for Leydig cells. Ma'am, why you are using that term? Because this is the interstitial space layer, so adanala I can call it that also, right? There is no hard and fast rule that I should not use it. Here students, understand. Now, how do I know that the sperm is going to help in fertilization 100%? I will get to know if the sperm is able to do the fertilization or not if the 60%, at least 60% of the sperm has normal shape and size. Because students, out of all the sperm that is formed, everything will not have proper shape, size, tail and all of that. Some of them will be completely gone. So, adanale, at least 60% of the sperm the normal shape and size. Among the 60%, nama 40% it should do vigorous motility. It should do vigorous motility. Then I can say that okay, sperm is successfully going to reach and fertilization is going to happen. Okay, va? Okay? Yes, understood? Here you want any more explanation? No, I hope, right? This also understood, na? Here, hypothalamus, gonadotropin releasing hormone, anterior pituitary will get activated, FSH will be released, which is actually going to help for spermatogenesis. Ana inhibin release, it will send a negative feedback mechanism telling that enough of your secretion, don't do overstimulation. Okay. And then students, LH is the Leydig cells, which is also called as interstitial cell stimulating hormone. Same thing. Okay. And it will release the testosterone. Okay. Testosterone. Now, this is also done. Yeah. Now, the structure of the sperm structure, students, please understand. This is called the acrosome. This is the head region, which will have the nuclear part. Okay, the nuclear membrane. I mean, not the nuclear membrane, the content. And then, students, here you have the neck region, which will have proximal and the distal centrioles. Proximal actually helps for cleavage, and distal will be the one that is helping for the tail formation. Okay, proximal cleavage you will understand later when I teach you fertilization. In the neck region, proximal and distal is going to be there. This is the middle piece, and in the middle piece there will be lot of mitochondria, powerhouse of the cell for the energy production, and then there is a tail structure. This is what your gameto, this is how your sperm is going to look like. Okay, this is how your sperm is going to look like. Clear? Structure of sperm is understood? Okay. Okay, now I'll teach you oogenesis, oogenesis and folliculogenesis, okay, because this two topic has to be taught simultaneously, otherwise you will not understand. Okay, now students, I told you, nama ovary is a almond shaped structure, and the almond shaped structure there is a cortex region, adukapra oru medulla region, and the cortex namakku follicles present aayirukku, and the follicles in the secondary oocyte release agum. But ana in the folliculogenesis and oogenesis You have to understand it together. Illana, you will not understand it. So look here. This is your follicle. Follicle is basically, see, girls love to dress up. Girls usually like to deck up, dress up, look very good, okay, do their hair. So same thing goes with our oocyte as well, or our ovum as well. They also like multiple layers around them for protection. So adu madri da inga vandu nama primary oocyte kedaikum students. First what you will have? Primary oocyte. This primary oocyte will be covered with granulosa cells. It will be covered with what? Granulosa cells. That is your primary follicle. That is what? Your primary follicle. Adukapram you will get your secondary follicle, but ana idile there is only primary oocyte. There is also going to be primary oocyte, just that the layers formation is going to become increased. You will start seeing theca cells as well. So inga vanda granulosa cell, inga vanda granulosa cells plus theca cell, and the layering vanda it is going to become additional. Okay, then students, this is the secondary follicle. Adukapram tertiary follicle. Eppadi ma'am na vanda in the tertiary follicle identify pandren students with the help of in the antrum, fluid filled cavity. When you see a fluid filled cavity, you should understand that, oh my god, it is a tertiary follicle. So here you can see granulosa cells are there. Then you have theca externa, theca interna. Two layers of theca will be formed. This is theca externa, this is theca interna. Adukapram a fluid filled cavity will be present, which is the antrum. Now, secondary follicle had primary oocyte, but your tertiary follicle will have the secondary oocyte. Now this tertiary follicle will develop to graphene follicle. Graphene follicle will rupture, release the secondary oocyte, and that is when ovulation will happen. Understood this much? Understood this much? Any doubts? Understood this much, or you have any doubt? Okay, now I'll teach you oogenesis. Understood, right? Okay, students, detailed lecture is already on the channel. Okay, under one shot playlist, detailed lecture is already there. If you're feeling like, oh my god, I want to study it slowly, you can watch the detailed lecture. Okay, detailed lecture. If you feel like, okay, no, I just want a revision, this is the best video to watch. Okay, okay, let's first understand oogenesis, then again I'll come back to folliculogenesis. So students, we have oogonia. Okay, we have cells called as oogonia. In the oogonia, cells will undergo. So here, one thing you have to understand. The gametogenesis will start at the embryonic stage. For males, it will start at the puberty. Males, it will start at puberty. And females, from birth, it is starting. And embryonic stage, start agum. So, in the oogonia, the embryonic stage, it will undergo meiosis. And here, it will form, sorry, it will undergo mitosis. And it will form the primary oocyte. Okay, primary oocyte. After that, it will get suspended. Meiosis 1 will be suspended at prophase. It will be suspended temporarily. So, in females, no. Students, they don't, okay, yeah. Females, no, they don't like saying yes or no very fast. How many of you have, you know, let's say you have asked something to your female friend, like, do I look good? Is my haircut nice? They'll be like, yeah, it is nice, but I feel like some color would do better. I feel like you should have just trimmed this portion. They'll never say you look good or not. Adu madrita in the entire process also. Instead of directly forming the secondary oocyte, girls will decide that I will do half of it in the embryonic stage. I will stop the process. I will do half of it in the childhood. I will stop the process. I will do half of it in the puberty. So, girls will never say yes directly. They will say like this, like this, like this, like this, like this, like this, like this. They will reach S. Adu madri da nama oogenesis. First nama embryonic stage la vanda we will form primary oocyte. And then when we were going to do meiosis 1, prophase stage la vanda suspend ago. Okay? Prophase, all the boys can relate. All the boys in the chat box is able to relate. Exactly. Okay. So, oogenesis is little complicated. So, look here. I'll make you understand. Don't worry. Okay. Oogonia one, the primary oocyte it will form. Okay. Primary oocyte in the embryonic stage. Adukapram students, meiosis 1 will get suspended at prophase. That means they were going to form secondary oocyte, but ana suspend acha. Then the suspension will be released when? Here. Childhood stage, slowly they'll be like, okay, let's think about it. And then in puberty, you will notice that this stage will continue. It will form the first polar body and the secondary oocyte. It will form the first polar body and secondary oocyte. Adukapramum ஒரு pause இருக்கு students, okay? One pause has happened in the embryonic stage. அதுக்கப்புறம் இன்னொரு pause in the puberty. That is, meiosis 2 will be suspended at metaphase. Meiosis 2 will be suspended at the, metaphase. Now the secondary oocyte is thinking what to do in life, okay? If a sperm comes in contact, then the secondary oocyte will become ovum. Understood? So first, oogonia was there. It formed primary oocyte. Then it was like, bro, I will become secondary oocyte. And the step la pause achu. That is meiosis 1 at prophase stage, diplotin stage. Okay, prophase stage. Adukapram childhood happened. Peace. Okay, I am suspended. Puberty happened. Puberty happened. Okay, puberty happened. It became secondary oocyte with the first polar body. Ana, before it could become ovum, or suspension. Where? At the metaphase stage. Okay, meiosis 2 suspended at metaphase stage. That means it is an equational division. Okay, equational division. My voice is gone, Black Panther. Okay, now students, if a sperm comes in contact, that means fertilization happens or sperm comes in contact, sexual intercourse has happened, then this secondary oocyte will become happy, telling that yes, I can finally become an ovum or an oocyte, and there will be a formation of a secondary polar body. Edukku ma'am in the first polar body, secondary polar body? Students, in the first secondary polar body vanda nama ploidy level maintain pandradhukku, because only one arrow is continuing. Only one arrow is continuing. You notice that, no? One side arrow is not continuing. So how do you balance the ploidy? To balance the ploidy only, you have first polar body and second polar body. Clear or not? Understood? So whatever the menstruation it is, the secondary oocyte, ovum will only form once the sperm has come in contact. Okay? Okay? Diplotin. Yes, yes. Look here. Meiosis 1 prophase, diplotin stage, the suspension will happen. Adukapram meiosis 2 metaphase 2 la vanda suspension nadakkum. Okay, now students, follicles vanda before birth we had around 6 lakhs. Okay, embryonic stage we had around approximately 6 lakh follicles. After birth we got around 1 to 1.5 lakhs. Okay, reduction happened. Adukapram nama puberty reach agumbodhu it became 60k to 80k. Okay, follicular atresia we say. Okay, follicular atresia. So idu madida idu continue agum. Clear or not? This is understood. Now I'll come back to folliculogenesis. Okay, folliculogenesis. Students, please understand, your polar bodies are just to maintain the ploidy. Okay, maintain the ploidy. Yes, yes. Who is that? Sabarish. Ma'am, first polar body have nucleus. Yes, it does, but I think cytoplasm is too less. Okay, non-functional cells, they are just there to maintain the ploidy level, because you can see only one arrow mark is continuing. So what about this N? Because in the diploid one the haploid ஆச்சு, so obviously இங்கே ஒரு N இருக்கணுமே, but ஆனா அது இல்லை. So அதனால அந்த ploidy level maintain பண்றதுக்கு தான் we have first polar body and second polar body. If you understood this, can we come back to this? So you can remember that first oocyte, primary oocyte is formed, then this follicular phase is going to continue, then only in the tertiary follicle you will see the secondary oocyte. Then that tertiary follicle will become mature graphene follicle. It will release the secondary oocyte, and that is when your ovulation will happen. Okay, that is when your ovulation will happen. So look here students, your oogenesis is a discontinuous unequal division. Polar body is there to maintain the ploidy level. Everything I have written. Okay, so here you can see. Okay, here you can see oogonia, each fetal ovary, prophase 1 of the meiotic division, temporarily it will get arrested, called as the primary oocyte. The primary oocyte then will be surrounded by granulosa cell, and it will form the primary follicle. Then the granulosa cells, above that theca cells will be formed, then it will be called as the secondary follicle. Theca cells will split into theca externa, theca interna, and then a fluid filled cavity will come, which is called the antrum. That layer is called tertiary follicle. Adukapram there will be rupturing, that is graphene follicle will form, it will rupture, release the secondary oocyte, by then ovulation will happen. Understood? Full one dance only I can do here. Okay, full one dance I will do here now. My god, my throat is gone. Okay, now we will come back to this. Okay, Yes, whatever we studied, let's go through a quick revision. I think we will be able to complete. Okay, look here students. At the puberty, spermatogonia is there? Yes ma'am. Mitosis differentiation will happen. It will form the primary spermatocyte. So 2n to 2n will remain adhe madri. Then primary spermatocyte, first meiotic division it will undergo. That is when you are going to get your secondary spermatocyte which are haploid. Correct? Yes ma'am. Second meiotic division na equational division. So whatever you formed in the secondary spermatocyte, adhe ploidy level da continue agu. Okay. And the ploidy level continue, you will form spermatids. Then spermatids will only do differentiation because namma sperm vanda, it should have a pointed structure or a tail irukkanam. Ana, namma spermatids vanda circle irukka. So there is no division that is happening but differentiation that is happening. Right, like let's imagine, okay, I want to become a model, okay, imagine I am becoming Golu, Molu I am becoming, okay, like I am becoming chubby, okay, I am becoming chubby, suddenly I want to become model, I will do differentiation and I will become a model. Adu madiri da. Spermatids vandu all circular it is. Sperm is like this. It will not move if it is circle. So adanale spermatids to spermatozoa differentiation will happen. Then the spermatozoa will leave the Sertoli cell. That process is called as spermiation. From here to here. Okay, from here to here. Where is it? Here. Sorry. From here to here. It's called as spermocytogenesis. From here to here. Okay, from here to here, spermiogenesis. When this fellow leaves, it is called as spermiation. Tata bye-bye. Okay, tata bye-bye. Clear? Now, what will happen? Okay. Oogonia, again, mitosis differentiation. Both the places, mitosis differentiation will happen. Okay. Adhikapra students, primary oocyte. Then first meiotic division will happen, completed prior to ovulation. This is the birth, childhood, puberty. Adhikapra secondary oocyte formation will happen. Secondary oocyte and first polar body will be formed. Then it will be like, okay, I don't want to do it. Yeah, I will get arrested. I don't feel like replying. I will not reply. Okay, because I am a woman. I will not reply. Okay, who are you to tell me to reply? You tell me. I will not reply. Then I will sit like this. Then if a sperm comes, then I will get activated. Then secondary oocyte will become ovum. This is what a secondary oocyte is thinking. Secondary oocyte is like, no, I want to get arrested. First, when did I get arrested? When I was a primary oocyte. I got arrested in the meiosis 1 prophase diplotin stage. After that, childhood happened, childbirth happened, okay, depression happened, puberty happened. Then I'm like, okay, fine, I'll form secondary oocyte. Then secondary oocyte formation happened. Then again, I'm like, oh, I'm bored. Yeah, I don't feel like replying today. So, Adanala, I will pause I will arrest it at metaphase 2 of like meiosis 2, metaphase 2. Then sperm will come. Then the secondary oocyte will become ovum. Okay. So adult reproductive life, the first polar, then it will form second polar body and ovum. Clear to all of you? Okay. Done students. Okay. I will become all India rank 1 if I study like this. Appa. Understood? Okay, understood? Now we have to study one cycle. Why is this chapter like this? Okay. When spermiation is when the spermatozoa is leaving the Sertoli cells, telling that enough of nursing, amma, let me go. Okay. So it will tell like that, it will go. That is called as spermiation. Okay. Verithanam. Okay, verithanam. Okay. Singapenne. That much I only know, students. Okay, every song I know only half of. Menstruation, shedding of the endometrium. Okay, menarche is the start of menstruation. Menopause is the stop of menstruation. Menarche can happen at any age. Like nowadays, age is different. Maybe our parents' time it used to happen in the degree time. Then it happened in PUC time. Now even in school students. Okay, school students also get it. So here. Menarche is the start, then the menopause is the stop. This is the only reproducible age, but in males it is not like that. Even when they are old, they can still form sperm. The sperm number, the count will reduce, but the sperm will be capable of conceiving. Okay, like that. So menopause will happen, students, 45 to 55 years. For women, that's all is the reproducible age. Actually, for women, it is even 30. Okay, 30 also they say. That's why they say you should conceive before 30 because after 30, lot of health issues will come. Okay. Thank you. Take rest. How da? Half midway of the chapter, how to take rest and go. Okay, look here students. Now I want you to understand a very important thing. There are two cycles that happens. Okay, there are two cycles that happens. First is called as ovarian cycle. Then you have the uterine cycle. So one thing will be happening in the ovary. One thing will be happening in the uterus. Why? Because by the time the Graafian follicle ruptures students and the secondary oocyte release agumbo, inga namma uterus la vandu ovulation nadakadam. Right? You should be ready for the pregnancy to happen or fertilization to happen. So, adhe madhida, adukapram menstruation nadakadame. So, the uterus should be ready for it. At the same time, the ovule should be ready for it. Okay? It looks like I am doing that Zumba dance. Okay? Dance. Okay? Clear? It could be harsha, it could be of lifestyle, hormonal imbalance, okay, mainly it is lifestyle, Students, my voice is gone, it's not my throat is gone, my voice is gone, so because of that kiki sound is coming. Okay, now, look here, uterine cycle, mainly your endometrium, glandular, it will become shedding will happen. That is what is happening. In the ovarian cycle, mainly the follicle development will happen. Students, one thing that will happen in both the cycle commonly is going to be ovulation. Secondary oocyte release and here ovulation will happen. Okay? Yes. Okay. Now look here. I got All India rank 45. It is a good rank. Yes, it is a good rank. But I don't know what are the gifts for each rank, students. Okay. Okay, let's start. Yeah, first this. Okay, now look here. Carefully listen. Okay. Ovarian cycle. What is happening in your ovary? Pre-ovulation. That means before your ovulation happens, follicular phase will be happening. That means primary follicle, secondary follicle, tertiary follicle formation will be happening. At that time, நம்ம uterusல வந்து என்ன நடக்கும் students? First 5 days and the menstrual cycle will happen. Okay, the periods we say no, menstrual cycle will happen. அதுக்கப்புறம் நம்ம endometrium வந்து positive ஆகி it will start going to a proliferatory phase. It will be like no, I want to do, I will become proliferatory. இது வந்து build ஆக start ஆகும். So proliferatory phase start ஆகும். அதுக்கப்புறம் in both the cycle நமக்கு ஒரு ovulatory phase கிடைக்கும் where the ovulation will happen. So இந்த டைம்ல Graafian follicle form ஆகும் and the secondary oocyte will get ruptured. Secondary oocyte will rupture and the Graafian follicle will rupture and secondary oocyte will be released and then ovulation. அதுக்கப்புறம் students post ovulation. Okay, அதுக்கப்புறம் வந்து post ovulation. Post ovulation ல வந்து corpus luteum will be present na. Corpus luteum will be ready to release progesterone, telling that yes, pregnancy is going to happen, So that phase is called the luteal phase. At that time, nama endometrium, it will become very positive. It will become secretory in nature. Okay, it will become secretory phase. Okay, yes or no? It will become secretory phase. So this is what is the whole cycle, students. Actually, this box makes it super duper easy. Now again, we can look here. Hormones also are going to play, no? So look here. In this stages, FSH is the important one. Follicular stimulating hormone. Why? Follicular stimulating hormone. Follicle formation ke help aga hormone will be FSH. Okay, wow. So students, follicle will be developing. Here mature follicle. So FSH will be gradually going. During this follicle development, these follicles are going to release estrogen. These follicles are going to release what? Estrogen. So here you can see the graph is slowly it is increasing. Now, when the Graafian follicle is rupturing, that time your luteinizing hormone will go to an LH surge. It will go to what? LH surge. Why, ma'am? Luteinizing hormone has to go to LH surge. Otherwise, ovulation will not occur. That is not exact 14th day. 13th day, your LH will be on peak. People tell that 14th day. But Anna, Nandini, do I look like a machine to you? Huh, Nandini? Do I look like one ChatGPT or like one AI tool I look like? I should only talk, no. So if you keep saying me fast, how will I do? Should have some, like, some basic sense. That ma'am is teaching continuously from past 4 hours. I am just sitting and listening. If you keep saying fast, obviously even I'll feel bad, no? So be patient. If you cannot be patient, then you leave the class. Then later you can come and attend the class. Okay? Even I am trying to make it faster, so your time doesn't get wasted. Anna, it will take time. It's teaching and it is 2 chapters which takes individually 5 and 7 hours to complete. If I take one shot, both the chapters, I take it minimum 4 hours, 45 minutes. So 9 hours chapter, I have to finish it faster. Okay, clear? Look here. So students, in the ovulation, 1 day before the ovulation, the LH is going to a maximum surge. Even FSH will be high, estrogen also will be high. Why? Because the follicles were secreting the estrogen, so adinale this is going to be high. Okay, adinale this is going to be high. Now ovulation happened, the secondary oocyte has come out. Ovulation will happen for all the phases. So at that time, students, here you can see when the follicle development is happening, here menstruation is happening. Then again follicular phase is happening. The body is like, okay, I want to build. Proliferatory phase will happen. Okay. Then what will happen? After the corpus luteum ruptures, it will form after the Graafian follicle rupture, it will form corpus luteum. Now these people are going to send estrogen, that is negative feedback will be sent, because of which estrogen will go down. Why? Because these people will be excited for progesterone. They are waiting for pregnancy to happen, so they will release progesterone. Okay, they will release progesterone. So this progesterone will start slowly increasing. Saying that pregnancy is going to happen. So in the corpus luteum will release progesterone, students, then corpus luteum will get converted to corpus albicans. By then here luteal phase will happen. Is this clear? Yes or no? Same thing here. Okay, same thing here. I have taught you again here also. Multiple times I have taught you in that class. Okay, in that detailed lecture. Okay, so here you can see, students, normally 28 days will be there. So the 14th day will be ovulation. 1 day before the 14th day, luteal LH surge will happen. Okay, and first these people are going to send positive, telling that we want estrogen, we are releasing estrogen. Ana in the stage level, the estrogen will be negative feedback mechanism. That's why you will see a dip of estrogen. Because these people will be excited for pregnancy, adinale they will release progesterone. Okay, clear? Okay, adinale they will release progesterone. So here you can see menstruation, follicular phase or proliferatory phase, luteal phase or secretory phase, and then the next cycle will begin. So students, this box is more than enough. Lot of images I have added. Okay, whichever image you want to see, you can add. This is the best thing to understand. Okay, this has everything. Okay, this has everything. Understood? Okay, this you understood, na? Again, I have drawn and taught you also. Okay, same thing. Positive feedbacks, then negative, because this estrogen will tell, and then this people will start increasing. Negative feedback will come, then this people will start decreasing. FSH and LH will start dropping after ovulation. Okay, FSH and LH will drop after ovulation. Clear? Hi ma'am, you are my motivation in studies. I am studying in 10th standard. Please say my name. Hi Kavya. Hi Kavya. I have one DNA student also with your name. Okay, next is going to be, students, ovum structure. I hope this is clear to all of you. Again, I am telling you, if you want to watch it slowly, detailed lecture, then you please go to one shot playlist. I have done detailed lecture where I have taught with around 5-6 slides. This is a fast revision, so one time only I will be teaching you that whole thing. Okay. Wait, students, I'll just put a Vicks chocolate in my throat, so like voice will become back to normal. Because after so many days, I am doing it at one stretch, I feel. Okay. What is happening? Ready? Okay, we are almost done. Okay, don't worry, we'll almost be done. Okay. Very good students who stayed. So proud of all of you. Okay, next is going to be ovum structure. Okay, in the ovum structure, students, look here. Namakku first oru layer irukku. Okay, see, first this layer is going to be called as corona radiata. So just imagine, sperm has more competition than anyone. Okay, it is not even close by to NEET competition. Sperm on the students, 200-300 million sperm. Sperm. Just one sperm will enter inside this, because the ovum will not, okay, ovum will not allow the entry. Okay, ovum will not allow the entry. Clear or not? Who is stubborn? Who is stubborn? Eda Vicks chocolate, give us chocolate, it seems. Vicks chocolate, see. Nothing these kids will believe. See, Vicks. You think I am like I'm eating chocolate, so I'm not giving you people, huh? You want my voice to be there, no, till the end of the class, or you want it to be like key and the radio madri. So adanale, okay? Okay, now start here, look here. Here students, corona radiata is the first layer, okay? Adukapram students, you have zona pellucida layer. In the zona pellucida layer is made up of a glycoprotein, okay? Adukapram, you will have the space which is there, which is called the perivitelline space. Then you will have the nucleus and obviously the plasma membrane. Now the sperm, all of this sperm will be waiting outside, telling that I will get the entry, I will get the entry. Anna, competition is so high that only one sperm will be able to enter. Now just imagine one of the sperm has started to enter. Immediately the zona pellucida will change the structure, like structure, students, composition, so that no other sperm will enter, because polyspermia cannot happen. That means many sperm cannot enter. So adanale or sperm vanda idu maadhiri zona pellucida touch ana polum students, zona pellucida will completely change. It will completely change so that no other sperm can enter. Now the sperm will move like this. It will go through these layers. It will reach the plasma membrane. It will release the nucleus membrane, okay, the chromosomes and everything, okay, the content, DNA content, and this both will fuse together, and that is how the zygote formation happen. So 23 chromosomes, 46 chromosomes, okay, 46 chromosomes. Who is talking? I'll remove you from the class now. Who is talking? Anyone who's talking? I will remove you people now. Okay, no talking. Okay, now look here. So the sperm is coming, corona radiata, then it will go to zona pellucida, then perivitelline layer, then the inside. Okay, by then the chromosomes will be released and they will fuse. Okay, they will fuse. Now look here. Who decides the sex of the baby? The father decides the sex of the baby. Anyone who is telling that the mother decides the sex of the baby, you should give one hittings to them. Okay. Father decides the sex of the baby because the woman will have only 23 plus X. Okay. Male will have 23 plus X and 23 plus Y. That means X and Y will be there. So it is the male who will decide. If let's say release of Y happens, then the boy child will be formed. Release of X happens, then the girl child will be formed. Female has only 23 plus X. Okay. 23 plus X. Okay, now we are going to start with fertilization. Fertilization vanda enna irukum students, sperm plus ova, same time, same location. That is, they have to reach the ampulla. Then only fertilization will happen. Okay, just because sperm is going this way, ovum is going that way, fertilization will not happen. They both has to come together in the same time. Okay, that is called as insemination, where the sperm will enter the vagina. Through the uterus, it will go to the ampulla region. Now, students, in the uterus, what this sperm will do, no? Very smart it is, okay? It will wait for 4 to 5 hours, telling that even I have demand. Okay, nothing like that. Okay, it will wait 4 to 5 hours. For what? For capacitation, that is where it will activate itself. Now, during this activation, many of the parvum sperms will die, students. Many of it will die. Okay, they would have come with full competition that yes, I am going to be the one. And now they will reach that. Like you people, no, you will run and you will reach till Jan and Feb, and then you will be like, oh, I want to give up, and then you will give up. Okay, till that you will be running, and then by the time you reach the peak time, you will be like, okay, I want to give up. Okay, adu madri tha. So here in the uterus, they will wait for 4 to 5 hours. That is called as capacitation, where they will activate themselves. Adukapram only they will enter inside. Okay, they will go to the ampulla region. Now students, look here. The sperm has certain things like hyaluronidase, that is which will dissolve the hyaluronic layer. Okay. Now acrosome will have sperm lysine. Sperm will have corona penetrating enzyme. Otherwise, how it will enter to the corona radiata? It cannot go and tell, excuse me, can I come inside? It cannot tell, no, it has to have a corona radiating enzyme. So it will go there and it has to move through the corona radiata. Then it will have zona lysine, which will allow it to enter through the zona pellucida. So for every layer, it has already its own enzyme telling that I don't want anyone's help, I will only enter inside. So inga vanda sperm, acrosome will have sperm lysine. Because students, and the tail will remove. Okay, by the time it comes in contact, tail will go. Okay, tail will go. Hari, you are not blocked, dear. You are not blocked. If you put continuously message, then the channel will put you on timeout. That's it. Okay, you are not blocked. I can see your message. Okay. Okay, done? So here students, what is happening? Sperm is coming. Okay, this will enter inside. The tail will go. Tail is not required anymore. No. So zona lysine, corona penetrating enzyme, hyaluronidase and sperm lysine. Is this clear to all of you? Yes. Now comes the actual story. Okay, everything I have drawn for you people, and still you people have not even told one thing in the comment section. Ma'am, your notes is nice. Nothing. Okay, nothing. Okay, look here. This is the ovum. Now the sperm has entered inside. What is going to happen? These both are fused. It will lead to the formation of zygote. In the zygote one, it will go through the cleavage. In the cleavage, your prostaglandins, which you remember seminal plasma, they will also help. Okay. So cleavage has happened, students. You got a 2-cell stage, then you got a 4-cell stage, then you got a 18 to 16-cell stage, which is called the morula. Adukapram, you are going to get a stage which is called the blastocyst stage. And the blastocyst stage, students, there can be also a cavity which is called the blastocele. Okay, blastoceles. You will get it in the description box. The notes you will get it in the description box. By the way, have you all subscribed? Yeah. Let me just check how many of you smash the like button and subscribe. You people will just come, tell hi, bye and you will go. Okay. Just tell hi, bye and then go. You won't subscribe, nothing. But hurry, competition is less. No, 29 students will fight for 1 seat. 29, approximate 29 students will fight for 1 seat. Here, Pavam, 200 million people are fighting. Okay, yes. So, student 8 to 16 cell, but then here they don't have relatives or family members to ask them. So, it is fine. Okay. So, here 8 to 16 celled morula stage, adukapram blastocyst stage will be formed. In the cavity, we call it as blastocele. Now, students, remember in this blastocyst stage, there will be two layers. Okay. One is called the trophoblast and one is called the inner cell mass. In the trophoblast will be the outer layer and this is going to be the inner cell mass. Now, remember this inner cell mass has the capabilities to continue and form all the organs. All the organs, okay? Yes, Dikshita you told, okay? Now students, look here. Monozygotic twins will be formed because of the splitting of the zygote, okay? Now, this zona pellucid. So students, how does this twin formation happen? Sometimes when the zygote outside zona pellucida layer will be there, okay? When zona hatching happens, zona hatching means where the zona layer, pellucida layer has to be removed. And the time by mistake the zygote can get split. That is monozygotic twins. Same to same, there will be no same gender, same look wise. That kind twins formation will happen, okay? These are the other layers, allantois, yolk sac, chorion and amnion, all this is there. Look here. First your sperm has entered, zygote formation, 4-cell stage, 8-cell stage, morula stage, blastula stage, blastocyst implantation. So blatocyst implantation, before it happens, the zona pellucida layer has to be hatched out, removed. That is called as zona hatching. Okay, wow. Now look here students, trophoblast, inner cell mass and the blastocele will be there. Now here, see here, the blastomere in the blastocyst are arranged into two outer layer called trophoblast and an inner layer called inner cell mass. The trophoblast there then gets attached to the endometrium and the inner cell mass will get differentiated into the embryo. Now it will go for what students? It will go for implantation. The blastocyst will start getting attached to the endometrium. By then, your endometrium has started building, proliferatory phase, it has built into many layers and in the blastocyst one it will get implanted. Adukapra in the endometrium will start covering, okay, because it has to give blood supply and everything, no, so it will start covering, okay, wow. Now look here, very important. After implantation, that is students, after 7 days after fertilization, like some people what do know, especially the women, after the sexual intercourse, they will immediately check for pregnancy, but they will not know because this implantation will only happen after 7 days after fertilization. So at least after missing the periods after 10 days, like after, if you miss the menstruation, after 10 days you have to do a test and see, pregnancy test, then you will be able to know because by then HCG will be known in your blood and in the urine. So because of that, you will get to know if the positive result will come. Okay, so implantation will happen 7 days after fertilization. Finger-like projections appear on the trophoblast, which are called as chorionic villi, which are surrounded by the uterine tissues and the maternal blood. The chorionic villi and the uterine tissues become interdigitated with each other and jointly form a structural and functional unit between the developing embryo and maternal body called as the placenta. So what are they saying? This is the placenta. From the placenta, your umbilical cord will stretch for the transport. So placenta is formed how, students? With the chorionic villi and the uterine tissues which are interdigitated to form the placenta. In the placenta vanda, okay, in the placenta vanda, they will supply oxygen, nutrients to the embryo and also removal of carbon dioxide and excretory waste materials produced by the embryo. The placenta will be connected to the embryo through the umbilical cord, which helps in transport of the substance to and from. Placenta also act as an endocrine tissue and they produces several hormones like human chorionic gonadotropin, HCG. So when HCG is high in the blood, okay, when HCG is high in the blood, it will, blood and urine, you will get to know if the results are positive or not. Okay, if the pregnancy is negative or positive. Clear? Now look here, students. Human chorionic gonadotropin, human placental lactogen, estrogen, progesterone. In the later phases of pregnancy, relaxin also will be released, which will do dilation and softening of the cervix. Why? Because through the cervical canal only baby, that birth canal, baby has to come. So we have to see the cervix is dilating or not. If the cervix doesn't dilate, the baby cannot come out. Okay? Okay? Clear? No food, nutrients. Okay? Now students, look here. This is the entire stage. Okay, you can see fetal growth and metabolic changes will happen. So these hormones are going to help for that. HCG, HPL, relaxin, estrogen, progesterone, all these hormones are going to help. Okay? And during pregnancy, we are supposed to avoid tetrogens, which are like nausea tablets. Okay, now HCG, HPL, students, see here, it will help for lactation, lactogen hormone. Okay, prolactin, milk formation, cortisol and oxytocin, uterine cortisol, oxytocin, uterine contraction, relaxin for dilating the cervix. Okay, yes, not gland, it has endocrine activity. We cannot call it a gland. Gland is what? Thyroid gland, parathyroid gland, pineal gland, pituitary gland, adada gland. But they can have endocrine activity. That means where they are releasing certain hormones. Okay, where they are releasing certain hormones. Is that clear? Corpus luteum, yes, I told you. Thank you, Stephen. Epiboly and emboly. Epiboly is where that layer formation, like the blastocyst will be there, no, that layer formation will happen. Emboly means where the gastrula goes inside like this. Like this. This is called as emboly, where the gastrula cells are moving inside. Epiboly means covering. Okay, something will be inside, covering will happen. That is the basic. Okay, very simple words. Okay. Endocrine activity we should say. Okay. Yes. The blood supply is there. No, blood supply is there. Amniotic fluid is there. Chorion is there. All those people will do. Okay, Sabrish. Okay, now students, look here. Let us remember that HCG and HPL relaxin are produced in the woman. And also it is happening in the very initial stage. No, Sabrish, it's not like happening in the later stage. It's happening in the very initial stage. So, in addition, during pregnancy, the level of hormones will increased by several folds in maternal blood. Increased production of these hormones are essential for metabolic changes in the mother and the maintenance of the pregnancy. All these hormones, okay. These are the germ layers, ectoderm, endoderm and mesoderm. They will help in the tissue development and organ development and all of that, okay. Students, trimesters. Okay, pregnancy gestation period வந்து 9 months இருக்கும். இந்த gestation period of 9 months ல என்னெல்லாம் நடக்கும்? First month of pregnancy ல வந்து the embryo's heart will be formed. First sign of growing fetus may be noticed by listening to the heart sound. நம்ம வந்து இந்த moviesல எல்லா students அந்த scan, okay, scan, yes, scan இருக்கும்ல, அந்த scanல they say no, see, can you hear the heartbeat? So that is what is happening in the first month of the pregnancy. Okay, adukapram students, by the end of the second month of pregnancy the fetus will start having limbs and digits. Okay, then by the end of 12th week, first trimester is first to 3 months. Okay, இதுதான் நம்ம most risky period னு சொல்லுவாங்க. first to 3 monthsல நம்ம வந்து pregnancy announce பண்ண கூடாது. அந்த டைம்ல நம்ம வந்து கொஞ்சம் கான்ஷியஸா இருக்கணும், because அந்த first 3 monthsல baby development வந்து கொஞ்சம் difficult-ஆ தான் இருக்கும். அதனால we have to make sure that in the first 3 months we are taking an extreme care. Then second and third trimester will usually be smooth. Okay, by the end of second weeks, that is the first trimester, most of the organ development will happen. That is why it is the riskiest period, because organ, heart, digits development has to happen. Other couple of students, for example, limbs, external genitalia are well developed. The first movement of the fetus and appearance of hair and the head will usually be observed in the 5th month. So during the second trimester, hair and movement, all that, like hair and appearance of the hair will happen. And then have you heard these people will say kicking, okay, like kicking and all that, they say, no, that one. Now by the end of the 24th week, that is the end of the second trimester, the body will be covered with fine hair, eyelids and eyelashes. Please remember that this entire thing, no, can come as match the following. Okay, it can come as match the following or match the column. So make sure that you are writing and studying it or revising it multiple times. Okay, you are revising it multiple times. Okay, now how does the woman get to know that it is time for delivery, or how does the doctor tell a certain date? Okay, and then the delivery will happen. Look here, the average duration of human pregnancy is around 9 months, okay, which is called a gestation period. Vigorous contraction of the uterus at the end of the pregnancy will cause a expulsion or the delivery of the baby, which is your fetal ejection impulse. The baby will tell, I am ready to come out, please take me out. Okay, I am ready to come out, please take me out. Okay, now here this process of delivery of fetus is called as parturition. Parturition is induced by complex neuroendocrine mechanism. The signals of parturition originates from a fully developed fetus. This is important. Who is giving the signal? Fully developed fetus, and the placenta is giving the signal telling that it is ready, I want to come out. Okay, I want to come out. So that vigorous contraction will happen, pain will happen, delivery pain, labor pain, they say. Okay, this trigger release of oxytocin from the maternal pituitary. Oxytocin acts on uterine muscle and causes strong uterine contraction, which stimulates further the secretion of oxytocin. The stimulatory reflex between the uterine contraction and oxytocin secretion continues, making the contraction very stronger. This leads to the expulsion of the baby through the birth canal. The placenta will also be expelled out of the uterus, not immediately, but in few time they will be expelled, and then the bleeding will start. So placenta plus fully developed baby will give the sign to the pituitary. Oxytocin will be released. More oxytocin, more uterus contractions. Okay. Oh my God. Okay. Understood? Yes or no? Next class, sir. Labor pain, water breakage. Water breakage is that amniotic fluid coming out. Ma'am, what is the use of amniotic fluid? Students, amniotic fluid is going to help in so many things. Okay, it is going to maintain that thermal condition, temperature. It will help the baby to do the basic movements. At the same time, it will also not allow, it will make sure that the baby doesn't stick to the amniotic membrane. So it helps for many things. Okay, many things. Okay. Last stage is, students, lactation. Now the mammary glands would have got activated. They will be ready for lactation and colostrum will be released. Now milk formation happens with prolactin. Milk ejection happens with oxytocin. Okay. And the colostrum will have IgA. The colostrum will have IgA, which is going to help in antibodies. Okay. Chapter is done and dusted. Now we are ready for questions. Wait, let me take you. Done, no? Nothing we missed, no? See, I completed it. I told you I'll complete it. I hope if you have any doubts, let me know. I'll go back and revise. Okay, wait, let me see. Find the questions, students. So many questions are there. 300 slides are there. All this is sexual reproduction in flowering plants. So many questions are there, but I didn't do all of it. I think this only. Start. Start. Okay, I'll sit here and I will control. Okay, I'll sit for some time. 5 hours I'm standing straight. Okay. Yeah. Thank you, Stephen. Students, if I sit here, can you see? Don't tell me I have to move. Okay, yes. Start. First question. Okay, where are we on the target? Come on, guys, very few more. Okay, just, oh wow, Kirtana already counted. So good at maths. Or are you telling the answer? I think it is the same number. Correct, Kirtana, correct. Okay, what is the answer? The testes are situated outside the abdominal cavity within a pouch called scrotum. This is necessary for maintaining the temperature. Okay, maintaining the temperature. So, answer 1, the scrotum helps in maintaining the temperature. Very good. Next question. Each seminiferous tubule is lined on its outside by spermatogonia, primary spermatocytes, Sertoli cells, both 1 and 3. Answer? Very good. Okay. It's going to be both 1 and 3. Next question. Leydig cells are present in the seminiferous tubule and secrete androgen, are present in the seminiferous tubule and help in maturation of the sperm, are present in interstitial space and secrete androgen, are present in interstitial space and helps in maturation of the sperm. What's the answer? Very good. Answer is 3. Okay, next question. Seminal plasma is rich in sucrose, calcium, certain enzyme, glucose, sodium and certain enzymes, fructose, calcium and certain enzymes, fructose, sodium and certain enzymes. Yeah, I will do it, okay? I'll do it maybe next week. Because I have to give them the questions, okay? It is an entire format. So, we cannot do it immediately. We have to make the questions and give them in advance. Again 3, ma'am. Okay, very good. Fructose, calcium and certain enzyme like inositol, etc. Next question. The spermatids are transformed into spermatozoa by the process called as. Quickly answer, others also. Everyone should involve, okay? Everyone should involve. Don't delete your answers. Okay, I saw your answers. Why are you deleting your answers? Don't delete your answers. Again 3. Why? Okay, spermiogenesis. Correct. Okay, spermiogenesis. Okay, I'll ask you one more question. Answer? No, no. Leaving of spermatozoa from Sertoli cells. Leaving of spermatozoa from Sertoli cells. Yes. What is that? Spermia latina. Yes, permeation. Very good. Okay, both you understood, no? Next question. What causes the onset of puberty in males? What causes the onset of puberty in males? What causes the onset of puberty in male? Quickly. I don't know. What do you mean by I don't know? I told you, no, it is increase of what? Secretion of GnRH. GnRH. Okay, that is what happens. That is why I taught you spermatogenesis and I taught you the hormones together, because you should understand that when puberty happens, the GnRH will be released by the hypothalamus. GnRH again, anterior pituitary, FSH, LH, that whole continuation. Okay. Okay, next question. For normal fertility, what percent of sperm in ejaculation must exhibit vigorous motility? Vigorous motility. Come on. Correctly you should answer. For normal fertility, what percent of sperm in ejaculation must exhibit vigorous motility? Yes, 3 is the correct answer. I don't know why everything 3 is the correct answer. Okay, 3 is the correct answer. Okay. Students, this is the telegram. Okay, make sure that you join. Okay, we need Tamil Nadu telegram. Link is in the description box. Notes also will be in the description box after the class. Okay, since tomorrow is Sunday, maybe notes will be uploaded by Monday. Okay, don't worry. This notes already you have. You have this notes, okay? And also if you want to join my telegram, it's Gopika Biology. You can also follow me on Instagram as well. Okay, yes. Now we will go back. Okay, now we will do reproductive health and then we will be done. See, today you will feel an accomplishment, okay, which you would have not felt if you were sat at home. Okay, Chuma, if you were sitting at home and missing class, you would have not felt it. Today you will feel that yes, I have done something in life today. Okay, finally I finished the first 3 chapters that I have to only see later. Okay, you will only have to see it later. You will not have to see it again. Okay, let's start reproductive health. Okay. Okay, now look here students, WHO has given a definition about health, telling that health is actually, students, can I just fill water and come? Can you wait for 1 minute? Okay. So scared. What happened? Students, just give me 1 minute. I will fill water and come, but I cannot find my one sandal. Okay, mission. Yes, mission successful. Yes, today's comment shall be that. Mission successful. How is that? Oh, no, no. Mission accomplished. Okay, mission accomplished will be the comments today. Okay, every other student who has not attended the class should be like, what is happening? So we will put mission accomplished. Okay, mission accomplished. Hashtag. Okay, mission accomplished. Okay, not now da, in the comments da. You people are then. Okay, I'll get water and come. Wait, don't go anywhere. We are going to do reproductive health. Hello, I'm back. So fast when I tell you people will message, no? So fast people will message, no? Hi, bye, how are you? Did you eat? How was current and my husband and wife and everything? Okay, look here. WHO, according to WHO, the well-being or when I say that a person is healthy, WHO is telling that person should be socially, physically, emotionally and behavioral healthy and also reproductively active. Okay. RCH full form on the students, Reproductive and Child Healthcare Program. This was an awareness program which was done with the help of media, media, speech and all of that. Okay, RCH, and this RCH mainly was focusing on family planning. It happened in 1951. Okay, 1951. Clear? Yes or no? I'll only teach you the important points. Okay, wait. Here, okay. Next one is one important topic here is amniocentesis. Yeah, ma'am, in the amniocentesis. Now, amniotic fluid we studied, no students. And the amniotic fluid, if we take it out, we will be able to understand the chromosomes. Okay, chromosomes. Sex determination can be done. With that, we can also understand if the child has any genetic disorders, Down syndrome, Klinefelter syndrome, like that. Certain disorders we can understand. So, on the disorders, the identify பண்றதுக்கு தான் amniocentesis இருக்கு. ஆனா people have misused amniocentesis for sex determination. அதனால sex determination அந்த கான்செப்ட் வந்து completely ban ஆச்சு இந்தியால. amniocentesis இப்பவுமே நடக்கும், but that is only for genetic disorder, to identify the genetic disorder. Okay, so amniocentesis, the amniotic fluid will be taken and that will do sex determination. It is banned in India. Genetic disorder now also you can utilize to identify. Okay, now population rate, students, in 1900 la the population rate was 2 billion. 2000 la it was 6 billion. Okay, so in India what happened, 1900 la 350 million irunducha, 2000 la 1 billion in May, and 2011 it was around 1.2 billion. So basically out of every 6 people in the world, the 6th person is going to be Indian. Okay, yes, identify the chromosomal pattern and then we would understand. Okay, yes, okay. Now look here. Yeah, so they are saying out of the 6 people, the 6th person is always an Indian, because look at this, students. 2000 la 6 billion was the whole world population. Nama India was the top most. Nama India was the 1 billion. India was 1 billion. It's like 6 billion. No, I am 1 billion. What is the big problem? Okay, so adanale people told that out of the 6 people, the 6th person will be an Indian. Why did this increase happen? Because of better healthcare, decrease in MMR and IMR, maternal mortality rate and infant mortality rate, more people in the reproducible age. These were the 3 reasons why the population kept drastically increasing. Now, next one, contraceptive methods. Yeah, ma'am, in the contraceptive methods. Now, contraceptive methods, students, the pregnancy avoid pandradhukku, we can use certain contraceptive methods. Contraceptive methods are divided into mainly 5 categories: natural or traditional methods, your barrier methods, then you have pills, implants, then oral contraceptives, injectables, sterilization. Okay, these are the main technique, thing. Natural or traditional method, barrier method, IUD, intrauterine devices, oral contraceptive pills, injectables, implants, and sterilization and surgical method. So, in the 6 methods are the main. Now, in a contraceptive method, let's say you, no, you cannot be a contraceptive method. I was basically trying to say, let's say this substance, okay, this substance wants to become a contraceptive. Okay, then what are the things that they have to follow? First of all, easily accessible. Everyone should be able to purchase it. Second thing, it should not affect the sexual desire of the person or whoever is wearing it. Third thing, it should be effective. It should be able to protect from the unwanted pregnancy. Fourth thing, reversibility. After using the contraceptive also, the person should be able to reverse it. That means the person wants to conceive anytime, any point of time, just by stopping the contraceptive, the person should be able to conceive. And last one, students, user friendly. Last one vandachu, user friendly. Okay, va? Okay, understood? So these are the main things that a contraceptive should keep in mind. I mean, contraceptive, no, people who make the contraceptive. Now look here. Only GM Express we are going through, okay, because I have written everything. Okay. Now, natural or traditional method, you have three types: periodic abstinences, withdrawal or coitus interruptus, lactational, amenorrhea. Periodic abstinences, now students, 10 to 17th day, nama menstrual cycle, now only we studied, no, first to 5 days, 10 to 17 days, and the time la fertile window will be open, okay, that is 10 to 17 days, you have to abstain from coitus, because that is the time where the fertile window, it is where the fertile window, biodegradable, let's not count it. It's not counted. Okay, look here. 10 to 70 days, 10 to 17 days, the couple has to abstain from coitus. That is called as periodic abstinences. That means during the fertile window, avaru the sexual intercourse perform panna kudadu. Adukapram students, withdrawal or coitus interruptus between the sexual intercourse just before ejaculation, penis withdraw panna process withdrawal or coitus interruptus we will call it, where the male partner will withdraw the penis before ejaculation. Adukapram students, lactational amenorrhea. In the lactational amenorrhea, na, when the woman is lactating milk, when it is feeding the baby, and the first 6 months there is no chances for ovulation. So on the time it is very safe to do sexual intercourse. Why so no ovulation? Because prolactin will be extremely high, because of which FSH and your LH will be very low. Because of that, ovulation will not happen. Okay? Clear? Yes or no? Understood? Okay. Now look here. Lactational amenorrhea, prolactin will be high, FSN and LH will be low, because of which no ovulation will happen. So during peak breastfeeding, no periods also will happen. Okay? Yes? Understood that? Okay, that is the three methods. Then students, barrier method, you have male condoms and female condoms. Male condoms are the most widely used one. Female condoms are comparatively less, preventing the physical meeting of sperm and ovum. So, male condom has to be worn on the penis, and female condom has to be worn near the vulva and the cervix. Okay, and now the male condom, students, the most common one is Nirod. Okay, the most common brand is Nirod. Male condom, the most common brand is Nirod. And students, in the female condom, the diaphragm, walls, cervical caps, idellame additional which is made up of rubber, and they can cover the cervix. Then we have spermicidal creams, jellies and foam, which will increase the efficiency of these people. Okay? Clear or not? 4 more likes, sir. Awesome, awesome, awesome. 1 second, students. Let me just... Okay, yeah. Okay, yes. So here, male condom, Nirod, diaphragms, walls and cervical caps, which are made up of rubber, and they are also going to cover the cervix. Spermicidal creams, jellies and foams are used to increase the efficiency. Students, the condoms or the barriers are not just used for preventing the physical meeting of the ovum and sperm. It also prevents from STD, sexually transmitted diseases. Okay, sexually transmitted diseases. Okay, next one, intrauterine devices. Intrauterine devices, 3 categories are there: non-mediated, copper releasing and hormonal IUDs. Non-mediated, na students, Lippis loop, that means which does not have any hormonal or chemical, okay, this one. And then students, copper releasing IUDs, na CUT, CU7, multi-load 375. Did we achieve? Wow, that's awesome. Okay, Lippis loop, copper releasing IUD na CU7, CUT and multi-load 375. Hormonal IUD na progesterone and LNG 20. Progesterone and LNG 20. Okay. Now, this IUDs, how are they going to help students? This copper releasing IUD will do phagocytosis of the sperm. They will reduce the fertilizing capacity of the sperm, and they will reduce the sperm motility. In the hormonal releasing IUDs, it will make the uterus unsuitable for implantation. It will make the cervix hostile for the sperm. It will not allow the sperm to happily survive there. It will be like, hey bro, go out. Okay, adu madri. Okay, understood. So this is one of the most widely accepted method, and it also helps for like most of the women will use this when they want to have a gap between the first child and the second child. Is that understood? Is that understood? Okay, now students, the tablets or the pills that you have to take. Those are called as pills. So how do we have to take? So in the first 5 days your menstruation will happen. So any of the one day you can have it, and then you will take it for 21 days, and then you will take a 7 days break. Okay, like that. So your pills have to be taken daily for the period of 21 days, starting preferably within the first 5 days of the menstruation. After a gap of 7 days where your next menstruation cycle will happen, then it has to be repeated in the same pattern. This way ovulation can be ovulation and implantation will not happen. That is what is the plan. Ovulation and implantation will be inhibited, and they will also alter the cervical mucus, which will not allow the sperm to happily survive. Okay, happily survive. Clear? Yes or no? No. Pain is not there. It is not. But the only thing is, IUDs have to be inserted by nurses or doctors who are experts in it. It cannot be done at home. But barrier methods can be utilized by just reading the instructions. So, adanala IUDs you have to go to a nurse or a expert to fit in the IUDs. Okay. Saheli students, it's an oral contraceptive. It's a non-steroidal oral contraceptive which was found in your CDRI, Central Drug Research Institute. Central Drug Research Institute, it was found. Okay. And the Central Drug Research Institute found this Saheli. Edukku ma'am இந்த Saheli special இருக்கு because அது non-steroidal and it is a once in a week pill because அந்த pills 21 days continuous எடுக்கணும். ஆனா இது once in a week pill, so அதனால Saheli became very famous and it is very affordable. ஒரு strip 24 rupees அவ்வளவுதான். Okay, 303. Awesome. Kirtana is keeping a track. Then Ragis is keeping a track. Okay, Deepak is keeping a track. Very nice. Okay, Saheli, see why students it became famous. Non-steroidal, less side effect, affordable, once a week pill, and it was found in CDRI, Lucknow. Okay, next is sterilization. Sterilization students, two types is there: vasectomy and tubectomy. Vasectomy na where you cut the vas deferens and tie it. Tubectomy na where you cut the fallopian tube and tie it. Yes, obviously the secondary oocyte cannot travel like this, so obviously fertilization will not happen. Okay, but menstruation, everything will happen by itself. Okay, menstruation will happen by itself. Students, tomorrow I don't think my throat will be, there. Okay, next is MTP, medical termination of pregnancy. The medical termination of pregnancy, students get it confused with contraceptives. Students, medical termination of pregnancy means pregnancy has happened. Adukapra namakku abort pananam. Contraceptive methods na pregnancy hasn't happened. For pregnancy to be avoided, we are using that. Here pregnancy has happened and then we are going for abortion. Okay. Thank you so much. Hepzibah. Okay, yes. Copper, side effects are not there. Side effects can be there, least side effects, but not like very extreme. Okay, not very extreme. Medical termination of pregnancy, students, unwanted pregnancy has to, when do we do medical termination of pregnancy? When it is an unwanted pregnancy. Second one, the students, when a rape has happened within the 72 hours, we can give pills. Adukapram students, mother or the fetal health has any problem. That is, mother is sick or the fetal, we feel like the baby will not come out healthy. And the time abortion pannalam, but one thing is, it is safe only during the first trimester. First 3 months la da idu safe irukka. Adukapram second trimester, third trimester la romba risky irukum. So adanal doctors recommend pannada. So MTP usually first trimester la da pannanum. Okay, 45 to 50 million MTPs per year out of 1/5 of the total pregnancy. Okay, even though MTP is actually not legalized in many places, still people do it. People do it with unqualified quacks. That means people who put the small clinic, they'll give the pill and they'll be like, okay, abortion will happen, periods will come after that. But what is the problem is, sometimes the cells, the complete cells have to go. No, students, implantation would have happened. So that shedding has to happen properly. If the shedding doesn't happen, some cells are there, then it can form cyst later. Okay, so adu madri problems irukum. Okay, next is art technology. Okay, art technology we have in vitro, in vivo. In vitro na students, it happens in the lab. Under in vitro you have your test tube baby, and in that you have to remember embryo transfer. Embryo transfer, students, 2 scenarios it will happen. So what you will do, you will take the sperm, you will take the ovum, you will fertilize it in the lab. Okay, in a petri plate, proper system la nama adha use pananam. Sperm and ovum will be fertilized. Adukapra nama and the embryo transfer pananam. So embryo can be transferred. If it is below 8 blastomere stage, we will do ZIFT. That is zygote intra-fallopian transfer. That means we will transfer the zygote into the fallopian tube, and then the female will continue the entire process. Zygote and morula, blastula stage, and then implantation. But Anna, if it is above 8 blastomia stage, then we will do IUT, intrauterine transfer, where directly it has to go to the uterus or the womb. Okay, intrauterine transfer. Adhikapra students, in vivo na it will happen in the female. Okay, inside the female body. Okay, inside the female body. Now, gift students, gift is gamete intrafallopian transfer, where I am transferring the gamete into the fallopian tube because maybe because of any XYZ reason, the gamete is not able to reach that. Okay, that is ovum can be taken from one person and then it will be transferred to another person who can only provide a womb but cannot form the ovum. Surrogacy and all we do, no, like that. Now here you can see intracytoplasmic sperm injection. Intracytoplasmic sperm injection na, and the sperm and the cytoplasm of the ovum la inject panita fertilization ka possibility we are making. Why? Because maybe because the sperm count is very low, oligospermia would be there, or like the sperms are not able to reach, not able to survive. So adanala what we do, intracytoplasmic sperm injection. Then students, we can also do artificial insemination, where the sperm will be directly injected into the vagina. This mainly happens when there is a low sperm count. Okay, now when do a person do ART technology? Couples who have been trying for past 2 years but they are not able to conceive, that time we can go for ART technology. Okay, okay, clear? My God. No ovum, it will just degrade. Okay. If the fallopian tube is cut and tied, then how will the ovum go, ma'am? Okay. As I told you, the ovum will not go, so obviously fertilization will not happen. The shedding will happen other ways also. Okay. Ovum will be released by the ovary. Okay. Ovum will be released by the ovary, but it will not enter into the fallopian tube. Clear? But students, see, these are all cells. No, they will be able to degenerate. It's not like it has to go to the fallopian tube only. Why it is going to the fallopian tube? Because if it goes to the fallopian tube only, it can go to the womb and implantation can happen. So, adanale, ovum will get degraded by itself in that pelvic cavity. Just that the ovum will not reach the fallopian tube or the ampulla. Okay? Oh, you are explaining to her. Okay. So, students, here it will inject the sperm. Okay? That is the whole chapter. That's all. Here I have just added diagrams. I told you everything's detailed lecture is done on the channel. So, if you want to check detailed lecture, you can do. Okay, answer the question. Answer the question. We are done with this chapter. I think we did it in 20 minutes. 20 or 25 minutes. Actually, technically, this chapter is only for that much if it is done just like a revision. Statutory ban on amniocentesis in India was necessary because it was very expensive. Tell students. Thank you, Anusha. Thank you so much. Guys, without fail, comment and let me know, okay? Because, see, you people will tell this and you will go. But for me to always see it, I want to see that comments, okay? It makes me more motivated, okay? Thank you, Anusha. Thank you so much, dear. Students, we, like, you know, we online teachers don't get enough love. Even though, you know, we feel like, okay, there is love, but then this, like, chromosome, it is abnormality. Oh, I'm so sorry. I did not see. Now I'm seeing. Abnormality. Okay. Abnormality. Okay, abnormality. Chromosomal abnormality. Okay, now you can answer. It is an invasive procedure, carries high risk. It can be used to prenatal sexual fetal. Okay, yes. Yeah, so what I was telling, online teachers, actually students, lot of comments will come, but somewhere down the lane, no, you people will not connect to us, or maybe you will connect to us, but it is always easier to be replaced. There are a lot of teachers. Some teachers will be very nice. Some teachers will be in your language, they'll be talking. Some teachers will be very close, or I don't know, like different aspects, and you will suddenly forget about us and you will go to them. So for online teachers, it's like if we get a comment, no, we will always look into that comment. We will like the comment multiple times. So and the madri or feeling. But offline teachers, see, even when I was in offline, I used to, for me it was a very normal thing to get compliments, very normal thing to, you know, students coming, running and hugging me. All that used to be very normal. But for me now, like, you know, it feels like, for me, I want to see how much students are, you know, interested in the class. Okay. No. They're asking why it is banned. Okay. Why it is banned? It is an invasive procedure. No. There's nothing like that. It's not an invasive procedure. It's a small a procedure. So there's no, the answer is going to be option number 4 only. Okay. Option number 4. 𝐂𝐸 is an NGO working for female child health, midwife working in villages to help in child immunization, a national helpline number for prevention of crimes against women, oral contraceptives for females. Okay, I should have the next VIX, students. My voice, I told you last time, no, my voice already, like, I had a throat problem. So after that, I think after a very long time, I'm taking a very long lecture. Don't worry, in NEET time, no, we will take marathons, like big marathons. Okay, we'll take 8 hours, 9 hours marathon. So don't worry. Thank you, Anna, okay? Anna Purani. Okay, correct, no? Purnima, I know. Anna Purnima, I know. Your parents all keep such fancy names for all of you. Okay? Yeah, full throat kitch is coming. 𝐂𝐸 is an NGO working for female midwife. No, it is an oral contraceptive for females. Okay. The ideal contraceptive for females who want to delay pregnancy or space children. Yes, I just told you. Space children, what is the technique that they will use? Come on. Fast. Thank you, Prajit. Thank you so much. Anna Purani. Okay, Anna Purani. Yeah. Me first, it seems. Okay, very good. The answer is going to be IUD. Oh, there's no answer, students. I am only the answer. Okay, I mean, I am only the Kaun Banega Crorepati. Okay, in test tube baby procedure, fertilization is in vitro, but embryo development is in vivo. Fertilization is in vivo, but embryo development is in vitro. Tell me, in test tube baby, what will happen? Test tube baby. Not necessarily every time. Both will be in vitro. Okay, it's not necessary. What is the comment for the day today? Mission accomplished. Okay, mission accomplished. Mission accomplished, and then you should tell the class. Okay, are you feeling confident? Are you feeling like not so confident? No, you shouldn't say that. How can you say that? Okay. Why da mix answers you're giving? Huh? 2, 1, 3, 4. Everything you people are telling. This will be what? Fertilization will be in vitro, but embryo development will be in vivo. Okay. Now, this we have done. Okay, students, that's it. Okay. If you want to do some SRIF questions, I can make you do. Okay, otherwise, that's it. Pollen grains lose their viability within 30 minutes of their release in. Thank you, Honeybee Gift. What names you guys have? Yeah, crazy names you guys have. Even I want to keep a name like this. Okay, pollen grains lose their viability within 30 minutes of their release in what? Yes, very good. Okay. So, students, we are done and dusted. Okay, I think I finished it before 6 hours. Okay, so we have how many likes? Let's see. I think we would have hit 3. Oh, that is awesome. That is awesome. Okay. Makes me so happy. So students, I will just guide you through the channel. Okay. First of all, see, this is the playlist. In the playlist, the students, here you can see the one shots. Okay, one shot. Need to the you. Yeah, 1 second. I can't see your people's reaction. Now I can see. So here you can see. Okay, here you can see all the one shots, NEET 2026 one shots. And then students, in video session, if you go, you can see, okay, so many things. So many things like body fluids and circulation, 99% students, hidden lines. Please watch this, okay, hidden lines, because so many hidden lines are there actually. And then, this also, question pattern, you can check out, okay. And then students, I want to tell you people one thing. See here, 150 plan, no, Verithanam plan, I have given 500 question PDF. See, give free giveaway. Okay, this 500 PDF, please take it and every day practice questions, because it is very difficult for you people to go and buy books. So if you don't want to just go and buy books, no, make sure that you are taking this 500 PYQs and you're solving. And this notes will be in the description box. You already have the notes, but still, if you want a combined notes, I will share it on description box. Okay, by Monday it should be done, because tomorrow is Sunday. I cannot ask my team members to work. And students, very, very importantly, what you have to do today, now only go here. Okay, go to the live. Okay, in the live will poi here. I want the attendance on this live. Okay, see, only 22 likes. What is this? Minimum 100 should be there. No, we are not a small channel anymore, right? We are a big channel. So students, in the short session, see, day 2, day 3, day 4, day 1. And you will not believe how many comments I have got. 125 comments. 125 students have answered this question. Okay. And that's why I'm telling you this is very important, because everyone wants that free gift. Okay, everyone wants that free gift. So even I want you people also to take the free gift. So go and smash the like button. And today itself, in the comment section, I want to see mission accomplished and what is your feedback on the class. How do you feel? Did you feel like, okay, it was a 6 hours class? Because I did not feel it was a 6 hours class. Only my throat felt it was a 6 hours class. But I felt it was like 3 hours class, because you people had so much of energy. I also had so much of energy. So we will do more videos like this, more sessions like this. Don't worry. We will also do longer session where I'll complete the zoology and botany, but that will take little more longer, because now itself I do not do it. Like this, I will do unit wise, so your backlogs can be covered. Okay, Vakana? Okay? Everything is okay? Okay, I can see your comments here. Safe score, medical call. Ma'am, please say safe score. Sure. Thank you so much. Thank you so much, guys. Okay. Yes, I will definitely drink hot water. Yes, honeybee gift. Okay, success panna mudiyum. Why you can't? Okay. Ma'am, watch sir's lecture. Sure. Okay. You guys are so funny. Okay. Yes. So thank you so much, Kanna. See you all in the next class. I hope all of you enjoyed. Please let me know in the comments. Take your 10 seconds of the time and let me know in the comments. And make sure that you're subscribing to the channel. Every subscriber that comes on this channel is so special for us. It is very, very special for us. And we will make sure to take care of each one of you. Because now I am that, like a mentor with 11,000 future doctors in my in my hand. So it feels very special. So I don't know how you people will feel it, but I feel it very special, because I started from zero. So and the ella step ella subscriber ella students romba important da enikku. So it is not like anyone else. It's very, very special for me. So thank you so much, students. Okay, thank you so much. I know you guys are with me. So thank you so much for all the love, all the wishes, and all the how much you took care of me during this live. It is very, very, very precious for me. So thank you so much. See you all in the next live, Kanna. Till then, take care. Bye-bye. Make sure that you are learning, preparing, be motivated. Do not lose your motivation. You are all awesome people. Just make sure that you're practicing enough questions and not just getting stuck to just NCERT. Okay? Okay, this is your hard work, ma'am. Thank you so much. Thank you so much. Yes, we will celebrate. Okay, we will celebrate. I'll come to your place one time whenever you people are telling me, ma'am, come. And then we will celebrate. Okay, by then I hope we are 25k and 11k, or I don't know what numbers we will be at. But you people can share it with your friends. Talk about the channel to your 2 or 3 friends in your offline centers and try to add people. Okay, so till then, take care. Bye-bye. Yes, I know. Because of your efforts, 11K future doctors are with you, ma'am. Thank you so much, guys. Thank you so much, Kana. Okay. Yes. Yes, thank you. Yes, definitely. I'll do it, Sabrish. Safe mark. Yes, yes, I'll do it.